to take advantage of this opportunity to update in top topics in plant biochemistry and molecular biology, such as development, environmental interactions, hormonal signaling, biotechnology, metabolism, stress, and technological breakthroughs. Once again, I welcome all the attendings from different countries. I wish you to enjoy yourselves and participate with questions and comments in this great meeting. Many thanks and have a wonderful week.
Greetings. On behalf of ASPB, it is my pleasure and privilege to welcome you to the 12th SAMB ASPB Mexico Section Virtual Conference. I personally had the privilege of experiencing the positive impact of this meeting in 2019, resulting in a fruitful collaboration with my colleagues. Indeed, the increased number of participants to 1,300 scientists from 50 countries in this meeting is a testimony to the positive and constructive experience of many other participants. I have long valued the power of science without borders. The long history of conferences and collaborative efforts is the prime example of how we cross the man-made borders and transcend political agendas of some politicians. Indeed, yes, we can. I am certain that this meeting will be no exception as it continues to strengthen our scientific relationships and enhance collaborations between scientists across the globe. I wish I could be there in person. However, from afar, I wish you much success in continuing this tradition, empowering the young incoming generation of scientists, and showcasing the power of science without borders. Be powerful and have a successful meeting.
These are challenging times. The world is standing up against one of the major threats we have ever encountered. And if there is one thing we have learned so far, is that in the face of the uncertainty, science, hope, and solidarity must always prevail. We want to give you the most enthusiastic welcome to our 19th National Plant Biochemistry and Molecular Biology Congress, our 12th joint symposium between Mexico and the USA, and our second American Society of Plant Biologists Mexican section meeting.
The pandemic transformed the way we interact with each other. We have been forced to change our daily routines to incorporate video conferencing as a major way of communicating with the world, and inadvertently, we became part of the largest social experiment ever run. We are a species entirely reliant on interaction with other human beings. And although there's no substitute for human touch, video conferencing has been instrumental not only to keep us communicated, but to keep our minds at ease, knowing that we are not facing the challenge alone.
Thanks to your tremendous support, we are able to bring together more than 1,300 participants from 49 countries. We are extremely happy that most participants are undergrad and grad students, as this means that you, the next generation of plant scientists, answered the call. We are convinced that you will get inspired by the work of all the amazing lineup of speakers that honor our meeting with their participation.
In addition, we will have the participation of over 100 speakers that will share with us their latest results through pre-recorded videos in an unprecedented way. All pre-recorded presentations will be streamed during the meeting in flash talk sessions marked in the program.
The organizing committee was formed by principal investigators from the Faculty of Chemistry, the Institute of Biology, the Biotechnology Institute from the National Autonomous University of Mexico, the Advanced Genomics Unit from Langevio of the Center for Research and Advanced Studies of the National Polytechnic Institute, the Institute for Biotechnology and Applied Ecology of the Universidad Veracruzana, and Michigan State University.
We want to thank the Mexican Society of Biochemistry and the American Society of Plant Biologists for their invaluable support that made it possible to hold this meeting completely free of charge to all participants. Finally, we would like to offer our sincere thanks to all the people that helped us make this event possible. Without any further delay, let's get started.
Okay. Good morning, everybody. To start our meeting, I would like to welcome our first keynote speaker of the day. We have Rob Martinson from Cold Spring Harbor Laboratory in US. The title of his talk is Germline Reprogramming and Epigenetic Inheritance in Arabidopsis. Rob, if you are ready, could you please share your screen with us?
All right. Thank you, Mario. Good to see you.
Thank you. And I will let you know when you have 2 minutes left.
Okay. Sounds
Thank
good.
you.
Can you see that okay?
Yes.
Okay, great. Well, first of all, thanks so much for the invitation. I would love to be in Mexico in person. It's a fabulous place. I've been many times, but congratulations on being able to put together this, this meeting virtually. That's what we're all doing these days. So I'm going to talk about germline reprogramming and epigenetic inheritance. I actually always like to start my talk with this iconic portrait of Luther Burbank, who was a famous plant breeder at the turn of the 19th, 20th centuries, painted actually posthumously by his dear friend Frida Kahlo in the 1930s. Despite being a famous plant breeder, Luther Burbank did not believe in the newly emerging subject of genetics and famously said that heredity was only the sum of all past environment. Well, now, of course, we know a lot more about epigenetic inheritance, and so maybe he had something to say. But of course, in mammals, it turns out epigenetic inheritance is relatively rare. And the reason for that is reprogramming. So the genome has to be reprogrammed in order to remove and restore epigenetic marks that might have been encountered because of the environment or because of developmental changes, and restore them to allow pluripotency or totipotency in the zygote. And so in mammals, DNA methylation, for example, shown here, as well as histone modifications and histone variants are extensively remodeled twice. First, in the very early embryo, right after fertilization, when methylation is lost and then restored. And a second time in the germline, where most of the DNA methylation is lost, but not all. So there is some possibility for epigenetic inheritance in mammals. But in general, the germline and the embryo completely remodeled the genome, making epigenetic inheritance very difficult. In plants, of course, we have a great deal more epigenetic inheritance in general, and we've been studying the issue of germline reprogramming for quite some time now, mostly in Arabidopsis. In Arabidopsis, we've been focusing on pollen. This is just a fluorescent micrograph of pollen showing you the two sperm cells labeled in green. One, of course, fertilizes the embryo, the other the endosperm. And the vegetative nucleus, which acts as a nurse cell during pollen tube growth, but doesn't contribute its DNA to the next generation. And in fact, pollen is extensively remodeled. DNA methylation, as I'll show you in some detail in a moment, but also histone modifications, histone variants, and small RNAs all change extensively in the pollen and are involved in a reprogramming of the genome. As a result of this reprogramming, actually both in mammals and in plants, transposons, which are major targets of epigenetic silencing, are reactivated. And this happens in the early embryo in mammals, as well as in the germline. And in plants, we found some years ago, just looking at publicly available microarray data, that transposable elements are in fact strongly upregulated, both in the pollen and in the immature seed. And this observation was made by Keith when he was a postdoc in the lab many years ago. It turns out that these transposons are the source of small RNAs, just as they are in mammals and animals in general. And these small RNAs in pollen are triggered by a special microRNA, at least in many species of plants, certainly in Arabidopsis. This microRNA is called miR845. It comes in two forms, a 21 nucleotide and a 22 nucleotide form. And it's extremely abundant. It's by far the most abundant microRNA in pollen. And it's very specific to pollen, at least under ordinary conditions. It's not really expressed very much anywhere else in the plant. If you introduce a target site for microRNA 845 into a GFP reporter gene, it will trigger large numbers of these secondary small RNAs, 21 and 22 nucleotides in size. And in fact, it will silence GFP. I'm not showing you that data very efficiently in the pollen. Now, the important question is the targets of miR-845. And it turns out that miR-845 has the remarkable ability to target many of the transposons in the Arabidopsis genome. And the reason for this is that it actually targets the primer binding site, which is required for retrotransposon mobilization. So these are LTR retrotransposons, the most abundant type of transposon in the Arabidopsis and in all plant genomes. And just to remind you, retrotransposons are replicated using a tRNA primer that primes first-strand cDNA synthesis through the LTR at a particular site, the primer binding site, or PBS. And that first step of reverse transcription is critical for the entire replication and transposition of retrotransposable elements. What's even more remarkable is that almost all transposons in plants use the same tRNA primer. This is actually the tRNA for the initiator methionine codon. And that tRNA then, as a result, the primer binding sites of most Copia and Gypsy retrotransposons in the Arabidopsis genome are closely related. And as a result, miR-845 actually targets hundreds or even thousands of different retrotransposons in the genome. And of course, this is a fabulous strategy for silencing transposons, because the transposon can't really mutate the primer binding site to escape or to evade microRNA recognition, because by doing so, it would also mutate the primer binding site itself, which wouldn't allow replication. So this is a very powerful way to silence transposons. And remarkably, in the mouse, the same thing happens. And we showed this a few years ago now. But rather than using a microRNA in the mouse and in human, tRNA fragments themselves, which are also 22 nucleotides in size from the 3' end of the tRNA fragment, do exactly the same thing. They behave like microRNAs and silence retrotransposons by targeting the primer binding site. Okay, so what about the small RNAs that are generated in pollen? I'm not going to show you all the data again. This was published a few years ago. But in fact, a whole suite of small interfering RNAs are generated or triggered by this microRNA. We can demonstrate that by looking at a mutant, which has lost actually only one of those two forms of miR-845, but it's still enough to have a pretty dramatic effect. And as you can see, transposable element small RNAs are greatly reduced in the mutant, and they're reduced in these size classes, in the 21s, 22s, and even some 23s. In pollen rather than the 24s themselves. So it seems that it's triggering these secondary small RNAs, which are very well known in Arabidopsis. So then we have lots of these secondary small RNAs coming from transposons in pollen. And many years ago, Keith, who discovered this class of small RNAs when he was a postdoc in the lab, published a wonderful model suggesting that perhaps these small RNAs, which seem to be generated in pollen under the control of the vegetative nucleus, were transported into the sperm cells to silence any transposons that might have somehow escaped or been reprogrammed. And in fact, he was able to, in his own lab, show that very nicely in a paper that came out a couple of years ago in Nature Plants. So you can actually demonstrate this very well. Okay, well, what about reprogramming then? So we have all these small RNAs, we have transposon activation. What about DNA methylation, histones, and so on? Well, just to remind you, pollen arises from the inappropriately named pollen mother cell, which then undergoes meiosis to give you microspores, and then these give rise to the vegetative cell and the generative cell after the first mitosis, and then in a second highly stereotypical mitosis, the germ cell divides again to give you those two sperm cells. And of course, the sperm cells contribute to fertilization, whereas the companion cell does not, or at least does not contribute its DNA. Some years ago now, Joe Calaco, who was a student in the lab, and Philippe, who was a postdoc, we're able to sort both microspores and sperm cells and vegetative nuclei very efficiently and in sufficient quantities to be able to do bisulfite sequencing to look at the pollen methylome. They were able to do this in both, as I said, both in the microspore, sperm cells, and the vegetative nucleus. This was published almost 10 years ago now, but I just want to summarize the results. So just to remind you, DNA methylation in plants is almost exclusively in cytosine, 5-methylcytosine, which occurs in 3 different sequence contexts, depending on whether the adjacent nucleotide is a G. So CG methylation, and I'll go into this in a lot more detail in the next few slides, is mediated by a DNA methyltransferase with an excellent homologue in mammals called MET1, and it's replication guided. CHG methylation, where H is anything except G, CHG methylation is actually chromatin guided, and it has to be for reasons that I'll get into in a moment. And this is conditioned by a different methyltransferase called chromo methyltransferase. And finally, CHH methylation, which is completely asymmetric, doesn't have any Gs. This is mediated by a third methyltransferase, and this is guided by RNA interference, and I'll show you that in a moment. So again, just to remind you, what we found when we sequenced the methylome, I'm just blowing up one chromosome here, chromosome 1, is that CG methylation, largely speaking, is unchanged. And that's very interesting, and it probably explains why epigenetic inheritance is so much more common in plants than it is in mammals, because CG methylation is unchanged and so would be inherited, and indeed is inherited in many very well-known examples of heritable epigenetic inheritance. CHG methylation changes a little bit, and in fact, I'll show you in a moment, it does really change, especially in the sperm cells, and also to some extent in the microspore. But the most dramatic change is in CHH methylation, which essentially disappears from sperm cells and is almost completely lost in the microspore. And it turns out, with some lovely work from Xiaoqi Fang and colleagues, that it's actually lost even earlier than that in meiosis at that stage, just before the microspores are produced. Okay, so there is then a reprogramming of DNA methylation in pollen, and we can summarize it in this graph, which is sort of borrowed from that famous mammalian reprogramming graph that I showed you. CG methylation is normal in the microspore and is maintained in sperm cells and into the next generation. But CHH methylation, for example, is reduced dramatically in meiosis and in the microspore, and it stays low in the sperm cell. Interestingly, it actually gets remethylated in the vegetative nucleus, even though this doesn't contribute its DNA to the next generation. We think this has something to do with the small RNAs that are being produced in pollen, both the 21 nucleotide small RNAs, which find their way into sperm cells, and of course, the more well-known 24 nucleotide small RNAs from transposons. And we believe that this has an awful lot to do with imprinting, and we know it affects the triploid block, but I'm not going to talk about that today. In the embryo, CHH methylation comes back, and this, of course, is very reminiscent of what happens in mammals, and so we were very interested in the mechanism of this reprogramming. So just to remind you, DNA methylation during replication can be maintained if it's symmetric. That's to say, if it's in CG dinucleotides, which are found on both strands. So this C and this C are both methylated. After replication, the 2 parental strands separate, and both of them are methylated. As a result, the apparatus, the enzymatic apparatus required for DNA methylation will fill in the other strand, the senescent strand, will recognize the methylation, the hemimethylation now on the parental strand, and fill in that missing methylation. And as a result, symmetric DNA methylation is maintained during replication. Asymmetric methylation, however, for example, CA methylation, is different because there's no C on the other strand. And as a result, the parental strands separate and only one of them retains that DNA methylation. DNA replication can now fill in the other strand, but now this daughter chromatid has no DNA methylation because it has not been inherited from the parental strand. And as a result, asymmetric DNA methylation is not maintained. And so it has to be maintained independently of replication by some other guide, not by DNA methylation itself. And thanks to the work of many different labs, especially my good friend Steve Jacobson, here in this great review written with Judy Law, we now know that CHG methylation, where H again is anything but G, so A, C, or T, CHG methylation, which is not strictly symmetric, because remember that CHG will read differently on the other strand, is maintained not by DNA methylation, but by histone methylation. There's a histone methyltransferase, actually several of them in plants, that methylate K9 on histone H3. And this is recognized by that chromomethylase responsible for CHG methylation. And it's this cycle that maintains CHG methylation. CHH methylation, oops, I'm sorry about that, is maintained, as we all hopefully know by now, by RNA-dependent DNA methylation, where small RNAs, particularly of the 24 nucleotide class, but actually we think in all classes, have the capability to trigger DNA methylation as well. They're produced by special polymerases, POL4, RDRP, DICER, and Argonaut, and so on. But then importantly, they interact with the transcripts of another plant-specific polymerase, polymerase 5, which then involves two very interesting histone lysine 9 methyltransferase homologs that were thought until very recently to be inactive, and yet somehow are required for DNA methylation. So it's a complex process, RNA-dependent DNA methylation, but a very important one, and one that I'll be talking about more in a minute. Okay, so in order to look at the inheritance of DNA methylation during DNA replication in replicating cells, we've been collaborating with Bill Thompson and Linda Hadley-Bodone now for many, many years at North Carolina State University. And they have a Arabidopsis cell culture system shown here, which is completely diploid. We've actually sequenced it many times. It doesn't have any obvious epigenetic mutations, or none that affect what I'm about to talk about. And you can grow it in culture essentially forever. And in fact, it has been in culture for probably for decades at this point. It incorporates BRDU very nicely, which is, of course, the nucleotide mimic that allows you to follow DNA replication. So what we do is we pulse for 24 hours those cells with BRDU, and then, or EDU actually these days, and then measure by FACS nucleotide incorporation against DNA content. And this allows us to separate nuclei that are in different phases of the cell cycle, G1, S, and G2. The S phase cells can be further separated, as I'll show you in a moment, into early, mid, and late S phase according to DNA content. And that reflects the differential DNA replication of different parts of the genome, because euchromatin is replicated early and heterochromatin transposable elements are replicated late. When we do the methylomes then, so we have enough nuclei to be able to do whole genome bisulfite sequencing on those methylomes, we notice the following things. First of all, CG methylation, despite being in culture for 30 years, CG methylation was largely maintained in these G1 cells. CHG methylation was lower, but it was still there. But CHH methylation had completely disappeared. So in that way, it sort of reminded us of pollen, as if somehow there's some sort of reprogramming event was associated with just a lot of cell division. We could look much more specifically at strand-specific methylation using a technique that was pioneered by Karin Schneeberger many years ago now, which looks at strand asymmetry in DNA methylation. So you take the entire methylome and you plot the methyl C level at each dinucleotide, CG, for example, on one strand, on the Watson strand, and on the other strand, the Crick strand. And you plot that as a scatter plot. If, as is the case in G1, CG dinucleotides are largely symmetric, then they will cluster in this heat map at the top right-hand corner. And this is because they're 100% methylated on each strand. If, however, they're asymmetrically methylated, then you get this much broader distribution showing asymmetry. And this is exactly what we see in S-phase nuclei isolated from those cultured cells. CHG methylation, on the other hand, is always asymmetric, as if it never gets fully symmetrically methylated, which of course is not really a tenable situation in the very long term, and it's lost an awful lot of CHG methylation. We can even separate into early, mid, and late S-phase using this technique, and what we found is that most of this asymmetry in CG methylation is in late S-phase, and that makes sense because the transposons and heterochromatin, where most of the methylation resides, replicates late, as it does in animals. Okay, so now armed with this technique, we can now look in other tissues of the plants, thanks to our own methylome data, but also methylome data that's been published from many other labs working in Arabidopsis. And what we found was that CG methylation is symmetric in most of the tissues and stages that we looked at. What we're looking at here, of course, is pollen. We're looking at myocytes, microspores, sperm cells, and the vegetative nucleus. But interestingly, CHG methylation was much more variable. We found in leaf and actually in almost all tissues that we've looked at, and we've looked at a lot of published methylomes, it's almost always asymmetric, as it is in tissue culture. Oh no, I think I'm losing my cursor. It's not good. Yeah. Sorry about this. It's really annoying. I thought I was doing okay. I'm going to have to abandon the PowerPoint and come back to it. Sorry about this. It's because the bandwidth isn't big enough. It shouldn't take too long. I guess we're all getting better at doing this now. All right. So, as I was saying, CHG methylation is asymmetric in almost all tissues that we looked at. But importantly and very interestingly, there were exceptions in pollen. So myocytes, shown here on the right, have fully symmetric CHG methylation, as if during meiosis, CHG methylation is restored, it's reprogrammed in an important way. And in the microspore, similarly, it retains almost all of its symmetry. But in sperm cells, it's gone again. And sperm cells are actually arrested in S phase, so that sort of makes sense. But the CHG methylation is now asymmetric again. So it's been reprogrammed in meiosis, just like in mammals, but then gets removed. In the embryo, interestingly, it comes back. So in the embryo, both CG and CHG methylation are completely symmetric once again. This is mature embryos taken from Arabidopsis. In contrast, endosperm, which of course has a rapid rate of cell division right after fertilization, is completely asymmetric, both in CG and CHG. So this is really rapidly replicating and doesn't really care anymore about its DNA methylation. Of course, the endosperm is not going to contribute to the next generation either. Okay, so this is very interesting. It's clear that cell culture, rapidly dividing cells, can give us a very powerful model for DNA methylation, epigenetic inheritance. And then by looking at all these other tissues, we had a good idea of how this worked. And so we were able, this was published just very recently, in the cell cycle. Then we think CG methylation is restored during S phase. CHG methylation we think is restored in G2, and that's because the microspore is actually arrested in G2. CHH methylation, on the other hand, may require that the cell cycle arrest altogether. We can't eliminate that possibility. It's our favorite theory, and certainly is much later than CHG methylation in the cell cycle. All right, so then we were interested then in how this DNA methylation gets restored in the embryo, given that it's lost from the sperm cell. Oh, sorry, and before I do that, what about small RNAs? Well, these tissue culture cells have very interesting small RNA patterns. Very much like pollen, a large number of transposable elements, including Attila elements shown here, actually accumulate not 24s, but 21 nucleotide small RNAs. And they do that in a cell cycle specific way, so that cells enriched in S phase have a lot more 21 nucleotide small RNAs than other cells. So there's something very important about that 21 nucleotide small RNA class that is also found in these rapidly dividing cell cultures. Okay, so what happens in the embryo? Well, recently, Jean-Sébastien Perron, working actually with Daniel Griminelli, who was on sabbatical in the lab at the time, looked in early embryos at DNA methylation patterns and looked in a variety of mutants, in RNAi mutants, mutants involved in RNA-dependent DNA methylation, and in mutants in that histone methyltransferase required for CHG methylation. And again, this was published very recently, but what we found was that there were two classes of transposable elements in the Arabidopsis genome. DNA class transposons and also DNA and some line retrotransposons are actually very sensitive to RNA-dependent DNA methylation, whereas other types of retrotransposons, the gypsies and copias, were much less sensitive to RNA-directed DNA methylation and instead depended on this histone methyltransferase. And that's shown here, looking at different levels. I'm sorry if the writing is a bit small, but CHG methylation shown here in blue is dramatically reduced in these DNA transposons in the RNA-dependent DNA methylation mutants. And whereas the converse is true for this other class of transposons shown here. So they're quite radically different. And we wondered then, given the dependence on K9 methylation, what happened to K9 methylation? And Jean-Sébastien and John Kahn in the lab were able to do ChIP-seq, looking at K9 methylation in very early embryos. And what they found was, sure enough, K9 methylation mimicked the CHG methylation that I just showed you. So it's reduced in the RNAi-dependent transposons and in RNAi mutants, but it's not reduced in those transposons in the canonical K9 methyltransferase mutants, which were thought previously to completely erase all K9 methylation. And in fact, they don't, because these RNAi-dependent ones are still maintained. And so we were very interested to find out how K9 methylation was maintained in this way in the embryo. Daniel Griminelli produced these beautiful micrographs looking at very early 8-cell embryos by immunofluorescence using an antibody to histone H3 lysine 9 dimethylation. And what he found was in wild type at the 8-cell stage, there's lots and lots of K9 methylation. But in mutants, for example, in Argonaut 469 triple mutants shown on the very right-hand side, that K9 methylation is not there in the early embryo. Similarly, in double mutants for the SUVH29 factors that I told you about earlier, which are required for RNA-dependent DNA methylation, but thought to be inactive as histone methyltransferases, they also lost K9 methylation at this early stage. I should say it comes back later. So it's only at this very early stage of reprogramming that these mutants seem to be important. And indeed, the Argonauts that I just described, AGO6 and AGO9, are expressed at the very early stages in embryos, just shown here. So this is just using mCherry AGO9 and AGO6 labeled Argonauts. So it makes sense that they would be involved in these stages. AGO4 is actually a bit later on. It's really in later embryos than 6 and 9. We were then very curious as to whether SUVH9 might be the K9 methyltransferase, even though it was thought to be inactive, responsible for this phenomenon. And so Jean-Sébastien, first of all, did some complicated crosses shown here to eliminate the function of all of the K9 methyltransferases that we know about. And when you do that, you get a very specific phenotype in Arabidopsis, these very curled leaves. It's sometimes called the STC phenotype. We know the gene that's responsible. And what he found was that he got that phenotype very strongly when he knocked them all out. But if he replaced SUVH9 wild type copy into that background, then the phenotype was rescued as expected. But then he made a mutant in SUVH9 in the catalytic domain, for the set domain for the K9 methyltransferase. And even though the protein is expressed at normal levels, shown in part A here, it completely failed to restore the phenotype. And sure enough, ChIP-seq revealed that it also failed to restore K9 methylation to those specific subset of transposons that require the RNAi-directed pathway to get K9. So in fact, SUVH9 must be functional, which was quite a surprise. And again, this was published just recently. All right, well, what effect does all of this have in the real world? So of course, most plants don't propagate themselves by cell culture. But in fact, in agriculture, of course, many plants are propagated by micropropagation. And a famous, maybe infamous example is the oil palm, which has really been subjected to relatively limited breeding because of its long breeding cycle. And as a result, cloning of elite palms that were known to have very high yields, these are hybrid palms, could be accomplished by simply taking like the heart of palm, those spear leaves, from a mature palm tree, putting them into culture, and then regenerating something like 10 to 50,000 genetically identical oil palms that in theory would all have that same very high yield as the original palm. But maybe it was the ghost of Luther Burbank, but in fact, it turns out that's not always the case. Although tissue culture labs in Malaysia and Indonesia are getting much better at this, there is still a real problem, which is that many of the palms, only when they're mature, so they're already occupying precious land in these oil palm plantations, turn out to have this very ugly and very, very low yielding phenotype called mantled. This is a parthenogenic fruit, usually without a seed, usually female sterile, and it also has these homeotic transformations and very little oil. In fact, those homeotic transformations are transformations of stamens into carpels, the very well-known B class of homeotic mutants in plants. And indeed, the phenotype looks an awful lot like deficiens in Antirrhinum or AP3 in Arabidopsis if you look at it in the right way. So you have all these transformations of stamens into carpels. And so deficiens was always a strong candidate gene from what might be going on, but no one could ever detect a mutation in deficiens. So I should say, full disclosure, this is work that was done with a company, Orion Genomics, of which I'm a founder, in collaboration with the Malaysian government, the Malaysian Oil Palm Board, who collected material from hundreds or even thousands of these mantled palms from all over Malaysia. And we subjected them to DNA methylation microarray analysis because it's much cheaper when you have thousands of samples than doing bisulfite sequencing. The material we used were trios where we had a mother palm called an autet and genetically identical clones called ramets, so either normal or mantled ramets. And then we looked for methylation, DNA methylation genome-wide, and looked for those regions of the genome that were always methylated in normal ramets and never methylated in mantled ramets. And amazingly enough, there was just three or four features on the microarray where they all clustered on exactly the same place that fulfilled that criteria out of many, many hundred mantled palms. This is effectively an epigenome-wide association study. It's looking at DNA methylation rather than SNPs. But what we found was that the single feature in the genome that seemed to be responsible was a transposable element. No big surprise there. It was actually a line element, and very satisfyingly, it was in the very long 30 kb intron found in the deficiens gene. So this all made sense. when we did some bisulfite sequencing now on selected trios. This is just one shown here, with M for mantled, N for normal, and O for the mother or tet. What we found was, once again, the same sort of thing. CG methylation was unaffected. CG methylation didn't change in the karma transposon, but CHG methylation and CHH methylation both disappeared in a substantial differentially methylated region of several hundred base pairs within the karma transposable element. You might be wondering why it's called karma. In fact, this type of line element had been discovered before. It had been discovered in rice, and in rice, the karma transposon was actually published a few years ago now, and called karma because it was activated in tissue culture, just like karma in oil palm. And in fact, it's one of only two transposons. The other is a well-known LTR retrotransposon that are activated in rice tissue culture. And it took a whole generation after regeneration from tissue culture for it to move. And that's why they called it karma, because it depended on what had happened in the previous generation. And so, of course, we could then name our epialleles good karma for methylated and bad karma for unmethylated. We could also figure out what was responsible for the phenotype, and it turns out that this is due to alternate splicing. So like all transposable elements almost all have introns, splice acceptor sites, line elements always do, and there's a line element splice site in the karma transposon, and when it's unmethylated, that splice site gets used, and when it gets used, it truncates the deficiency gene and gives a very strong phenotype, and this is just a developmental a series of fruit development showing that aberrantly spliced form of deficiency. We showed a similar dependence on this type of methylation in maize for splicing almost 10 years ago. Interestingly, small RNAs also change in oil palm tissue culture, just as they do in Arabidopsis, which is pretty amazing. So in oil palm, in normal inflorescence, we have mostly 24 nucleotide small RNAs, as you would expect, coming from transposons. But in tissue culture, we actually see 21 nucleotide small RNAs, much more abundant in oil palm tissue culture, as if those active transposable elements are once again being targeted for small RNA production, just as they are in pollen and just as they are in cell culture in Arabidopsis. But of course, this means that we can now test for karma, DNA methylation, or bad karma, if you like, in seedlings before they go into the plantation. And this type of predictive test is actually extremely valuable because it means that non-productive, low-yielding palms don't occupy precious space in the oil palm plantation. And this test is still under development, but it's working pretty well. And we're hoping that it will have some real world impact on this important epigenetic trait. And this actually led to one of my favorite nature covers, "Weed Out Bad Karma," which sort of sums it up. This is very important, of course. You don't need me to remind you, but oil palm is a very controversial crop, especially in these days of heightened awareness of environmental biodiversity and climate change issues. The extensive planting of oil palms in Borneo. This is a picture taken in Sabah, in the north of Borneo, by my colleague in Malaysia, Malina Ong Abdullah, who's the first author on that manuscript and runs tissue culture for the Malaysian government, MPOB. Of course, sustainable oil palm is an incredibly important goal for both Malaysia and Indonesia. And being able to reduce the footprint of cloned plantations by simply removing the palms that will never produce oil would actually potentially have a significant impact on the sustainability and biodiversity of the rainforest, shown here on the left compared to oil palm plantations shown here on the right. Okay, well, I'm going to wrap up there. I'm just going to summarize what I told you. So genome reprogramming in plants and in mammals occurs in the germline and in the embryo at two different stages, guided by small RNA. So epigenetically activated, or we sometimes call this easy RNA, in sperm cells are triggered by a very specific microRNA that targets active transposons in pollen at the PBS. And amazingly, in mammals, a very similar thing happens by targeting the PBS with tRNA fragments. Symmetric CG methylation in plants is not reprogrammed. It's retained in sperm. And that's probably why we have a lot more epigenetic inheritance in plants, for example, transposon silencing and so on, than we do in mammals, where CG methylation is much more extensively reprogrammed. However, asymmetric methylation, which is very important in the Arabidopsis genome at least, and in most plant genomes, is reprogrammed. Both CHH methylation, which is lost in sperm cells, and CHG methylation, which is asymmetric for most of plant development and only made symmetric again in meiosis and in the embryo. And so in the embryo, we've shown now that that methylation, DNA methylation is restored, guided by both K9 methylation and small RNAs, and critically through the K9 methyltransferase SUBH9. In cell culture, this reprogramming doesn't occur because you don't go through meiosis. And so you never remethylate or restore symmetric methylation to CHG and restore CHH methylation. And that's why we think tissue culture, somoclonal variants, which of course occur in all plants that have gone through tissue culture, occurs in such prevalence. And we've shown that this is due to the absence of the correct type of small RNA that would normally reprogram that methylation in the embryo. And as I said, this can have real world consequences to plants that are agricultural plants that are propagated in this way. So I have a lot of people to thank. The recent work I showed you was all done by former postdocs Philippe Bourges, who now has his own lab in Paris, and Jean-Sébastien Parent, who has his own lab in Ottawa. Daniel Guiminetti was on sabbatical, wonderful for actually a couple of years off and on, and really helped a great deal with all of the studies, the recent studies that I showed you, and of course the early work of excuse me, Keith Slupkin, Nilosh, Fred was also very important. Current lab members who contributed to what I told you include John Cohen, Jason Lin, and Chris Sousa Alvis, who I haven't had time to tell you all of the work they're doing that's related to the stories that I told you. Very importantly, the Malaysian Palm Oil Board, there's actually a cast of thousands here that help with the palm oil, the oil palm genome, first of all, the sequence of that, and then all the methylation screening. And at Orion Genomics, the CEO, Nate Lakey, head of bioinformatics, former head of bioinformatics, Steve Smith, and head of research, Jared Warwick. So thanks very much. I'd be happy to take some questions if there's any time.
Okay. Thank you so much, Rob. That was a wonderful talk. And we have minutes for a couple of questions, so feel free to raise your hands. And we're gonna ask you to unmute yourselves once we give you the access. So questions? Okay, we have Jose Luis Reyes Taboada. Can you unmute yourself, please?
Can you hear me now?
Yes, we can hear you.
Okay, just because I have a message saying that the host is not allowing participants to unmute themselves. Okay, anyway, so I guess host will have to be very active right now. Thank you, Rob, for the beautiful presentation. It reminded me a lot of things about small RNAs. And I cannot turn on the video, so you'll hear my voice only. With respect to the last part with the karma intron, which is really interesting, I cannot begin to imagine, and maybe you have some ideas about this, what would be the mechanism by which the methylation of the DNA sequence in the intron translates into the splicing machinery to to regulate splicing, the splicing process. Do we know anything about that?
Actually, we do know. In plants, I mean, we showed it years ago in maize, but other people have observed as well. We typically think of it as DNA methylation, and it's specifically CHG methylation, which of course is very provocative because the splice site itself, the consensus splice acceptor site, is CAG, right? So,
right.
You know, you'd almost think that the actual methylation of the, but in fact, it turns out it's chromatin. So it's K9 methylation that inhibits splicing. And this has been shown in mammalian cells now in several different papers, most recently actually by my colleague here, Adrian Craner at Cold Spring Harbor, about a lovely paper in Nature a couple of years ago showing this. So it seems that because splicing is co-transcriptional, K9 methylated chromatin slows down transcription, which has an effect on which splice acceptor site is used.
Okay.
Having said all that, I do wonder whether DNA methylation of the site itself, like you say, it's a complete mystery as to how that would work, but it's very provocative that that particular cytosine, you know, on the transcribed strand.
So it may have a more general effect then.
Yeah.
As long as you have splice sites being methylated, it will happen frequently.
That's right, exactly. So I think it's, I mean, we did an analysis of the maize methylome, just looking at a variation in DNA methylation and splicing between two different inbred varieties of maize. And there were hundreds of alternately spliced genes that correlated very, very well with methylation of the splice acceptor site. That was in our genome research a few years ago. But, you know, we believe that it's actually chromatin that's doing it. Yeah.
All right, thanks.
Okay, thank you, Rob. We have room for more questions. I will read one that comes from the chat. So this is from Julian Peña Castro. He's asking, many people doing transcriptomes see upregulation of transposons. Do you think it is a bad consequence of the stress or an adaptive response?
Well, in stress-related transposon activation, we know quite a lot about that now. It's typically very specific. So very specific families of transposons are activated by different stresses. And for example, I mean, a really beautiful example is a LTR retrotransposon in Arabidopsis called onsen, which is like the hot bath, you know, in Japanese. It actually has a heat shock promoter in its LTR. So temperature stress directly upregulates that transposon. And there are similar examples. There's some beautiful examples from rice, actually from 20 years ago, that have, for example, herbivory wounding response elements in their LTRs. So stress response seems to be really specific, but you're absolutely right. Transposons come on a lot in different stresses. But I think that's probably why. I mean, that could be a contributor as well, of course, to tissue culture, because tissue culture is very stressful. But we think that the actual DNA replication patterns that we're seeing in tissue culture may have more to do with it.
Thank you, Rob. We have another question from Tamara.
I think you can go ahead.
Yes, you can go ahead. Or if not, you can write it in the chat and we can ask it. Yeah, she's gonna write it. Okay, in the meantime, while she's writing the question, I have a quick question, Rob. Sure. Do you know if there is now strong evidence for either DNA methylation or histone modifications involving paramutation in any system? I know that in Drosophila, there smaller RNAs are involved, but like being smaller RNAs like a diffusible signal that can be inherited?
So there are now very good examples of paramutation in Drosophila, as you mentioned, which are completely dependent on pi RNAs and on K9 methylation. Pi RNAs don't guide DNA methylation in Drosophila, where there's no DNA methylation, but instead guide K9 methylation. So that seems to be the more conserved mechanism. And similarly, in C. elegans, there are great examples of inheritance, actually environmentally triggered adaptive inheritance that is triggered by small RNAs that find their way into the germline and condition K9. With respect to paramutation, absolutely. I love paramutation. I know you've worked on it a lot, Mary. And even actually some oil palm breeders that I was talking to a few years back said that they might have examples of paramutation from Karma. Isn't that amazing? So, I think that's right. I think paramutation, it's got to involve all the same mechanisms in plants as in these animal examples.
Okay, thank you, Rob. We have room for one more question. So we have Mariana. No, wait, I have now Tamara's questions in the chat. So Tamara is asking, in the last part, in the old cell cultures where you find 21 nucleotides, small RNAs, which could be easy RNAs, how are they generated? Is the same pathway that in pollen?
It's a great question. So in cell culture, most of the 21s we see are from Attila transposons, which is a particular family of gypsy retrotransposons in Arabidopsis. And we showed, I didn't cite this paper, we showed in a paper a few years ago that Attila retrotransposons are actually targeted by multiple microRNAs. They're probably quite old and they've had time to accumulate, a lot of enemies. And there's a lot of microRNAs that target Attilas. So in Arabidopsis cell culture, that's probably because it's mostly Attilas, the microRNA responsible. In oil palm, we don't know. We haven't done the analysis. We should. Again, it's not all transposons that make these small RNAs. Karma absolutely does, but not all transposons do, and presumably they're being targeted by some sort of microRNA. We just haven't looked for it. But it wouldn't surprise me. Many plants have either miR845 or they have a rearranged tRNA gene that is sort of like an evolving microRNA that also targets the PBS. This was published by some other groups working in strawberry and some other plants. So there's lots of ways that you can target that PBS, and I bet oil palm is doing that, but we haven't actually looked for it. Okay.
Thank you. There's now many questions. I think we can have one more. So I'm going to read it from the chat. This is from Roman Matias, and he's asking, on the replication of methylation marks, what would happen during natural asexual propagation? Would you expect the same rate of epigenetic variation than in a sexual plant? Or would they experience a general loss of DNA methylation?
What a great question. Yeah, so embryo, what we've now shown in Arabidopsis is that embryo development itself, from the early embryo onwards, can restore DNA methylation in a small RNA-guided way. So even in an asexually propagated species, so long as it produces seeds, then it's probably going to restore at least some of the DNA methylation that might be lost in the germline. Having said that, if it fails to do so, even if it's like at some statistical probability, then of course it's never going to come back, because if it never gets fertilized, then it will never have the opportunity to reimpose the sorts of DNA methylation that would be more common in the population. And so yes, I think clonally, even clonal seed would be more susceptible to epigenetic variation over the long term. I think it's a great question. Really interesting.
Okay, well, time is up. Thank you, Rob, once again for accepting our invitation and for your wonderful talk. And we're gonna follow up in our program. Thank you, Rob.
My pleasure.
Okay, so next in our program, we have Marisa Otegui from the University of Wisconsin-Madison in US. The title of her talk is Endosomes in Plant Development. Marisa, if you are ready, could you please share your screen with us?
Good morning. I would like to start by thanking the conference organizers for having me today. So my name is Marisa Otegui, and in my lab, we work with membrane trafficking pathways that lead to protein degradation. And today I would like to show you some of our results in our analysis of endosomal trafficking in plants. So endosomal trafficking is important in the context of endocytosis. And so when cells require their plasma membrane to change composition, they will sort them in, they will sort proteins into small endocytic vesicles, send them to endosomes, and at endosomes, they can be recycled back to the plasma membrane or degraded. And this is quite important in the context of responses to environmental cues or during development when cells are differentiated into different cell types. So, for example, here we have a cartoon of a human fibroblast. These cells can change completely the composition of their plasma membrane, every single protein in 16 minutes. And they do so by sorting these proteins into thousands of endocytic vesicles and producing thousands of exocytic secretory vesicles that will send new proteins to the surface. And again, it could be for playing new functions or new roles as the cells need to change. And this is also extremely important during development when tissues or cell types are differentiated or to responding to different cues. If this process fails, in most cases lead to death or very severe abnormal, very severe developmental defects. And so getting a little deeper in a higher magnification, let's say, of what is happening in these cells, we know that one of the most important signals for internalizations through endocytosis is actually ubiquitination. So at the surface, plasma membrane proteins are ubiquitinated, and that triggers their internalization into endocytic vesicles. Those vesicles travel to early endosomes. In plants, early endosomes coincide with the trans-Golgi network. And right there, they can be recycled back to the endosome if they lose their ubiquitin or retaining the endosomal system, where at late endosomes, also called multivesicular bodies or multivesicular endosomes, the escort proteins that are cytosolic proteins will bind the ubiquitin in these proteins, deform the membrane, and sort them into vesicles, intraluminal endosomal vesicles, in such a way that when the endosome is fusing with vacuoles, those intraluminal vesicles now can be degraded by local hydrolases. And so the ubiquitin usually is removed before the vesiculation process is completed and recycled back to the cytosol. In escort mutants that fail to sequester this plasma membrane into vesicles, the proteins end up being on the surface of the vacuole, where they cannot be degraded properly. And if we're thinking about receptors telling the cell to divide, or channels or transporters now located in the wrong membrane, you can imagine all these, why these mutants are usually lethal or have serious developmental defects. So the vesiculation process happening here, the two that I mentioned, endocytosis, is quite different from what is happening in the endosomal membranes. Here, the vesicle is budding into the cytosol, and in this case, it's budding away from the cytosol. And the different topology also imposes different constraints. So in the case of endocytosis, we know quite well the machinery here. And so if this is a cytosolic side, a clathrin cage, this is the most common type of vesiculation happening at the plasma membrane. This clathrin cage will directly engage with the cargo, right? So these are the plasma membrane proteins, here represented by these orange lollipops. And so they are physically connected to the cargo during the whole process. And then there are mechanoenzymes, dynamines, that will help constrict the neck to release the vesicle. For the endosomal vesicles, the situation is quite different. This is a cytosol, so the proteins the escort proteins act from the inside. So supposedly they are constricting the neck, but this is where we assume that they are located, causing some kind of hysteric problem on how they act. The other difference is that these plasma membrane proteins, as I mentioned, they lose their ubiquitin at some point, and escort proteins can only recognize them by their ubiquitin. So it is unclear how the escort proteins can retain the cargo in these early budding profiles when there is no physical interaction between the cargo and the escort machinery. So we have been very interested in understanding these very late stages in vesicle formation. And so the escorts are grouping complexes that they are recruited sequentially to the endosomal membrane. Here represented we have, and this is a cartoon, we don't really know how this look like, nobody has been able to see it. We have escort 1 and escort 2 was able to interact with ubiquitin. Escort 3 has been shown to form spirals. These proteins are 7 different escort 3 subunits, and they form spirals that help to constrict the neck. And then finally, SKD1, that is a AAA ATPase, is acting as a chaperone together with the positive regulator RIP5. So together they bind the escort 3 subunits, they remodel somehow these filaments. And the idea based on data in yeast is that then they will recycle all the escorts back to the cytosol for another round of vesiculation. So I'm going to be focusing on 2 main topics during my talk: the unique features of endosomes in plants and the plant escort machinery during development. And so one important thing that we found is that different from what has been proposed in yeast, where each vesicle form individually and the escorts are at the neck and recycle at the end of the vesiculation process, in plants, vesicles form in networks through concatenation. And so there is an uncoupling, a separation, a temporal separation between membrane constriction and scission. So they start to form from the limiting membrane, from the body of the limiting membrane, but they remain connected to each other during that process. We also showed, and we published this a few years ago, that the escort proteins remain associated with the membranes that are being internalized in this vesiculation process. They are retained in these bridges, and that should be important for trapping membrane proteins into these domains. And so we did a number of mathematical simulations, and we came to the conclusion that if the escorts are not retaining this position, forming very strong diffusion barriers, the cargo proteins would escape. And so, as you can see, the 2 patterns are quite different, and they impose different functions and dynamics for the escort proteins. Just to show you some of the data that supported this discovery, these are electron tomograms. So these are 3-dimensional reconstructions from transmission electron micrograph. And so we can see this is a wild-type multivesicular endosome in a Arabidopsis, in a root cell, and all these vesicles on the limiting membrane and form connected to each other. So this is what we call concatenation. And what happened is that one vesicle forms first, and the neck is established, and then another one forms on top, and the second neck is established, and so on. And again, the escorts remain trapped as this is happening. So quite different from what it has been described in yeast. And so we predicted that the concatenation should be important for cargo retention, but is that the case? And so I'm going to introduce now a couple of mutants. So here we have SCOR3 mutant, CHIN1. So just to remind you, SCOR3 forms these filaments that constrict the neck. And so the CHIN1 mutant in Arabidopsis is either embryo or seed lethal. And the other mutant down here is LIP5. So this is a cofactor for the chaperone, the AAA TPase that remodel the escorts during the last phases of vesiculation. The mutant is quite happy if grown under proper conditions, but is hypersensitive to both biotic and abiotic stress. So for these mutants, we were able to determine that indeed they mislocalize plasma membrane proteins. And so here we have examples of the PIN auxin efflux carrier, PIN3 and PIN1 are expressed as fusion with GFP. And this is a root cap of wild type and in LIP5. So PIN3 GFP is localized to the plasma membrane as expected in wild type, but in LIP5, besides being at the plasma membrane, there is a strong signal from the backclar membrane. And as you can see, I show you this at the beginning, is what you would expect for an escort mutant that fails to sort plasma membrane protein to trap them inside those vesicles. In CHIM, again, it dies as embryo and early seedling. So here we have a hard stage embryo wild type showing the localization of PIN at the plasma membrane. In the mutant, there is a strong signal from the vacuole. Now, obviously, these mutants are unable to trap or to sequester these PIN proteins on vesicles. But what is wrong with those endosomes and how is concatenation important? And so what we were able to show is that in wild type, again, the large majority of the vesicles are concatenated. We have placed in these tomograms that you can see here. These are tomographic slices from the raw data. We have placed some red dots just to visualize the vesicles more easily. And this is wild type. You can see here in the gallery some examples of LIP5 and some examples of CHIM1. And what you observe is that there is a reduction in the number of vesicles, but also they are not concatenated, and sometimes they are connected through very long and weird necks that don't seem to respond to the normal cycle of letting vesicle form constriction, vesicle form constriction. And so here is a quantification of what I just showed you. There is a decrease in vesicle density in the 2 mutants, but also concatenation is basically lost. You have more than 70% of the vesicles concatenated in wild type, but less than 20% in the mutants. So what is so unique about plants? Why they do this process in such a different way? And we have been, this is a long-term goal for us to understand, and we are studying a number of proteins that we believe are connected that they will be important to explain this difference. One thing that you notice immediately is that there are many more escort proteins in plants than in other organisms. So these are just the plant escort3 proteins, and so they are clustered together with their yeast and their animal homologs. In the case of IST1, that is just one of these escort3 subunits. I want to call your attention to this clade. There is only 1 in yeast, 1 in humans, but there are 12 in Arabidopsis. And when we look at those more closely, what we notice is that they all contain the IST1 domain that is so important for interacting with other escort proteins, so it's essential for the function as escort subunits. They all have that, but they differ quite a bit in their sizes, and we couldn't identify any other domains of known function that could help us predict what they could be doing. So to start somewhere, really, we decided to analyze IST like 1 through 6, because these are the most closely related to the yeast isoform, and because they tend to be more similar to each other as well. And so we checked their capacity to interact with other escort proteins. So the yeast IST1 protein interact with gene 1. Again, this is SCOR3 protein, just to remind you what these things are. With SKD1, that is the AAA TPase, and with IP5. So we checked for those 3 interactions and found that only AST like 1, 4, and 5 can interact with this SCOR3 component gene 1, and only AST like 1 and 5 can interact with both SKD1 and IP5. So at least their binding capacities and specificities, they don't seem to be fully conserved. We also analyzed the single mutants. You can see here AST like 1, 2, 3, and 6. These are the tDNA lines that were available at that time. All the single mutants were wild-type looking. And so then we crossed them with the LIP5 mutant to see if we could uncover some genetic interaction. And we did only for IST like 1. So the double IST like 1 LIP5 mutant is severely compromised. You can see the plants are really small. They have early senescence. They show constitutive pathogen response, and they died at this stage when they are grown at 22 degrees. But as for other mutants showing constitutive pathogen response, when you grow them at higher temperatures, like at 28 degrees, then they can grow a little further. And in this case, they were able to produce flowers, although they remain pretty much infertile. So we were interested in understanding why they are infertile. And so we checked for pollen viability and found that whereas the single mutants are able to produce perfectly normal pollen, the double mutant, in the double mutant anthers, up to 40% of the pollen was inviable, so infertile. And so curiously, if these plants contain at least one wild type allele of IST like 1 or LIP5, then the pollen fertility is normal. And so it seems to be based on this that the defect seen in the pollen grains is of sporophytic origin. There is something on the anther and in the parent, in the father, that is wrong. So it's a sporophytic defect more than a paternal defect, more than a gametophytic defect. We check more closely those pollen grains and notice that compared to wild type and the single mutants, the XII, the outermost layer of the pollen coat, may have some abnormal features in their pattern. So you can see here in this close-up, the patterns are irregular in some areas, and in some extreme cases where the pollen grains are completely shriveled, the XII is very smooth. By transmission electron microscopy, we were also able to show that whereas the XII is highly variable in thickness, the intine is not. And tryphin, that is another important component of the pollen coat, is made of waxes and hydrophobic material. You can see it here, embedded into the XII. In the double mutant is very irregular in the position and also have some kind of crystals represented here in white. And so it gives us idea that there is something abnormal with the deposition of tryphin and waxes. And so we decided to check the composition of waxes in these anthers. And we were able to confirm that the double mutant has lower content of sterols and alkanes within their waxes. So the XII, both the XII and the tryphin, are actually produced by a sporophytic cell layer called the tapetum, right? So if this is a diagram of an anther in cross-section, the tapetum recovers the pollen sac and surrounds the microspores while they are developing. And so as the microspore develops through the stages that were so characterized in Arabidopsis, and stage 7, we have a tetrad of microspores. At stage 8, the microspores are free in the lochiole. So the tapetum at this stage is secreting material for the formation of the XII. At stage 9, is secreting tryphin components, like boxes, that it can be incorporated into the pollen wall. And finally, at stage 10, the tapetum will undergo programmed cell death. So these cells actually live only a few days, 2-3 days at most, and then they just die as part of their developmental program. So we assume that the pollen lethality that we were seeing in the double mutant were due to defects in trafficking in the plasma membrane protein that were needed for pollen ball formation. And there were two good examples to follow. ABCG16 and ABCG9 have been shown to be at the plasma membrane of tapetal cells and needed for the formation of the exine and the triflume. So we decided to express those two as GFP fusions in our mutants. So this is ABCG9, and there is endogenous promoter. It's most highly expressed at stages 8 and 9. And so here you can see the wild type. This is a longitudinal view of the anther. So this is the tapetum, this is the lochiole, and this is the anther wall. So the magenta spots are chloroplast. And this is just another view of the tapetum. So at this stage, the ABCG9 transporter is at the plasma membrane, the same thing in the single mutants, with the caveat that in LIP5 there is also signal from the backyard membrane as expected from an ESCOR mutant, but still very strong signal from the plasma membrane where the transporter plays its function. However, in the double mutant IST-LAC1 LIP5, what we notice is that there is accumulation in endomembrane, but pretty much there is no signal, very little signal from the plasma membrane. This is different from all the other genotypes. And we can quantify this by calculating the fluorescence intensity ratio between the plasma membrane and the endomembrane. And you can see here a very strong decrease in the double mutant, indicating less localization or lower signal from the plasma membrane. This is not typical of ESCOR mutants. This would suggest that somehow the localization was affected either because there was too much endocytosis happening or the plasma membrane, the protein never reached the plasma membrane. And that would be a defect in exocytosis. So then we tested ABCG16. But ABCG16 is expressed earlier. So again, these are under the endogenous promoter, and ABCG16 is seen in stage 7. And you can see here, wild type is at the plasma membrane and the double mutant as well. These dots are just endosomes. But the transporter is in the right place at this stage in the double mutant. At stage 8, the transporter is degraded, and that happened in both the wild type and the double mutant. You can see very little, it's almost the signal is gone in the anther by this stage. So now we have this situation where one of the ABCG transporters is not at the plasma membrane at a later stage, and the other transporter is properly localized but an earlier stage. So it looked like there is a temporal problem going on. And there is also indication that secretion to the plasma membrane may be affected. So we decided to analyze another marker, another cargo. And in this case, we use the lipid transfer protein fused to GFP. So this is a soluble protein that it gets secreted through the Golgi, through vesicular trafficking and into the lochiole. And so if there is a defect in exocytosis, maybe we could see it with this other marker as well. So the advantage is that the LTPs are expressed all during development of the pollen grain. So at stage 7, we can see signal from the tapetum. Then we can start the accumulation between the tetrads in the lochiole when the microspores are free, stage 8. And then we see more and more accumulation on the microspore walls at stage 10. You can see the wild-type tapetal cells almost depleted of all signal because everything has been secreted into the lochiole. Now, for the double mutant, everything progresses just like in wild-type until stage 9, when we start to see accumulation within tapetal cells. So all these dots here, you can see in this detail So large accumulation of LTP, the lipid transfer protein inside the tapetal cells, indicating that somehow they are not being secreted into the locule. So these results showed us that the combination of these two mutations, IST-like1 and LIP5, is actually affecting secretion, not so much endocytosis, secretion. And this is quite unusual for an ESCOR mutant. We check the features of the endosomes, multivesicular endosomes, and as expected in the tapetum of the double mutant, they are abnormal. The multivesicular endosomes are smaller, the intraluminal vesicles are larger. But more surprisingly, what we found is that the DGN, the early endosome, that also is involved in exocytosis and secretion to the plasma membrane, is severely affected with these big, large bulges. So what we have found, again, is a novel function of these ESCOR proteins somehow in regulating exocytosis at this particular stage during microspore pollen grain development. And just for the last couple of slides, I would like to show you what we are doing right now to further understand the function of these enigmatic IST-like proteins. So I mentioned that we have tDNA mutants for 1, 2, 3, and 6. More recently, we have developed CRISPR mutants for 4 and 5. So now we have a very nice collection of mutants to understand what is the functional meaning of this diversification. So this is a quadruple mutant for 1, 2, 3, and 6. Plants are indistinguishable from wild type, perfectly normal. The 1, 2, 4, 5, 6 quintuple mutant is severely dwarfed, but it still can produce flowers, normal-looking flowers, and is fertile. Now, the combination of IST like 3, 4, and 5 results in these very small abnormal seedlings with large urapical meristems, abnormal roots, that somehow are able to keep developing and forming an inflorescent meristem that results in something rather abnormal, with the flower buds replaced by more meristematic regions. So this is a project that a new postdoc in my lab, Ariadna Gonzalez-Solis, has started, and so we are very excited, and hopefully in a few months we will be able to tell you more about these unusual IST-like proteins and what they may be doing in plants. So just to wrap up, I mentioned that vesicle concatenation in plants is very important for trapping cargo, that the double mutant IST-like1, LIP5 plays an expected function in exocytosis in tapetal cells, and that we are making progress in understanding what the other IST-like proteins do in plants. And with that, I would like to thank everyone who has been working on this project so hard, Kaya Goodman and Julio Paez Valencia for their work on the anthers, Ariadna Gonzalez-Solis for the most recent work in the IST-like mutants. Thank you so much for your attention.
Thank you, Marisa. We have room for one quick question. Okay, so we have Luis Cárdenas. Go ahead, please.
Thank you, Mario. Nice talk, Marisa. I would like to ask you about it's a very nice talk about tapetum and how this is affected. Do you know anything about the pollen tube tip growth? Because this is highly dependent on also from exocytosis and endocytosis. Have you seen any defect?
Yeah, so that's actually what we thought was happening first. Actively growing cells by tip growth would be affected in this mutant. However, if we manage to make double mutant pollen, like in heterozygous mutants, those pollen grains are able to produce pollen tubes normally. And based on the segregation of the mutant alleles, it looks like they are as competitive as a wild type pollen. So it seems to be that this combination, these two mutations have a very particular role in tapetal cell. We also noticed some defects on the secretion of waxes in leaves. So it may be something connected with this type of transporters and in the cells that are expressing them, epidermal cells. But no, we couldn't find any defect in pollen tube growth.
Okay, thank you.
Thank you.
Thanks.
Okay, so we're running on a tight schedule, so we need to move on. Thank you so much, Marisa, for your wonderful talk. Now I will, we will go now with Jocelyn. Dan, if you want to come in.
I am here. Thank you, Mario and Marisa. Continuing the plant development session, we're going to learn about Plasmodesmata. It's my great honor to introduce Dr. Jocelyn Benitez Alfonso from the University of Leeds, and her talk is PD Wall Mech, Dissecting Cell Wall Properties and Cell-to-Cell Signaling via Plasmodesmata. You can take it away, Jocelyn.
Thank you, Dan. I'm just checking that you can hear me all right.
Yes.
Yes, okay. Let me share my screen. And there we go. Hello, buenos días para aquellos que están en México. My name is Jocelyn Benitez Alfonso, and as Dan has introduced me, I am Associate Professor in Plant Science at the University of Leeds, and a proud Cuban Spanish for your knowledge. First, I want to thank the SMB committee for inviting me to this amazing conference, especially Dan, who was the first one to contact me. Today, I will share my ongoing work in the discovery of new cell wall components and the mechanisms facilitating intercellular transport via plasmodesmata. So plasmodesmata are these membranous pores that you can see here inserted in cell walls, through which has been described the transport of proteins, RNAs, small molecules, and connect almost all cells in the plant body.
Hello, everybody. It seems like we have some connection issues with Jocelyn. Give us one moment and we'll try to reconnect or get the presentation, a recording of the presentation going. 1 second, please.
Hello again.
You are back, Jocelyn. We can hear you.
Oh dear. I see that there is some issues with the connectivity. I don't know what is going on. So hopefully.
No worries.
So some viruses has been shown to move via plasmodesmata. And in the picture here, you can see the tobacco mosaic virus, which has been filtrated in this mature leaf, from where it can move intercellularly and into the new tissue. So clearly, controlling plasmodesmata transport could be very important tool for crop improvement. But unfortunately, we still lack knowledge on what are the factors controlling plasmodesmata formation and what are the regulatory cues. In my lab, we have focused on understanding the role of cell walls in plasmodesmata regulation, with the vision of getting new tools to improve crops, but also to develop new biomaterials. So today, I will introduce you to the objective of this work, specifically how we are developing knowledge and tools on the composition and regulation of cell walls around plasmodesmata, how we are using this knowledge to develop, to target new cell components in order to modify plant growth. And finally, if I have the time, I will introduce you into our work in biomaterial design based on the knowledge acquired from plasmodesmata. This work has been carried out by a team of researchers in my lab, but also count with the support of amazing collaborations from the plant science to the cell and the physical science and the mathematical modeling. Some of the names are mentioned here. So the work has been funded by UKRI, which is the research council in the UK, and is under this acronym named PD WOLMEC, which is a stand for plasmodesma wall mechanics. And the reasons is because we are gonna complement our studies on the structural and cell biology of plasmodesmata transport and formation with studies on the mechanical properties of the cell walls that surround plasmodesmata in order to get a better understanding and integrated models in which we can clearly predict functional targets to modify plant growth through targeting plasmodesmata. Finally, this knowledge on cell wall components will be integrated into material science type of approach to generate new composite materials. The work is supported by companies such as Futamura and others, and by the Centers of Innovation at Leeds, including the ASPB Center and the Bragg Center for Material Research. So this is a real cross-disciplinary area that is inspired really by the understanding of the role of callose. Callose is a cell wall polysaccharide that accumulate at plasmodesmata site. This is seen here in immunolocalization in cell walls of tomato pericarp, where callose can be seen fluorescent in green based on an antibody that is specific to callose. So this beta-1,3 glucan has been shown or has been suspected to modify plasmodesmata transport through increasing the accumulations of cell walls around plasmodesmata, thus limiting the cytoplasmic aperture that allow molecules to pass through the channel. Callose synthase and beta-1,3 glucanases are the enzymes that synthesize and degrade callose, and the activities of these enzymes has been shown at plasmodesmata. Moreover, we now have mutants in these enzymes, such as this PD-located beta-glucanase, which I described in 2013, which shows really exciting phenotypes, such as in here, you can see the formation of groups of lateral roots, which is very unusual in Arabidopsis. In other mutants, such as the callose synthase 3, expressed on an estradiol-inducible promoter described by Ika Hilariuta lab, you can see that there is an induction of callose upon estradiol application. And this induction of callose lead to general reductions in plant growth in Arabidopsis, suggesting again that callose play a really important role in plant development. We have been exploiting further the tools that we have developed to modify callose by applying this knowledge into novel signaling processes, such as the formation of nodules during legume-rhizobia interaction. As many of you probably know, nodules are organs that are formed to host the bacteria rhizobia. Here, these are shown here in Medicago truncatula plant, and within nodules, the rhizobia is able to fix nitrogen. Therefore, this is a very important process for sustainable agriculture. And we know that different factors modulate signaling to and control the formation of nodules in legumes. These are long-distance signals from shoots, which are controlled by the receptor, clavata receptor son, and also short-distance signaling that occurs between the epidermal and the cortical tissues, which has been related with the expression of the transcription factor nodulin section 1 or NIN. So we were wondering if plasmodesmata could play a role in these transport processes. And in collaboration with Fernanda Carballo-Nivel in Toulouse, we studied this aspect by designing a line that expresses YFP, a mobile version of YFP in the epidermal cell layer. As you can see here, this YFP gets restricted in the mature epidermal cell layering roots, suggesting that the epidermis is disconnected to the underlying tissue, this in non-inoculated plants. But when rhizobia is present, somehow these channels are open and transport of GFP start to occur in between the epiderma and the underlying tissues, suggesting that plasmodesmata is active. Further work identify a beta-1,3 glucanase, which localize and express in these nodules, and since the very beginning of the infection process and into the nodule development. Ectopic expression of this beta-1,3 glucanase in legumes, in this case Medicago truncatula, really increase the number of nodules that are informed during infection, whereas silencing of this beta-glucanase using an RNAi line lead to a reduction in nodules. Again, suggesting that this beta-1,3 glucanase participate in nodule formation. Further work, especially the fact that the transcription factor NIN fails to be inducing the nodules in lines in which calosyntase 3 is activated, led us to propose these models in which plasmodesmata mediate the transport of signals between the outer cell layer into the inner cell layer to regulate the nodule formation in response to rhizobia infection. And these beta-glucanases are a part of this mechanism. But questions remain on how these mechanisms are actually integrated with other mechanisms that regulate nodule formation, specifically the long-distance regulation of nodules in response to nitrate and in response to the plant physiological status, which is controlled by the clavata factor SON. We have made a little bit of progress in this area by identifying a novel receptor-like kinase, which is expressed in a small amount after rhizobia infection in Medicago roots. But this expression gets very well increased in mutants in SON, suggesting that indeed this, the regulation of this receptor-like kinase is relying on SON. This receptor-like kinase locate at plasmodesmata, as you can see here in this confocal micrograph in which the fusion protein in this receptor-like kinase co-localized with aniline blue and callose in the cell wall periphery, suggesting that is plasmodesmata locations. Ectopic expression of this receptor-like kinase really didn't show us any natural phenotype. So we were kind of wondering what would be their role. And due to the role of SON in nitrate, we started to look at what is the effect of this overexpression of this line in the presence of nitrate. And we found that although control plants are able to reduce callose in the presence of nitrate and rhizobia in Medicago roots, this was not possible in this overexpression line of this receptor-like kinase, suggesting that this receptor-like kinase really integrate with the process of callose regulation in response to nitrate in Medicago roots and in response to rhizobia. Concomitantly, we can see that the infection threats, or the formation of infection sites and the formation of nodules in nitrate increase when ectopically expressing this receptor-like kinase. So we're still figuring out how nitrate and alterations in both SOM and this plasmodesmata located receptor mechanistically modify callose and symplastic intercellular transport. But watch this space, is what I will say. So far, I have shown you examples in which targeting callose regulation at plasmodesmata can be used as a tool to modify plant growth. But the project in general comprises several other cell wall components. And we are starting to understand and dissect what are these other cell wall components that might contribute with callose in plasmodesmata regulation. This is an example of the type of techniques that we are using. This is Raman spectroscopy. So we are basically looking at regions in which callose is deposited using Raman and applying a principal component analysis to identify peaks that are correlated with the callose. We can see here that certain hemicelluloses and pectins are identified in this principal component 3, which is rich in callose. And further analysis using cell wall probes identified by Paul Knotts and plant probes led us to identify which type of pectins are those. So here, for example, we can see that these peaked fields in cell walls are depleted in rhamnogalacturonan 1 with the linear galactan side change, whereas they are enriched in branch galactan side change. So very small modifications in these pectins leads to a different conformation of these pectins, and seems that specific variants of these are surrounding plasmodesmata and colocalized with callose and potentially modifying plasmodesmata transport. But to test this hypothesis, we need to identify which proteins are modulating the regulation of these other cell wall polysaccharides. And for this, we are carrying out a bioinformatic approach in which we integrate the knowledge obtained from the different plasmodesmata proteomes which has been published so far. So this has generated a pipeline by one of my students, which integrate the different subfamilies identified in these proteomes and identify the genes within these subfamilies that encodes domains characteristic of non-plasmodesmata proteins, such as signal peptides, transmembrane domains, and GPI anchors. These classify the output in different lists according to the likelihood to find these proteins at plasmodesmata. Such as in Arabidopsis, 206 proteins were identified in that list A, which contain these structural features and a likelihood to target plasmodesmata sites. What we can do with this list is now create interactomes. So co-expression analysis to understand which of these proteins are expressed in relation to normal regulators of plasmodesmata, such as callose. And by looking closely to this type of interactomes, we can identify not only callose modifying enzymes, but closely related enzymes that modify cellulose, hemicellulose, pectins, and encode cell wall proteins. So this is now a new pool of enzymes and proteins that we are starting to study to dissect what their role at plasmodesmata are by using genetic mutations in these proteins and studying closely what is their intracellular localization. So to summarize this second part, not only callose is accumulated at plasmodesmata, but cell walls around plasmodesmata has a specific cell composition of pectins as hemicelluloses, and we are starting to identify proteins that regulate those and understanding what their role is in the regulation of transport. But how really cell wall composition regulate plasmodesmata transport? The general idea or belief is that callose and other cell wall components physically constrict plasmodesmata aperture and thereby reduce the cytoplasmic sleeve that allow transport through the channel. This model has been challenged recently by the group of Emmanuel Bayer, which discovered this type 1 or clue plasmodesmata with not visible cytoplasmic sleeve. Despite no visible cytoplasmic sleeve, though no means to where the transport can occur, these plasmodesmata seems to be highly permeable, or at least are highly present in very permeable tissue, suggesting that they are very functional. So how this happened This question was partially addressed by using computational models with Eva Dijnen in the Netherlands, which use the mathematical description of the geometrical features of plasmodesmata to devise a formula to calculate the effective permeability of the wall based on plasmodesmata features, but also on plasmodesmata density. When Eva compared the differences in permeability between a straight plasmodesmata, like the ones defined as type 1, in which the cytoplasmic sleeve and the neck region is of the same size, with type 2 plasmodesmata, in which we have a higher cytoplasmic sleeve, she could see that there was very little differences in thinner cell walls, so in cell walls that are young. Whereas these differences are more significant in mature cell walls. Does this make sense, thus, that we actually have this type 1 plasmodesmata existing in very young cell walls? But that doesn't explain how callose or other cell wall polymers act at plasmodesmata. To kind of investigate this question, I took on some kind of physical model to mimic what happened with callose at plasmodesmata. So I created callose cellulose mixtures in ionic liquid, which then are reprecipitated to form hydrogels that can be studied using a scanning electron microscopy. So these hydrogels of various concentrations were obtained, and the mechanical properties of the hydrogels were determined using FM nanoindentation and a textured analyzer. Our findings shows that Young modulus was reduced as callose concentration increased, whereas the ductility of the gel, represented here as the rupture point, really increased with increases in callose concentration, suggesting that basically callose acts as a plasticizer to cellulose, so giving it a more elastic feature. Interestingly, these changes were nonlinear, so very little callose induced higher differences in these Young modules, suggesting indeed that callose and cellulose might interact. Thus, it's very difficult to predict the properties that these mixtures will generate. This model kind of makes sense if we consider callose as an structural component of plasmodesmata. So maybe callose is necessary in this type 1 plasmodesmata to give or to provide some kind of elasticity to these cell walls, thus allowing these cell walls to be modified for the transport of bigger molecules such as GFP. These findings also open the door to identify new biomaterials obtained by mixtures of callose and cellulose, which is an area that we are exploiting with industry. So, but we are interested in understanding this further. So within this PD Wall MEC project, we are understanding the contribution of other cell wall components, pectins and hemicellulose. And we are asking the question how these cell wall properties around plasmodesmata really vary with different cell types and plant species, and how this contribution really integrates with plant development and responses to the environment. And I'm going to give thank you to all the ones that deserve done this job in my lab: Lian Yerma, Philip Kirk, Candela Spaniagua, Sam Asbury, Mercedes Hernandez, Radwa Busalet, and Rocio Gaudioso Pedraza specifically contributed to the slides that I have shown here today. I have counted with amazing collaborators, including Eika Hilariuta, Fernanda de Carballo Niver, Emmanuel Bayer, Paul Knox, Mike Rees, and Eva Dijnen. And with the support of several agencies within UK and the Liverhume Trust. And please connect with us via Twitter at Benitez Alfonso Lab and visit our website at Benitez Alfonso WordPress. Muchas gracias. Thank you for your attention. And I hope that we have some time for questions.
We have plenty of time for questions, and that was a wonderful talk, Jocelyn. I really love the modeling, and especially the material science and mechanical modeling. That's really cool. We have a first question from Luis Cardenas, and you should be able to unmute now, Luis.
Thank you. Thank you, Jocelyn. I got two very short questions. As you said, the overexpression of the glucanase induce a higher nodulation. Do you have any idea about the mycorrhizal interaction? Do you see also, do you expect to have also an effect on this kind of interaction? And my second question is talking about the cell wall composition. Yeah. My second question is about the composition of the cell wall in the plasmodesmata. Do we have any idea about the pectin composition? I'm talking about the nature of the pectin. Is this most de-esterified or this is esterified pectin?
Okay, that's good that I have answered to both of your questions.
Okay.
Maybe no successful answers, but the answer that I have so far. So in terms of the mycorrhiza fungi, we are starting to look at that. So far, what we have done in the lab is investigating if arbuscular mycorrhiza induce also changes in callose deposition and in intercellular transport. And what we have found so far is that arbuscular mycorrhiza do not reduce callose, but instead increase callose in the root, so suggesting that is a different mechanism. In any case, we have starting to use the mutants, including the SON mutant, to exposing them to rhizobia and arbuscular mycorrhiza to actually understand if there is interaction between the two symbionts that could kind of dissect a crosstalk between these pathways. So the arbuscular mycorrhiza fungi is ongoing. The problem with arbuscular mycorrhiza is that we have to do the infection in soil. So it's very difficult from the cell biology point of view, whereas we can do in rhizobia in plates, so we can nicely have nice roots to actually visualize what is happening all the time. In terms of the, does that answer your question?
Yes.
Okay.
Yes.
In terms of the cell wall composition of pectin composition, we are studying the pectin composition not only by using these molecular probes that you have seen here, but also by using other approaches such as the kind of immunoprecipitation with callose. So what we have been starting to do is taking callose and a protein that binds callose and precipitating this protein in cell wall extract and seeing what else is coming up with the callose. And this is unpublished data, but there is some the esterified pectins that comes with the callose, suggesting that potentially they are at very close proximity at plasmodesmata. We have used probes against the esterified pectins, but we haven't found specifically accumulated at plasmodesmata, but that doesn't mean that they don't interact with the callose. So what we are seeing in these probes is that sometimes when we increase callose, we also increase some of these esterified pectins. So there is a more complex interaction between these polysaccharides that the one that I have described today that we are just starting to dissect.
Thank you, Yosemi.
I think we have two quick questions in the chat. The first is from Gerardo Valdez. He says, what about senescence? Callose is used to accumulate in several tissues according to phenophase. Maybe about senescence?
Yeah, okay. So we haven't studied senescence specifically because senescence is when actually plasmodesmata seems to stop functioning in the way that we, so we are more focused on meristematic tissue or developing tissue. But in the past, in Cold Spring Harbor, where I was in Cold Spring Harbor, we identified GAG1, which was a mutant in a thyroidoxine that indirectly modified plasmodesmata transport. And what we found is that when we topically express this protein under a senescent promoter, we could delay senescence in these lines, which was really interesting. We didn't expect that, but that's something that we saw. That's probably due to the export of sugars that occur during senescent tissue. But we haven't followed up that area of research.
Well, thank you. We're at the end of the time. That was a great presentation, Yaselin. Thank you so much.
Thank you. Gracias.
And up next, we have Stuart Gilmore. He's coming from Lanhebio, and his talk will be the zygotic genome activation in Arabidopsis. You can take it away, Stuart.
Okay, let's see here. Okay, can you see my screen?
It looks good, and we can hear you.
Okay, thanks. Thanks so much, Dan. Thanks to the organizers for organizing such a great meeting and for inviting me. It's really a pleasure to be here. Let me just fiddle with my screen here a little bit. Hold on a second. Sorry, people.
No worries, take your time.
I'm just trying to like minimize a window here. Here we go, okay. Yeah, so I'm gonna talk about zygotic genome activation in Arabidopsis and work we've been doing on this in my lab for about the last 10 years. So in my lab, we work on seed development and we use Arabidopsis thaliana, which I'm sure you're all familiar with. We use it to work on embryo development in particular, because Arabidopsis seeds are very easy to clear. And so it's really easy to look at them under a microscope, which has been very important for a lot of our work. And seeds are really interesting because they're composed of tissues of different origins and of different ploidies. So if you have, here's a seed, this is about 3 days after fertilization. We have a globular stage embryo. This is the seed coat, which is maternal tissue. And then you have the embryo, which is the product of fertilization of the sperm and the egg. So it's diploid with one maternal and one paternal genome. Then you have the endosperm, which has 2 maternal genomes and 1 paternal genome. So there's all kinds of interesting genetics going on here. And what I'm going to talk about today is zygotic genome activation, as the title says. We also study pattern formation in my lab. But zygotic genome activation is what goes on in the first few days or so after fertilization, when the newly united maternal and paternal genomes become zygotically active, and it's part of the maternal to zygotic transition in early embryogenesis. So this is something that also occurs in animals and that people have been studying for a long time, and there's quite broad agreement about how this works, how the maternal to zygotic transition works in animals. And the consensus is that there's an early period of maternal regulation of embryogenesis, and then depending on the species, it could be a few hours later or about a day later, the zygotic genome takes over the regulation of development, and maternal transcripts are actively degraded. So you have early maternal regulation, zygotic regulation starts up, and then the maternal transcripts and proteins are often actively degraded so that you have zygotic regulation of embryogenesis. Why does this happen? There's several different hypotheses. In some species, for example, in Drosophila, the early divisions of the nuclei are so fast that you can't actually transcribe anything more than a short gene. So they couldn't regulate their own early development. Another idea might be to allow reconciliation of chromatin marks that come in from the sperm and the egg, something that Rob Martinson was talking about earlier. And another idea is it's a way of avoiding too much paternal control in early embryogenesis, where the mother wants to allocate resources equally to all of her offspring. And so she would tamp down on paternal gene activity early on. So I'm going to talk about work obviously today in plants. And people have been studying this topic in plants now for about 20 years. And we're still trying to get to a consensus about how this works. There were early hypotheses, really two. One was similar to animals, where early embryogenesis in plants is under maternal control, and after a few days, the zygotic genome takes over. Other evidence supported very early zygotic regulation of embryogenesis in plants, so almost the role of maternal regulation would be very, very short. I mean, that's what I'm going to talk about today. Let's see here. Okay, so here's an outline for my talk. It's very important to distinguish between studies that people have done using isogenic embryos and using hybrid embryos that you need to do parent of origin transcriptome profiling. So in the first part, I'm going to talk about studies of zygotic genome activation in isogenic embryos, so transcriptomes and functional analyses using embryo defective mutants. The next part, I'm going to talk about genome activation in hybrid embryos, also transcriptomes and functional analysis with embryo defective mutants. And in the last part, I'm going to talk about a recent meta-analysis we've done of transcriptome data from different studies of hybrid zygotes and embryos. So as to when transcription starts in the zygote in plants, I think pretty much everyone agrees now that it starts very early. So these are studies from maize, rice, and Arabidopsis over the last few years, and they all came to the conclusion. So these are studies where they were comparing egg cells and zygotes, isogenic, okay, so self plants, and they found that about 10% of the genome, the transcriptome, is upregulated in the zygote compared to the egg cell within a few hours of fertilization. So transcription of a good portion of the genome starts early, but it's not the whole genome. It's really about 10% according to these studies. So there were also, from a number of years ago, other experiments using enhancer trap and different kinds of markers where they found signal for the markers very early, but the observation was that it was the maternal allele that was more highly expressed than the paternal allele. So those first observations were from Jean-Philippe Vier about 20 years ago. Then Daphne Autran made other observations about 10 years later, looking a little bit more carefully. And it's not that there's no expression of paternal markers, but it was less than maternal markers. So, but the idea that there might be more expression of maternal alleles in early embryos was controversial from the beginning. There are a number of examples of markers where they were expressed essentially equally between the maternal and paternal alleles. Here's a bunch of references. And I think also people were skeptical about a more important maternal role early on because of the large collection of embryo defective mutants which exist in Arabidopsis. There are about 500. This is work from David Meinke's lab over many years, as well as others. And so embryo defective genes are genes that are absolutely required for embryogenesis. And so in the absence of gene function, you get phenotypes. The embryo could die or it could continue growing, but just be abnormal. And if you take a heterozygous mutant plant and you self it, and you look at saliques like that I'm showing here, of a late stage salique, you see about 25% mutants consistent with a zygotic effect. So if they were maternal effects, you would see 50% mutants because the plant is heterozygous. And so it was about this time that I started my lab and we started thinking about this issue. And one of the first things that we did was to order a bunch of embryo defective mutants from the Arabidopsis Stock Center. And this is work by my former PhD student, Gerardo Del Toro. And what he did was, to start, he grew all these embryo defective mutants and looked carefully for phenotypes starting at 2 days after pollination, 2, 3, 5 in mature embryos, and to look at segregation ratios. And what he found is there's a lot of variation, of course, because you're looking here at penetrance, expressibility, all kinds of things are going on. But there were some of these embryo defective mutants in self plants where at 2 days after pollination, you get more than 25% mutants. Some of them actually have 30 or 40 or almost 50% mutant phenotypes during a short window. And then during development, the embryos, the percentage of mutant embryos decreases to where it's about 25% later on. So this suggested that something a little bit more complicated is going on. It's not just purely a zygotic effect. But a more clear way of testing for maternal effects, and another way of putting it is to test if the paternal allele has full activity, is to take an embryo defective mutant as a mother and cross it with wild type pollen as a father. And in the case that the wild type alleles are fully active, fully transcribed, fully translated, etc., these wild type alleles, unless there's haploinsufficiency, should be able to cover the lack of a mutant, the lack of a functional maternal allele. And so in the case we have full paternal activity, if you look at the embryos after developing, after doing this cross, you won't see any mutant phenotypes because they'll all be either heterozygous or wild type. 50% will be heterozygous, 50% will be wild type. But in the case where there may be less paternal allele activity, what you would expect, and the paternal alleles might not be able to compensate for the lack of a maternal allele, and then you'll get some mutant phenotypes that you see, but the percentage of phenotypes that it's segregating in a sleek actually decreases during development. And so Gerardo did this fairly giant experiment where he crossed, in the end it was 49 embryo defective mutants. He crossed them with wild, this is all in Colombia ecotype. He crossed them with wild type pollen, and he found, this is a heat map summarizing his results right here. And these are clearing preps. So these are what the embryos look like when he's doing this assay. Here's an example for ATSY3A, where the wild type allele from Colombia can complement the lack of a functional maternal allele. Essentially, all the embryos look normal. Here's another example of BMB 2804, where the wild type paternal allele essentially at the beginning is not totally functional because you see mutant phenotypes 21% at 2 days, 4.9% at 3 days. By 5 days, there are very few mutant phenotypes. And so of these 49 embryo defective mutants, 80% showed maternal effects. So that's this purple here. So purple is 50% mutants and beige is 0% mutants. So 80% of them showed some kind of maternal effect, which was transient, okay? While 20% of the genes that he tested showed no maternal effect whatsoever. So apparently the paternal allele here is perfectly capable of complementing a maternal allele in early embryogenesis. And more recently, Axel Orozco Nieto, a master student in my lab, went back to these experiments and used 24 additional embryo defective mutants, and he found similar things. So here we have maternal effects on the left, paternal effects on the right. So he tested both maternal and paternal effects. So again, for a maternal effect, you take a heterozygous embryo defective mutant as a mother, you cross it with wild type as a father. To test a paternal effect, you take a wild type mother and you cross it with a heterozygous embryo defective as a father. And when the embryo defective was used as a mother, we found a number of, maternal effects for all these mutants. When it was used as a father, some of them, about 10 of them, had some paternal effect. The rest had essentially no paternal effect, but the paternal effects were always less severe than the maternal effects. So from this first part of the talk, the conclusions are that that transcription of the genome does start soon after fertilization. It's not the whole genome, but it does start early. And the work with the maternal, the embryo defective alleles shows that at least for these 73 genes that we tested, the maternal allele is initially more important than the paternal allele. So to talk about work in hybrid embryos, obviously to look at the genome level, you need to do transcriptomes. To assign reads to a mother or father, you need to use polymorphic ecotypes. So for example, it could be Landsberg and Columbia, and you take advantage of the polymorphisms in the parents to assign reads in the zygote to one parent or the other. Okay, and so a number of labs have done these kinds of experiments. One was published by Nodine and Bartel in 2012 using the CVI Columbia hybrid, where they found essentially equal maternal and paternal reads at the 1 to 2 cell stage and at the octant stage and at the globular stage. Just a couple of years ago, another study was published by Zhao et al. In Developmental Cell, where they used a Columbia Landsberg hybrid, and they found at 14 hours after pollination, so quite early, they did find more maternal transcripts, but by 24 hours after pollination, the number of total transcripts was quite equal between the maternal and paternal genomes. Here's a study from Rice where they found something quite different. They found a massive maternal bias in early zygotes. So these studies with hybrid embryos didn't all come to the same conclusion. And especially just thinking about the Arabidopsis results, it's hard to reconcile equal maternal and paternal transcripts with the results that we found with functional results with embryo defective mutants, where we found more maternal effects than paternal effects. One way to explain this might be that hybridization that's required for producing these parent of origin transcriptomes has effects on paternal allele activity. And so to look at that, this is data from Axel Orozco that analyzed by Jaime Alanis. And so what this is showing is it's taking, Axel did crosses, and instead of using wild type Columbia as the father when crossing to an embryo defective mutant in Columbia as the mother, he used different ecotypes. So he used CVI, Landsberg. So these are the two hybrids that I just showed you in the previous slide. He also used TSU and Burr. And what he found, this is a very nice result, as you might expect in Columbia CVI especially, and also in Columbia Landsberg hybrids, the maternal effects are less. So these have been normalized to the same cross, but using Columbia as the father. So one would be the hybrid is exactly the same as an isogenic cross. So these hybrids show less maternal effects. They do have a maternal effect, but it's less, or earlier paternal allele activation. Whereas these other two hybrids here showed maternal effects that were exactly the same as isogenic Columbia. So luckily, Raju Datla and Daoquan Chang had actually produced a transcriptome from the Columbia 2 hybrid, which based on our functional analysis was very, very similar to isogenic Columbia in terms of maternal effects. At several different stages, crosses in both directions, a good number of reads. And so we analyzed, this is Jaime Alanis analyzed this data, and we found something different compared to this Columbia CVI and Columbia Landsberg experiments, which is that there's an early maternal bias at the zygote 1 cell and octant stage, but beginning at the globular stage, there's essentially equal maternal and paternal reads. And there's another way of showing it. This is looking at statistical analysis, looking at the reads to see if they have, and the different kinds of parent of origin behavior you see here. Thousands of genes at the zygote 1 cell stage and at the octant stage show a maternal bias in both directions, suggesting that again, there is a maternal bias in this hybrid in early embryos. We don't think this maternal bias is due to contaminating transcripts from the sporophyte, from the seed coat, for example, because when Jaime, he did these analysis where he graphed the maternal fraction of reads per gene and then he divided the genes based on the zygote to ovule ratio. And there's no relationship between genes which are expressed more highly in the ovule compared to genes that are more expressed more highly in the zygote. So we think this is quite good evidence that our results are not swayed by maternal contamination. Another really interesting thing that he found is that the maternal bias of transcripts in the zygote is not correlated to the relative level of the transcript in the egg. So, suggesting that this maternal bias might actually be due to active transcription in the zygote. Here, Jaime looked at, these are genes which are expressed in the zygote but are absent in the egg. They're highly maternal. And he found a similar thing where he looked at genes that are expressed at 10 times higher in the zygote compared to the ovule. And then he had this very nice idea of looking at intron reads. So you can think of intron reads as a kind of proxy for active transcription because they're still nuclear. And he found that at the zygote and octant stages, the intron reads are also highly maternal, whereas at the globular stage, they're at about 50-50. So again, consistent with an early maternal transcript bias that may be due to active bias towards transcription of maternal alleles. So the conclusion of this part of the talk, embryo defective mutants in crosses between Columbia and Sioux and Columbia and Burr show maternal effects that are indistinguishable from isogenic Columbia. Transcriptomes of Columbia-Sioux hybrids show a bias in maternal transcripts in early embryos. And this maternal transcript bias is likely, or at least possibly, due to preferential transcription of maternal alleles. So, and just for the last bit of my talk here, so getting back to these other transcriptomes, different scientists came to different conclusions. Here's a summary here of the total reads. Okay, so again, Nodine and Bartel found 50% maternal and paternal reads at these stages. Zhao et al. Found an early maternal bias, but it was 50-50 by 24 hours after pollination. Whereas in our analysis of the Sioux-Columbia hybrid, we found about 80% maternal reads at the 1 to 2 cell zygote stage, 73% at the octant stage, and then about 50-50 at the globular stage. But these are total maternal and paternal reads, not reads per gene. So that's an important thing to keep in mind. And another source of difference between these studies could be the way you analyze the data and the way you represent it. So Nodine and Bartel and Zhao et al. Represented their parent of origin data as log2 fold change of one ecotype over the other. And you get these very nice curves here, which are centered around 0, which is equal maternal and paternal transcripts. Here in Zhao, these ones that are a little bit offset from 0 at 14 hours when there are more maternal transcripts, and then by 24 hours, it's exactly at 0. When we analyze our own data, graphed our own data using this log2 fold change, we also found that at the zygote, 1 cell, and octant stages, there's a maternal bias, which is essentially gone by the globular stage. Okay, but it turns out that representing the data as log fold change makes genes which are transcribed from one allele and not the other disappear. You don't see them. And so when we graphed the Nodine data and the Zhao et al. Data and our data also as maternal fraction, this is the same way that I showed you our data a little bit earlier, the conclusions of Nodine and Bartel are essentially the same. There's almost no parent of origin behavior in this hybrid. There's small peaks here. So the maternal fraction is 1 is the gene, the reads are exclusively maternal, and 0 would be the reads are exclusively paternal, and 0.5 is equal maternal paternal. So Nodine, consistent with their conclusions, there's almost no parent of origin behavior in Columbia CVI hybrids at all. However, in the Zhao data, we saw what they saw, which is that there's a maternal bias here at 14 days, and then by 24 4 days, there's nice peaks here at 0.5, which is equal transcripts, but there are also very prominent peaks of reads that are either exclusively maternal or exclusively paternal. And in the case of our data, we saw there's a maternal bias here early on, which shifts to being more biallelic during early embryo development and very little genes with paternal bias. So this was kind of unexpected. Just looking a little bit more at this data and these reads that are either highly maternal or highly paternal, they're actually a very large part of the data set. At the 14 hours after pollination stage, there are about 42% of the reads are unidirectional, and at the 24 hours after pollination, about 24%. And so this is really was a little bit surprising, but does go, I think, a long way in reconciling the previous conclusions from this transcriptome with our conclusions from functional data. And it just also looking at intron reads, so as a marker of active transcription, it's quite interesting. Here in the Zhao data at 14 hours after pollination, you do see a large bias towards maternal transcripts. These are intron reads, whereas at 24 hours after pollination, the different directions of the cross are quite different. When Landsberg is the mother and Columbia is the father, the reads are more distributed evenly between equal maternal and paternal transcripts, or only paternal or only maternal. Whereas when Columbia is the mother, you see actually very strong biases towards either being maternal or paternal, but not both. And in the Columbia 2 transcriptome, we saw something, active transcription that actually looks quite a bit like total transcripts, again, suggesting that these biases may be due to active transcription. So from the third part of the talk, the conclusions here, there are thousands of genes in Columbia Landsberg hybrid zygotes that are transcribed from either the maternal or the paternal allele, but not both. So it's a scenario that's closer to non-equivalent parental contributions to the zygote. Columbia CVI hybrids show almost no parent of origin behavior at all, so more like equal transcripts. And Columbia 2 hybrids show a maternal bias in transcripts and zygotic transcription early on. So just to kind of sum up here, this is a model that we published a number of years ago. And if we look at and the idea of the model was there's differences between isogenic embryos and hybrid embryos. In isogenic embryos, the maternal contribution here in green starts quite early and is quite significant, whereas paternal contributions on a gene by gene basis, many of them are initially lower and then increase to being equal to maternal, while there are other genes whose paternal alleles are active early on. And the idea of this model is that this trend that you see in isogenic embryos can be deregulated or changed in different hybrid combinations. And so we think that Columbia 2, the hybrid that we used, that Raju Datla's lab used for the transcriptome piling, shows delayed paternal activation and is most similar to isogenic Columbia, where you have on the other end of the spectrum, Columbia CVI hybrids, where they have early paternal genome activation, and Columbia Landsberg, where it seems to vary depending on the gene. So just to sum up, it's kind of a twist on the model that I presented at the beginning of my talk, where, again, it depends on the hybrid, but we think in isogenic Columbia and in Columbia 2 hybrids, there is more maternal regulation of early embryogenesis, but it's zygotic maternal activity, not inherited maternal transcripts that are playing a more important role in regulating early embryogenesis. And so the people that did this work, Gerardo Del Toro started this project when he was a PhD student in my lab. He did the first part of the experiments. Axel Orozco followed up with more functional analysis of embryo-defective mutants. Jaime Alanis, Axel was a master's student. Jaime Alanis, who's currently a PhD student in my lab, has done all the computational analysis. And Daniel Lepe actually worked on another aspect of this project that I didn't have time to talk about. So I'll be happy to, I think maybe I have a little bit of time. I'll be happy to take any questions. Maybe one question.
There's plenty of time. We're running up into a break. So if anybody
right. Okay. Good.
But raise your hand if you have a question, or if you feel more comfortable, you can write it in the chat and we can ask Stuart.
See, there's a number of chat questions here.
Oh, I think these might...
Oh, maybe it's one. Oh, no, sorry. I don't. Maybe there's one chat question from Julian Peña Castro.
If you can read it, Stuart, I've been direct messaging too many
Do you
people.
think these observations may be based on transcription factors and or promoter boxes competition? Well, they're certainly related to transcription. I think they're probably more related to epigenetics and what's going on in the gametes and what's going on after fertilization. And our results suggest that, I guess, like a lot of things in biology, it's less simple and more complicated than it originally was going to be. And so we think that probably in different hybrids, there may be different things going on. And we're really interested in understanding it better, obviously.
I have a question. You were mentioning at the very end that different genotypes behave very differently with respect to maternal and paternal contributions. And I know it's probably a very open-ended question, but you focus so strongly on transcriptomics and developmental biology. How does that tie in back to genetics itself? And is there ties with like hybrid vigor, or how does that connection between the genetics and development play out?
Right. So I would love to have a better answer for you than the one that I have, which is that we, for years, we wanted to actually look at different hybrids to see if there are differences in cell division in the embryo. All these hybrid embryos, it's not like they die. They're all, okay. So Mengshan Sun's lab recently published a paper where they showed that the suspensor length in hybrids corresponded with the maternal ecotype that you used in the cross. That Landsberg has a slightly shorter suspensor. When you cross with Columbia, the suspensor length depends on the maternal genotype. I think actually the most interesting thing that we've discovered is this hybridization effect, because it was quite unexpected for me. And I would like to better understand what's going on. Is it a kind of heterosis that starts really early? That would be really cool. We need to do the experiments. But yeah, I think the hybridization aspect is actually the most significant part of our work.
Thank you. We have a question from Cano Bravo. I'm trying to ask Cano to unmute, but it doesn't look like it's working. Oh, can you unmute, Cano?
Hi, good morning. Yes, my question is there any way to know which will be the dominant gene and the recessive gene in the embryo, or not? Is indistinct?
You mean depending on the parent of origin? I mean, so if you're bringing up haploinsufficiency, that is a very good question. So all of the embryo defective mutants that we use are recessive. Well, they're recessive in later embryogenesis. I didn't show this, but I think I mentioned this in some of the experiments that Axel did. We did find mutants where there were maternal and paternal effects. The maternal effects were always stronger. So that suggests that there is haploinsufficiency also, which basically haploinsufficiency just means that one copy of the gene is not enough to cover the function. And so that's the null hypothesis for all the maternal effects, that is actually haploinsufficiency, but we did quite a few experiments to look at paternal alleles. But yeah, the only way you can determine recessivity of a gene is by crossing with the wild type and looking at the phenotype. That's a good question. It looks like Mario has a question.
Yeah, it's a really quick one. So how you take into account heterosis when you do your analysis? Do you go, let's say, for one generation, and then when you look at the hybrids, and then can you go for a second generation to try to see what's the contribution of heterosis to the phenomena you're studying?
We have never done that experiment because we're always looking, we're taking, we're crossing one. Okay, so to do that, we would have to use a heterozygous plant as a father and cross it with, we've never done that kind of experiment. It might be interesting to do, although it's a little bit tricky how we can conclude, because again, the hybrids we're looking at are just in embryogenesis and really just for about 5 days, although the hybrid plants grow perfectly fine. I'll think about that, Mario. That's a good question.
Let's take the next question in the chat from Takehiro Ozawa. He writes, how is it correlated the hybrid vigor and whether there is a maternal bias effect or not during plant early embryogenesis?
Yeah, so that's what Dan was asking me. And it's sort of a pipe dream of mine is that you could use this kind of analysis because people still don't completely understand how heterosis works and which combinations of isogenic lines are going to be most heterotic. It would be great if you could somehow do an early assay looking at crossing different lines and looking at embryogenesis to see if there was a heterotic effect in embryos. We would love to figure out how to do that. And again, we could do experiments just quantifying the growth of embryos in these hybrids. There's not a huge effect, I can say, because we would have noticed it, but we haven't looked carefully enough to see if there are small effects.
And now we have one last question from the president of the symposium himself, Felipe.
Felipe. Yeah, thank you. Yeah, my question is if there are some paternal difference in the endosperm development at the beginning of this?
Have we ever noticed, like, for example, phenotypes in the endosperm of embryo defective mutants? Is that what you're wondering about?
Yep.
So actually, I had a student, Karina Roscoe, who started looking at that. The pandemic kind of got in the way, and actually her project is to look to see if delayed paternal gene activation also happens in the endosperm. We have some preliminary results that it does using some gene expression markers, and I think Daphne Autran had looked at this a little bit. So we have a little bit of preliminary evidence that there is silencing of paternal alleles in early endosperm development. Also, it's something we're very interested in because delayed paternal genome activation, you could think of it as a kind of just temporary imprinting, and we're interested in if we can separate genome activation and imprinting in the endosperm. So that's one project we're working on, but I don't have a better answer except to say that we're interested in it, and we think there is some paternal allele silencing. Almost all, pretty much all of the experiments that have been done to look at imprinting in the endosperm, to look at genes that are maternally biased or paternally biased, they're all done. I think Gerardo del Toro did one at 4 days after pollination. All the other ones have been done at 6 or after, and that's too late to catch genome activation. By then, at least in the embryo, it's over. So that's something we're very interested in looking at ourselves and profiling gene activation in early endosperms and looking at what's going on in hybrids.
Thank you.
I want to mention to everybody that technically we're on break. Maria Jazmin Abraham Juarez speaks at noon in 10 minutes, but we're on break. So sorry, Stuart, if people are going to keep on asking questions,
okay,
we'll go through them. We have one from Itzel Amasende, and she says, hi, Stuart. What is your opinion about the zygote genome activation in apomictic species? Which lack of parental contribution to the formation of an embryo?
Right. Well, so it depends on the kind of apomixis, but you have some kinds where the egg cell just does the whole thing itself. You have other kinds where you differentiate an embryo from the nucellular tissue that's around the embryo sac. since, well, let's see. That's a really good question. I think it might, what you see might depend on the kind of apomixis you're talking about. If you're just, if an embryo is getting generated from sporophytic tissue, you should already have equal maternal and paternal expression, so there's probably no difference. In the egg cell, good question. I mean, there it's exclusively maternal. And there are results from Sundar's lab showing that you can achieve, with just a little bit of paternal contribution, egg cells to develop. So it might not even be, I guess it's relevant in that in lots of kinds of apomixis, you're getting maternal generation of zygote, and our results support the fact that the maternal genome is a little bit more important, but there are also other studies that show that the paternal genome is absolutely essential to get things going. So yeah, I don't know. It's definitely some relevance there, but I'm not sure. In the big picture, I think it's relevant in that, yeah, there's the maternal genome is more important than early embryogenesis. Just waving my hands here.
Sorry, Dan, just to pitch in really quick. So I want to make a quick reminder that we're sending an unmute request that you need to accept in order to be able to ask a question. Therefore, the speed at which you can ask a question relies entirely on both the speed of your fingers and the speed of your internet. So please remember, first, you need to accept the unmute request, and then you can ask your question. Thank you.
Well, if people aren't getting tired of hearing me talk, someone else has a, Gerardo Valdez Eleuterio says, if you mind, what about transparental plasticity? I don't quite understand what you mean. Like of genes in particular, or epigenetic inheritance, or?
Yeah, perhaps they mean, transgenerational. Oh, I guess it's transparental. I was interpreting it as parental contributions to transgenerational inheritance.
So I mean, it's definitely highly related to epigenetic marks that are inherited from the gametes. I mean, and that's always so. At least in hybrids where you see delayed paternal genome activation, and we think in isogenic Columbia also. So you have paternal alleles are not quite as active early on. The null hypothesis would be that it's just because sperm cell chromatin is so highly packed that you need a couple of cell divisions to reset the epigenetic landscape so that things can take off. That might be true. Actually, I have another slide that I didn't show, but in the Zhao et al. 2020 paper, where they have very nice transcriptomes of apical and basal cells after the first division. There's something super interesting there. So in their transcriptomes, you have the zygote, which they published in 2019. Then they have the apical and basal cell, and then the globular embryo and suspensor, all parent of origin. And we analyzed their data also, and to put it, and they looked at this also, but in a way that I think is a little bit more complicated. What we saw is that in the basal lineage, so the suspensor, the parent of origin gene expression is very similar to the zygote, whereas in the apical cell, it's reset, and you start to have more biallelic transcription, whereas in the suspensor lineage, it stays more either maternal or paternal, depending on the gene. So that's a good example of how you have a change in cell fate. Must be due to epigenetics, probably.
Well, with that, thank you, Stuart, for a great talk. It was a great question session too. And how about we take a well-deserved 7-minute break? Sure. Jasmine Abraham Morez will be speaking in 7 minutes, and see you all soon. Thank you,
Thanks
Stuart.
a lot, Dan. Take care. Bye.
Thank you, Dan. Hi, everyone. Can you see my screen?
Yes, it looks great.
Okay. First, thanks to the committee for this invitation. Today, I am going to talk about novel protein interactions in maize, which reveal a crosstalk between development and immunity. This project has been carried out in the University of California, Berkeley, in EPC and in Langevio Simvestab. First, maize is one of the most important crops in the world, and plant architecture has been studied using different mutants. Here you can see the wild type in P73 background and mutants affected in vegetative development. For example, the ligule mutants and other affected in plant size. Many of them have been very helpful to improve the plant architecture in the field, like the leaf angle and the overall plant size. But some mutants show a different phenotype depending on the genetic background. Here you can see the NOT mutant, which shows a very extreme affected phenotype in the V73, but in the Missouri 17, it is very mild, very similar to the wild type. And presumably, these differences are due to genetic modifiers. Here, I will be focused on the narrow path dwarf mutant. This is an EMS mutant, which shows a pleiotropic developmental phenotype. As you can see here, since very early stages of development, it is smaller compared with the wild type, and in the maturity, the size is strongly reduced, and making a phenotypic analysis, we know that it is because reduction in the cell size and also in the number of the cells in the organs, but also this mutant shows defects in patterning, like in stomatal defects, and also in the ligule formation. Here you can see the ligule in the mutant compared with the wild type. And here in these pictures, I am showing some stomatal defects in leaf development. So these phenotypes suggest that this mutant this gene plays a very important role in tissue patterning at the cellular level. The NOT protein was first identified in the cell number regulator family in maize. And in this tree, I am showing that in grasses, this is a single copy gene, but in the most of dicots, it has 2 copies. And then it was characterized in Arabidopsis. This gene is MCA1 family because it is a mid-complementing activity in Arabidopsis, and it is related with calcium transport and also with mechanosensing. And now we know it is transmembrane protein, but the most of the structure of the protein is in the cytoplasm. And here you can see the phenotypes in Arabidopsis. Even with the double mutant, the phenotype is not so affected like in maize. And also in rice, there's a mutant, but it is only affected in size. But in maize, it is showing many defects related with patterning. So to study the function of this protein, we want to explore the protein interactions. And here you can see that in the public databases, the information about the interactions either in Arabidopsis, the MCA1 protein, or in maize, all the information, the most of information is based on text mining. There is no information based in experiments. So we decide to explore these interactons by using a specific antibody recognizing the NOD protein. We have two different alleles, but only the NOD one is a null mutant. So we use this one for all the experiments as a negative control. Here, you can see the immunoprecipitation of this protein from shoot apical meristem tissues, because in here is where this protein is more abundant. And then we analyze that complex by mass spectrometry. Here I am showing the number of proteins found in 3 replicates. And also in parallel, we made a GIS2 hybrid screening using a library from male and female inflorescence and from the shoot apical meristem tissues. So we identified a group of proteins which are very strong interactors, not interactors based on all of these replicates. And the most of them are related with signaling and immune response. Here you can see the pathway with the proteins we found in that replicates, but many of them don't have orthologs in Arabidopsis or in other model plants. So if we see the same pathway but using orthologs from Arabidopsis, we can see these interesting categories related with endocytosis, membrane trafficking, autophagy, and also immune response and racinosteroid signaling. So focus on the protein kinases found in these categories. Here I am showing the deep-sea experiments to confirm the physical interaction between the NOD protein with these two protein kinases. The positive control is the NOD protein forming homodimers here in the first line, and the negative control is a mutant version of the protein which lost the interaction. And here you can see the signal in the membrane with these two protein kinases, which are GRAS-specific kinases related with autoimmune response or with racinosteroid signaling. Then to test if these proteins are phosphorylating NOD, we used recombinant proteins and in vitro phosphorylation assays. As you can see here, the ligulous SNARU protein is phosphorylating itself, and the negative control is a version of this protein, which is a kinase death. And then we tested the NOD protein with the ligulous SNARU kinase, and we can see here the phosphorylation of NOD. Then to confirm this result, we used analysis by mass spectrometry, and here you can see The phosphosites found in the NOD protein after the phosphorylation reaction and here before the phosphorylation. And the negative control is the kinase dead, ligulous narrow, and the positive is the ligulous narrow wild type. So from here, we know that NOD is interacting with some protein kinases at the plasma membrane, and the ligulous narrow is phosphorylating NOD. But as these two kinases don't have orthodoxy in the other models plants, and we don't have any other information about their function, we decide to explore the function by using transcriptome analysis and phosphoproteome of the ligulous narrow mutant and making the double mutant, not ligulous narrow. Here, I am showing the results about the RNA-seq analysis. And we found that they have a constitutive activation of biotic different responses, which is a characteristic of autoimmune mutants. Also, they have other traits like dwarfism, phenotype variance depending on the background, and also different phenotypes depending on the temperature. Here you can see the very similar phenotype in the single mutants compared with the wild type, and here the most represented categories differential expressed genes from the RNA-seq. And in stress, the biotic stress is very highly represented. And so we want to analyze the changing in expression of the pathogenesis-related proteins. Here you can see upregulation of many of them in the two single mutants. But very interestingly, as I showed you in the first slides, in this permissive background, Missouri 17, the phenotypes are mild. Here, the NOD mutant is very similar to the wild type, and here the ligulous narrow mutant, which is very affected in the ligule formation, is almost not affected in the Missouri 17 background. So, as you may know, this phosphorylation in MAP kinases 3 and 6 is a classic marker to identify biotic stress induced by PAMs or DAMs. And here, many reports have shown rapid induction with PAM treatments like flagyellin 22. Here at the 5 minutes or 5 to 10, we can see very high induction of this phosphorylation, these proteins. So also these reports are from Arabidopsis analysis, and also some reports have shown even we can separate PTI from ETI effect using inducible systems, but also we can see the very fast response with PAMS induction. So in maize, we used the same antibody used here in Arabidopsis to test our mutants in these phosphorylations. And here in B73 background, you can see a very high induction of this level, but it is not in the Missouri 17 permissive background. So we have the question of about if this PTI is inducing in the V73, but is not in other backgrounds. And which makes a thing about genetic modifiers present in the Missouri 17, but not present in other backgrounds. Related with that result, Alisa Anderson published in 2 years ago the identification of one ligulous narrow modifier in Missouri 17, which was called sympathy for the ligule because it rescues the ligule defect in the mutant ligulous narrow. And then we found that this modifier is an ortholog of the enhanced disease resistant 4 from Arabidopsis. And like this one, when plants are treated with a chitin, the transcriptional induction of the gene can be observed at a very rapid, like 30 to 60 minutes. And this response is very similar to the pathogenesis-related genes. But also, this mutant, the ligulous narrow, is rescued either by growing at low temperatures or having the version from Missouri 17 of this modifier. As you can see here in these near isogenic lines, the phenotype is very similar to the wild type in the low temperature, and in the high temperature, this near isogenic line is recovered compared with the mutant. So then, with all the information we had about the transcriptome and the phosphoproteome in this mutant, we have this model which is showing that ligulous narrow is a negative regulator in an immune signaling cascade, and also it is affected by temperature, and also is affecting the development. But in the mutant, a MAP kinase cascade is activated, leading to the PTI response, which can be dampened either by low temperature, cool temperature, or for the presence of the Missouri 17 modifier sol. Also, it is all of this information we are generating is related with the hypothesis about the ligule involvement in development and in immunity, which established that a phosphorylation brassinosteroid signal propagates from the ligule to the leaf margin, and it is affecting the shape. And this signaling cascade is involved in establishment of developmental patterns and also in immunity. From other mutants affected in genes related with brassinosteroid signaling, and also with these mutants we are finding in maize, we are helping to make strong this hypothesis. And now we have more mutants because now already we have the double mutant and not ligulous narrow, which you can see here in this picture. This is in a different background. This is in A619, which is a mirror phenotype between the very severe B73 and the mild Missouri 17. But the double mutant is strongly affected, as you can see here in the pictures. And here in this plot, you can see the height of the plant and the size, the leaf size. And here in these red dots, you can see the double mutant. And also, it is very affected in the ligule formation compared with the single mutants or with the wild type. And the RNA-seq analysis is showing an even stronger induction of immune response in this mutant. And now we already have the specific antibodies recognizing the ligule narrow protein and also the SOL, which is the modifier. And now we are making another experiments exploring the proteome and the phosphoproteome, and also more interactions, protein interactions, to try to find what is the molecular function of these protein kinases, and also the modifiers found in the permissive backgrounds. In summary, these results suggest that node and ligulus narrow are novel developmental regulators in a crosstalk with immunity. Also, that MACE mutants are a powerful tool for the identification of new protein interactions pathways in crop species. And these novel protein interactions may contribute to the identification of effectors in crops, regulating the development and also stress response. I would like to acknowledge Sarah Heck from the University of California in Berkeley, who is a very important part of this project, and also Alisa Anderson, who was working with the modifier, and Jacob Brenker from the University of Wisconsin in Madison, who is now helping to understand the results about the RNA-seq and the phosphoproteome, and also funding, which is very important for this project. Thanks. If you have any question, please ask.
Thank you, Jasmine. Great talk. We have plenty of time for questions. Our first question comes from Arturo Guevara Garcia, and hopefully you can unmute yourself, Arturo.
Thank you very much, Chris. Nice talk, Jasmine.
Thank you.
In the phosphorylation assays that you showed, what do you know if phosphorylated form correspond to MPK3 or MPK6?
We still, yeah. In the in vitro phosphorylation assays, we only use our interest proteins, the node and the ligulus narrow. And the that you mentioned is using a specific antibody recognizing the phosphorylation in the MAP kinases 3 and 6. And this is a generic antibody used in animals and in plants because this is a very conserved response in stress, mostly biotic stress. So we haven't done any in vitro phosphorylation with this specific MAP kinases.
Oh, okay. Thank you very much. I know the functionality of the antibody, but my question is, do you think that could be important to know if it's MPK3 or MPK6 or doesn't matter?
Oh, in this point, it is not very important to distinguish between them because in other species have been shown that both of them are induced, the phosphorylation of them are induced. So I think it is not specific. We cannot say which one is induced, only the mix of them, because we are not very sure which one it is.
Do we have more questions for Jasmine? Well, more people are asking. I have one, Jasmine. You show so clearly that these mutants are so important for the ligule and for the development, the proper development of the leaf. And they have these connections to disease and to heat and stress. Do you think that the changes in leaf shape are adaptive with respect to disease and stress, or is it just a developmental constraint or consequence of how these pathways just happen to be?
I think they are mostly related with development, in changes in development and patterning, and the connection with immunity is a parallel function, which in maize maybe it is important to propagate the signal from the ligule. Because, well, until now in maize, we don't have very much information about immune response. And so these proteins, the working in the ligule, are first changing the architecture of the plant. But then also related with immunity, which is not still very clear, how it is working with the adaptation.
I see, thank you. We have a question from Naftali Cruz. Let me unmute you. You should be able to unmute yourself, Naftali.
Hi, Jasmine, very nice talk. Thank you very much. And my question is, if you have studied the phosphosites of Ligulus narrow, and if you have complemented with, I don't know, phosphomimetic and phosphodead alleles of this protein, and if it could complement the phenotype of the mutant that you have. I don't know if you have considered these type of experiments to understand the role of the phosphosites that you identify by mass spectrometry.
Sure, you are right. These phosphosites are very important to study using phosphomimetics, but we haven't done yet. We only know some of them are in vivo phosphorylated because based on the phosphoproteomics data, and also in the North mutant, some of the affected phosphosites are changing the shape of the protein because they are also affecting the interaction with itself or with other proteins maybe. So in the future experiments, of course, we are planning to analyze the phosphosites using phosphomimics.
We have a few more minutes. Any more questions for Jasmine? Just one last question, Jasmine. You talk a lot about steroids and it's the Ligulus narrow. I'm thinking of like the work of Mike Scanlon and the growth of the maize leaf margin and how, and a lot of times it's like adaxial, abaxial, but there's a lot of evidence that like this margin outgrowth is independent of adaxial, abaxial patterning. Do you think it's, yeah, how do you interpret the lack of blade outgrowth in some of your mutants?
Yeah, the ligule is not affected in all the blade. In this Ligulus narrow mutant, it is affected only in the in the middle of the leaf. So that's why the hypothesis says that this signal is propagating from the center of the leaf to the edges. So we also have another mutants like the GUAP1, which is affected in abaxial, adaxial auricle structure, and they are now used for making double mutants with the ligulus narrow to try to elucidate what is happening with this patterning, establishing the wave in this structure. But we don't know yet.
Thank you. Fascinating work. Thank you, Jasmine. And we should get on to the next speaker. So Mario will take it from here.
Thank you.
Thank you, Dan. Okay, so our next speaker is José López Bucio from the Universidad Michoacana de San Nicolás de Hidalgo here in Mexico. The title of his talk is Cell Death and Regeneration Within the Root Meristem. Jose, if you're ready, could you please share your screen with us?
Well, thank you, Mario, for your kind presentation. And I'd like to thank the organizers for the opportunity to be here. There are several reasons to study plants, particularly plant roots. Here we have some of the functions that these organs play. And you see that they're very important for ensure the photosynthetic and reproductive tissues to the soil, to the substrate, and also to take up water and nutrients. The photosynthesis process, in fact, makes sugars and other nutritional compounds for microorganisms which inhabit the rhizosphere, the part of the soil which is in close contact with the roots. And in this manner, the roots support the most of the plant microbiome. And by all of these characteristics, the roots have been considered a major target in the search of the next green revolution. In fact, in order to perform this, all these functions, the root system is always producing new branches. A single plant may have, in fact, thousands of root branches performing all of these functions. So every root has a meristem. What is a meristem? Well, I think the very basic property of the meristem is the cell division process, which is continuous and which happens over the entire life cycle of the plant. So you can see here a few photographs of the meristem, in this case of Arabidopsis, where you can see the mitotic cells, okay? And this region is also related to auxin gradients, shown here in blue color. These orthogonal gradients, in fact, are responsible of the identity of cells within the primary root tip, okay, shown here. Here we have the root cap. 4 cell layers of columella cells are protecting the meristem, located upstream. And inner within the meristem, we have the stem cell niche. But what is the stem cell niche? Well, we have a magnification here. You see that there is a quiescent center, a few cells, which are somewhat mitotically inactive cells. Around the quiescent center, we find most initial cells which proliferate and produce all tissues that take part in the mature root in this case. So the meristems are very interesting zones regarding the basic cellular programs, cell division, cell differentiation, cell elongation, and also cell viability, because these cells are involved in rapid DNA replication. And you know that DNA replication is always presenting a problem, because the cells are exposing the DNA, and there may be opportunity for mutations. So the meristems are regions that are very sensitive to environmental stimuli, particularly those related to genotoxic stress. Okay, so there are many environmental stimuli which can modulate the shape, the branches, the length of the root system of plants. Here we have, in order to compare these strategies to grow, we have the normal indeterminate growth of the primary root, which makes a long dominant axis. In the case of Arabidopsis, this is named the primary root, the first structure, what is produced after germination. But in the case of limitation of phosphate, you see that there is no more growth in the primary root, and the growth changes. The root indeed forms many branches, and the root system is believed to be more efficient in the take-up of phosphate, particularly from the upper soil layers. So there are other stimuli, such as, for instance, molecules, secondary metabolites, volatiles, which are produced by microbes, and also by stressors, abiotic stressors, such as salt, pollutants, metals, to mention a few. So there is a very huge developmental plasticity, just shown here, and this always depends on the activity of the meristems. Here we have a normal meristem which is proliferating, which is showing the hormonal gradients around the root tip. But when the primary root encounters problems, there is a change to the terminate growth. The mitosis is inhibited, and then there are there is a stimulation of the differentiation process, which gives rise to the formation of the root hairs or lateral roots. Here we have a very nice comparison of the structure of the meristem. Here we have a meristem in a plant which is growing with a high supplement of phosphate, a macronutrient, an essential macronutrient. And we have here the same region, but in this case, in a plant that is growing under limitation of phosphate. So you can see here that we have no more meristem here. We have no more cell division here. We have differentiation. We have production of root hairs. We have differentiation of the vascular tissue, which approaches the very root tip here. And we cannot see anymore the root cap or the columella cell layers. So this happens just by changing the availability of an essential macronutrient within the soil. So we have been working in this topic for several years, and we recently started a screening in order to try to identify genetic factors that are involved in the growth of the primary root. In this case, using Arabidopsis and in vitro systems, where we can control the availability of phosphate in the medium. And you see that we have a mutant which is named HPR1, which refers to the inhibited growth in the primary root. Here we have a comparison of the growth of the primary root between the wild type parent and also the HPR1 mutants. You see the constant growth of the primary root, and here we have the inhibition in the growth of the primary root in the mutants. When we check the phenotype of the roots, we have here the root tips. In the case of the wild type seedlings, we have the meristem, the cell elongation, and differentiation regions. But in the case of the HPR1 mutants, we cannot see the meristem or the cell elongation region, and instead we have root hairs covering the root tip and also lateral roots. In fact, when we observe the phenotype of the lateral roots, we again see these changes. In the mutants, we have affective meristems in the lateral roots. These structures, in fact, look like callus-like structures instead of normal lateral roots. So as auxin is important for the activity of the root meristem, we tested whether there will be an alteration in the auxin response in the mutants. And to answer this question, we performed experiments supplying very low concentrations, increasing concentrations of indole acetic acid. You see the location of the auxin response at the primary root. And in the wild type seedlings, which express the DR5 reporter gene, we see that there is an increase in the expression as the concentration of auxin in the medium increases. In the case of the mutants, we can see that we have two different expression domains. The first one that is shared with the wild type seedlings, and a second one here, which is located in provasculature cells. And upon addition of auxin, you can see that there is a magnification in the response, particularly at this second auxin maximum within the primary root. And this also correlates with the formation of root hairs with differentiation processes at the very root tip. So what is interesting is that when we compare the structure of the primary root in the wild type, you see 5 days after germination or 10 days after germination, the structure of the meristem is comparable. We have here the root cap, okay, and the meristem here. But in the mutants, you can see that 5 days after germination, we have an almost normal structure of the meristem, and later on, all the root tip is completely disorganized. But if you observe this region, okay, where the second auxin maximum was observable, we see that there is a red patch. This red color here is produced by propidium iodide staining. Propidium iodide, in fact, can penetrate damaged tissue. Those cells which are dying receive the propidium iodide, and therefore we see this red patch, which is indicative of cell death or damage. This damage was also present in lateral roots, as you can see here, and this correlates again with the transit from normal meristem to distorted abnormal meristems. So this was an indication that there was a problem with the cell division that later on is correlated with the inhibition of the growth of the primary root. So to try to support this hypothesis, we isolated mutants which manifest not really determinate growth, but instead the primary roots. The primary roots grow slowly in these mutants, and this slow growth is sufficient to induce the formation of branches, and the branches also grow very well, as you can see here. So the phenotype is not exactly, is not just comparable to the HPR1, and these mutants are defective in the mediator 18 subunit of the mediator complex. What is the mediator complex? Well, this is a transcriptional coactivator which plays a critical role in the transcription of genes in eukaryotes, from yeast to humans. The mediator complex helps the RNA polymerase II to find out the promoters and to start the transcription of genes. So this particular subunit is critical for the configuration of the root architecture. When we tested the phenotypes at several days after germination in the normal plants and in the mutants, you see that in the case of the mutants, we have again the same problem. We have this red patch showing the cell death area, and this problem is exacerbated with time. By 7 days, when we have fully photosynthetic leaves, this light signaling is part of the exacerbation of this phenotype in both the primary roots and also in lateral roots. In fact, this was published recently this work was done, in fact, mostly by Javier Raya in our lab. And this was correlated with the DNA damage response, this particular cell death phenotype. So by the same time, we were working, trying to understand the effects of environmental pollutants on the activity of meristems. A few years ago, we reported in a nice collaboration with Miguel Martinez in order to understand how chromate could be affecting the growth of the primary root in Arabidopsis. It has been, it is thought that chromate is a very toxic pollutant. It is accumulated in water bodies, particularly in industrial zones. So when we tested the effects of low concentrations of chromate, 20 micromolar, for instance, we see that there is an stimulation of growth, and higher concentrations, we see a strong repression of growth, as you can see here. It was of interest to see that the repression of growth was related to auxin signaling, because auxin mutants, which are insensitive to auxin, in this case the solitary root 1, this is an auxin repressor, and the mutation in solitary root is dominant, so we have here a strong repression in the auxin signaling. As you can see here in this comparison, in the wild type, we have no growth, because we are growing the plants with a very high toxic concentration of chromate. But in contrast, the solitary root mutants grows very well. As you can see here, the roots are growing very well, and later on, they are inhibited by, there is a much more concentration of chromate needed in order to have this growth repression. So the surprise came here. We expected actually that if the MET18 mutants have a problem in the meristem, their cells are dying. So the hypothesis was that if we provide chromate to these mutants, there will be an exacerbation of the cell death phenotype. This, in fact, was not the case. We compared side by side four plants, mutants, and 4 wild-type seedlings side by side in medium without chromate. And when we add 60 micromolar of chromate, we see that there is a 50% inhibition in the primary root growth, okay, in the wild type, and then there is a further inhibition in the mutants. But when we check the primary root tip in the mutants, surprisingly, we did not find a single primary root. We find a couple of root tips, which under the confocal microscope looks practically identical. So it seems that chromate triggered the process to convert a single meristem into a twin meristem through a process of splitting the pattern root meristem. So this was a very nice opportunity to investigate how can a single meristem be converted into twin meristems. And we checked by several genes involved in the cell death process. This is the ethylene response factor 115, which, as you can see, is very nicely expressed at the center of the meristem, where the cell death takes place in the mutants, but not in the wild type. And in the case of the mutants, you can see that supplementation of 60 micromolar of chromate at several times after treatment with chromate, you see that 3 days, 4 days, 5 days, until 7 days, and you see that at all stages, there is a marked expression of this transcriptional factor until we have the couple root tips already formed in the previous region occupied by the meristem. So the quiescent center is a very nice marker of the stem cell niche. You see the quiescent center here in the control in normal plants, and you see also the red patch here in the mutants and the location of the quiescent center. What happens with the quiescent center is when we supplemented chromate, we see the formation of a withered primary root, of course. And by this stage, 5 days after germination, we see the formation of two opposite zones where the quiescent center is located. And later on, you see the twin roots with each with its own quiescent center already formed. When we analyze the auxin response, we see the comparison in the control and in the mutants. We see that there is an increase in the response, in the auxin response in the mutants. Here we have the red patch. And the auxin response is very dynamically controlled during the process of twin root formation in the case of the mutants. By this time, you can see here the formation of a pyramid-like structure, okay? Later on, you see the separation of the auxin streams at the center of the root. And later on, you see the formation of both twin roots. So these results suggest that there may be an important role of auxin in this organogenesis process. And we have the advantage in the case of research with auxin that we have at hand inhibitors in order to check whether auxin transport will be important or not in the transition from a single meristem into a twin root. So we performed the analysis using the DR5 gas reporter gene and applied 60 micromolar of chromate. We see nicely the formation of the twin roots in the mutants. But when we supplied 4 micromolar of NPA together with chromate, we did not see the separation of the twin roots. In contrast, you can see the very different phenotype of these roots when compared with this one. All pictures are at the same magnification. You see an increase in the expression domain of the DR5 reporter here. This is a very wide meristem. We have very wide roots, and we have the formation of long root hairs along this new structure, which is formed after application of NPA and chromate. Of course, we have an induction of the cell division process and also the coordination with differentiation processes, which will give rise to this change in the root phenotype. This was also verified using a different reporter gene, the cyclin B1, which is an excellent marker of the transition from the transition to mitosis. You see again that we have the formation of the twin roots here. Later on, when we supply NPA with chromate, you can see the meristem. Just compare this meristem with this one. Of course, we have perhaps 10 times greater meristems here just by supplementation of chromate. And you also see that the root is very robust when compared with this one. This might be an interesting model in order to check or to analyze organogenesis. Okay, we don't know if we have more cell layers forming here, or we don't know what is the identity of these cell layers. And this is, I think, an interesting research to perform in the next weeks. So, as you remember, we initially identified the solitary root as an important genetic factor in the response to chromate, in the response to the inhibition of the primary root to chromate. So we made the double mutant, in this case, the MED18 and the solitary root double mutants were generated. And you see here, supplementation of chromate induces the twinning of the meristem. And when we checked this in the solitary root mutant and the MED18 double mutants, you see that there is no transition from the single meristem into the splitting that gives rise to these twin roots. So this confirms, in fact, that auxin is very important by both strategies, either pharmacological using MPA, and in this case, genetic using the solitary root mutant. So what is the importance of this organogenesis process? Well, I think that this is a curiosity, in fact. There are a few reports in the literature that have been related to the formation of these twin roots. This is in fact called dichotomous root branching. A few families in plants, particularly in Selaginella, in the genus Selaginella, and in the bryophytes, the root branching process, in fact, occurs in this manner by formation twin roots. So this is one of the first cases in angiosperms, in this case in Arabidopsis. And it will be of interest to check whether this process is also or has an important adaptative advantage in the conditions of phosphate limitation, nutrient limitation, or perhaps the presence of toxins around roots, or perhaps as a response to conditions in the environment that impose a strong stress to plant roots. When we transfer the plants that formed the twin roots to chromate-free medium, you see that these roots are able to grow independently, which indicates that their meristems are functioning in an adequate manner. So I think we are close to finish this talk. Let's see. Okay, so just a summary. I don't want to repeat the main results that you have seen. Just that the chromate seems to be an important factor in the organogenesis process, which in conditions that impose genotoxic or cell death pressure to tissues, we can have the transition from cell death and for development of this beautiful process of twin root formation. Of course, this is at the center of cell regeneration. Hopefully, something like this process will be happening in animal or humans in order to try to test whether we could regenerate complete organs by just applying treatments in cell cultures or cell tissues in laboratory conditions. Well, just to thank the people to have supported this project, many students, particularly Javier Raya, and Miguel in the case of chromate, Professor De Beilder, Luis Herrera, and Arturo Guevara also supported this research at several stages. And the financial from several institutions are also presented here in order to recognize the important support to this research. So thank you so much for your attention. If you have some questions, I will try to answer them. Thank you so much.
Thank you, Jose. We have time for one quick question. Please, you can unmute now, Professor Dubrovsky.
Yes, thank you. Jose, thank you very much. I have a question related to stability of this phenotype. Let's say what is the percentage of seedlings you have this dichotomy, and whether this dichotomously branched apex can repeat this dichotomous branching after it's already formed, and whether lateral roots also are capable to do that.
Well, both questions are interesting. The first one, I think the process is very reproducible, as you could see. This allow to check several genes, several markers during the transition from the single meristem into the splitting of the parent meristem. So the percent of the roots that start this program upon supplementation of chromatin the mutants is higher than 90%, close to 1,000%, I think. So it is very reproducible. The other question is, well, we don't know also how this dichotomous root branching process influences the lateral root formation program. We don't know. This is, I think, a very interesting question to follow, and we need to check this.
Right, but lateral root can branch also dichotomously?
We don't know, Professor Dubrovsky. We don't know. We have just focus at this stage of the project. We have just focus on the primary root. Okay. We have to check it.
Yeah, thank you, Jose.
Thank you for your question.
Thank you so much, Jose. That was a wonderful talk and really interesting. And thank you so much.
Thank you, Mario. Thank you all.
Okay, moving on to the next talk. Our next speaker will be Nayeli Marsh-Martinez from Symvestav and Irapuato. And the title of her talk is Dual Cytokinin Regulation by Transcription Factors that Control Development. This talk is a recording, so give us 1 sec to start the recording.
Hello, I am Nayeli Marsh. I work at the Biotechnology and Biochemistry Department at Symvestav Irapuato in Guanajuato, Mexico. Thank you for connecting today to this talk. Also, many thanks to the organizers of this nice symposium for the invitation to present our work. This time, I want to talk to you about the role of the transcription factor no-transmitting tract, also named WIP2, as a connector of hormonal pathways. I want to start thanking the people that were involved in this work that was mainly done by David Diaz in collaboration with more people at Symvestav. Here we see Yolanda, Rosa Esmeralda, Eric, and Humberto, and more collaborations at other institutions. This work has not yet been published, so please treat the information as so. The role and importance of hormones in many plant processes is well established. From them, auxin and cytokinins are considered to be two of the most important hormones that guide plant development. They have been considered to have an antagonistic relation. However, as more research has been done in the last years, it is clear that they act more in a complementary way. I really like the title of a review that was published in 2015 by Schaller and collaborators, where they compared the cytokinin and auxin interactions with the concept of the yin-yang, because it reflects the interconnections and complementarity of these pathways. At the end of each pathway, that includes biosynthesis, inactivation, transport, perception, signaling steps, there are transcription factors that regulate the transcriptional response to each hormone. For auxin, these are auxin response factors. And so there has been a lot of research There's still also a lot to understand about what is downstream of them. We work in the lab with a different kind of transcription factors. This is one of them, WIP2 or no transmitting tract. It can remodel plant architecture when misexpressed, as you can see here. It belongs to a family of 6 members in Arabidopsis, characterized by the WIP domain that starts with these 3 amino acids and contains 4 zinc fingers. In Arabidopsis thaliana, the lack of entity function causes the loss of the transmitting tract. This is a tissue inside the pistil that is necessary for proper pollen growth to reach the ovules. Therefore, mutants present this phenotype of fruits that are not completely full of seed. Entity is also involved in replum development. This is a tissue that divides the valves of the Arabidopsis fruit. We have studied some of the mechanisms of entity action, finding that it interacts with different regulators that guide gynecium and fruit development. Also, together with one of them, called seed stick, entity regulates genes involving lipid metabolism and transport and cell wall remodeling. Here we see the phenotype of the loss of function of one of these genes that produces a very striking gynecium phenotype. Because of this phenotype, Humberto named this gene Kawak, that means storm or thunder in Mayan. Entity has also been found to play a redundant but key function in root development. The triple mutant of Entity or WIP2 with its closely related genes WIP4 and WIP5 named NWW does not develop properly. What happens is that in the embryo, the first division of a cell called the hypophysis does not occur properly. In a wild-type embryo, the hypophysis divides asymmetrically and gives rise to the root apical meristem. But this does not occur as it should in the NWW triple mutant. The authors of this work noticed that this defect was similar to another mutant called Monopteros, here indicated as MP, and that while entity was expressed in the cells that give rise to the root apical meristem in a wild-type embryo, it was absent in the Monopteros mutant. They found that entity was a target of Monopteros, which is an auxin response factor, ARF5, indicated that entity acts downstream of the auxin pathway. If entity is downstream of the auxin pathway, which genes are acting downstream entity? We wanted to have a broad view of these genes, and for that, we used the inducible line 35S entity GR, where we can induce the entrance of entity to the nucleus when dexamethasone is applied, to compare the transcriptomes of induced versus non-induced plants. When entity was induced, as we had seen before, leaves developed in a very different way than wild-type plants, producing very clear serrations or even a sort of projections at their margins. Here we are looking at induced plants plus dexamethasone and without dexamethasone, and here we are looking to constitutive overexpressors. This leaf phenotype reminded us of plants that have been treated with cytokinins, and though the phenotype is not exactly the same, we thought that it was worth to explore whether these hormones could be related to entity. Root development was also affected when entity was induced. As you can see here, the root presents a severe reduction in growth after induction. The blue arrow indicates the length of the root at the moment of induction, either with a MOX solution, when the plants were treated with a MOX solution, or with dexamethasone. The green arrow indicates the length of the root 3 days after the treatment, and as you can see, there is a clear reduction in growth, which is quantified in this graph, comparing also wild-type plants that were treated with or without dexamethasone. You can see the severe reduction here. Interestingly, Root growth is also affected when plants are treated with cytokinin. NTT is not only expressed in the gynecium or the embryo, it is also expressed at the vegetative stage in the vasculature of different organs, as we can see here, the leaves, hypocotyl, roots, and in this line we also see expression at the shoot apical meristem region and at its incipient lateral roots. And because these are all different tissues, we wondered whether it could be regulating different genes depending on the context in which it is expressed. For the global expression analysis, we decided to separate the aerial from the root tissues, also to reduce the problem of masking differential expression due to the mix of tissues. We were also very interested in finding early and somewhat late downstream genes. For this, we took samples 30 minutes and 8 hours after induction, and for the samples taken at 30 minutes, we also added cycloheximide, that is a translation inhibitor to reduce the secondary effects of other genes after NTT induction. We gotten data for 4 comparisons, roots and shoots at 30 minutes and 8 hours. And we found that a different number of genes had changed expression depending on the tissue and the time. We also analyzed enriched geo categories. Here I only show some hormone-related categories. We found that some were present in all the contrasts, but others were only found in some of them, as occurred also with many genes. We can see this clearly in this heat map depicting auxin-related genes. Because NTT is a target of R5 or monopteros, we decided to look first to auxin-related genes and found that genes that participate at different steps of the pathway were differentially expressed. Most of these genes presented moderate changes in their expression. And actually, the genes that had the most change in expression were genes that are known to be early auxin response genes, such as GH3, that are genes that code for enzymes that conjugate auxin to amino acids, small auxin upregulated RNAs, also called SAURs, and the ARF-negative regulators IAA-OX. Since the auxin and cytokinin pathways are often interconnected, and because of the NTT phenotypes that we had observed, we also looked to cytokinin-related genes. Again, genes involved at different steps of the pathway were affected. So genes that code for enzymes that are involved in cytokinin metabolism or for cytokinin transporters, and also components of the signaling pathway, an important cytokinin receptor, phosphotransfer proteins, and both the transcription factors that regulate the response and their negative regulators. Some of these genes had higher changes in expression, like this, for example, or were affected in at least 3 of the conditions that we studied. Therefore, we chose 3 genes to study further. One of them is involved in cytokinin biosynthesis. This is the IPT5 gene. One involved in cytokinin degradation. This is CKX7. And the negative regulator of cytokinin signaling, AHP6. Both AHP6 and CKX7 would have a negative effect in the cytokinin pathway, while IPT5 would have a positive effect. First, we wanted to confirm the changes in expression of these genes after NTT induction. For this, we crossed the inducible line to marker lines. When we crossed the inducible line to an IPT5 Gauss marker line, we observed that Gauss staining increased after NTT induction, as you can see here and here. Conversely, the induction of NTT in the cross of the inducible line to an HP6 GFP marker line caused a reduction in AHP6 expression, and this was coincident with what we found in the transcriptome data. As we had seen in the crosses and in the transcriptome, qRT-PCR analysis confirmed further that AHP6 expression decreased 30 minutes and 8 hours after NTT induction. On the other hand, IPT5 increased expression 30 minutes and 8 hours after NTT induction, and something similar occurred with CKX7, which rapidly and strongly increased its expression after NTT induction. After observing the changes in expression of these cytokinin-related genes, we wanted to know what was the effect of NTT induction in the cytokinin reporter line TCSN-GFP. For this, we crossed this reporter line to our NTT inducible line. We found that the induction of NTT altered the expression of this reporter line, as we can see here. When we analyzed the root apical meristem of the main root 2 days after induction, the changes were very subtle. But one week after, all TCS signal was lost in induced roots. However, GFP signal was still detected in emerging lateral roots of these same plants. And when we looked a little bit above the root apical meristem of the main root, 2 days after induction, GFP signal was detected at the region above it in induced plants, in contrast with not induced plants, where the signal was not detected. It also seemed that the size of the meristem was affected. The mature region of the root also showed GFP signals that were not present in the non-induced plants. Therefore, we concluded that NTT induction caused alterations in cytokinin signaling. It was clear that the expression of CKX7, IPT5, and AHP6 was changing when NTT was induced. So we also wanted to know whether NTT was able to bind regulatory or genomic regions of these genes. We used different fragments, either complete promoters or smaller fragments to test this. First, we used a yeast-1 hybrid assay. And while some fragments could not be used because of autoactivation, the rest produced positive results, as we can see here. We also tested the regulation of NTT in these genes using a bioluminescence transactivation assay. This picture was actually taken in one of those experiments. We tested the promoters alone, as indicated here, and in combination with NTT, and found that NTT was able to regulate the expression of these 3 promoters in the same sense as we had found for the transcriptome or the crosses or the qRT-PCR experiments. Finally, since CKX7 is one of the genes that was activated the most at the early time, we tested whether it played a role in the misexpression phenotype of NTT-induced plants. For this, we crossed the inducible NTT line to the CKX7 loss-of-function mutant. Here we see both the inducible line in the wild-type background and in the mutant background treated with a MOX solution. They actually don't seem very different from each other. However, when induced, while the inducible line presents again the serrations or these projections in its leaf margin, these serrations or projections are lost or are very attenuated in the CKX7 mutant background, indicating that this gene is very relevant for this phenotype and suggesting that cytokinin homeostasis plays a role in it. We also treated these plants with cytokinin, and they responded also differently. You can see the big difference in the shape of the leaves of these plants. In a way, this was expected because CKX7 is a mutant that is impaired in cytokinin degradation. Therefore, we would expect that it's more sensitive to cytokinin treatment. Regarding the roots, the induction of NTT in a CKX7 mutant background produced a new phenotype, the development of Kali along them, as you can see here. This was not observed before in the wild-type background. And when cytokinin was added to the induced plants, Kali developed also in the inducible line in the wild-type background, suggesting that the excess cytokinin that results from CKX7 inactivation plays a role in the formation of this Kali. This also suggests that there are other genes that are being regulated by NTT that are able to trigger the formation of Kali when CKX7 is not present. Based on the results that I just presented to you, we have concluded that NTT connects an auxin input signal through R5 or monopteros to a cytokinin output through the regulation at different steps in the pathway, as we can see here. Here is our working model, where auxin input or other inputs will affect the activity of R5, also called monopteros, which in turn will regulate NTT. And downstream NTT, besides other genes, we can find different cytokinin-related genes that will affect this pathway either in a positive or negative way. We also found many other genes in the transcriptome that are worth being further studied. And therefore, the regulation of NTT in the cytokinin pathway is complex, and it seems to affect cytokinin homeostasis at different levels. Well, this is the end of the story for the moment. Of course, there are many questions open now, and we would really like to keep on working on them. Thanks a lot for listening to this talk. Also, thanks to all the people that collaborated in this work that were presented at the beginning. And I would also like to thank Symbestab, the institute where I work, CONACYT, who provided the funding for this work. And recently I started participating in this international consortium with the European Union. Please let me know if there are questions. I will be happy to hear them and try to answer them the best I can. If there is no time, you can also write to me. I put my email in the screen. Thanks again.
Excellent talk, Nayeli. We have plenty of time for questions.
Thanks a lot, Dan. Thanks to everybody. Sorry, I was worried that my internet would not work, so I prefer to record the talk.
Seems like it's working great now. We have a quick first question in the chat from Luis Cardenas. NTT overexpression seems to induce big trichomes in the leaves. Is this correct?
Yes, yeah. We didn't look further to it, but yes, yeah. You have very good eyes. Yeah.
Anybody can raise their, we have one from Professor Dubrovsky. Let me unmute. Can you talk?
Yes. Nayeli, thank you very much. Very interesting talk. I would like to ask you if you have verified in CKX7 mutant background when you have this callus formation, is that related to lateral root primordia or it is independent?
We think, we have not checked very carefully, but we think it's related to lateral root primordia. We think that the lateral roots, instead of becoming lateral roots, that they start to develop as Kali. But we need to look, more carefully to that. From the position, it seems that they are lateral roots that change.
Okay.
But in the case when we add cytokinins, then the whole root starts to become a callus.
Oh, okay. Okay. Thank you very much.
Thank you.
Any more questions for Nayeli? I have one also looking at your leaves. I love leaf shape, of course, but the serrations are just so prominent. And in your overall model, you start with auxin and the ARFs, but then it goes to cytokinin and things like cali. And it seems like when I think of auxin and ARFs, I think more of like actual patterning of serrations. Whereas when it goes to like cytokinin and callus, I start thinking more of like meristems or like compound leaves. What is your interpretation of those like serrations or like leaflets?
Yes, I always thought they were serrations, but recently I have started to think more as leaflets. Actually, I didn't put all the genes that we found in the transcriptome, but we also found COG genes, for example. So then, yes, maybe they are starting to become kind of leaflets. And even sometimes in the leaf lamina, we have seen some cases in which you also get some growths there. But no, we have not really used markers, for example, to see what is that. But the presence of the COG genes suggests that it may be more related to, like a small leaflet. On the other hand, I don't think this is the only thing that NTT does. Probably it's orchestrating very different processes at the same time, and this is only one of them.
Thank you. And Mario, we'll get to you, but let's first go to Patricia. Can you unmute, Patricia? Try it now.
Yes, I think I can now. I have a question, Natalie, about the serrations and the leaf shape. Do you know if any environmental conditions such as light might change the serrations phenotype, or is completely independent of what light conditions have? Do you know something about this?
Thanks, Patty. Thanks a lot for the question, because actually we do see changes. So sometimes there are super pronounced serrations and sometimes they can be just undulations. This depends more on the line, on the transformant itself. But actually we have never checked if it also depends on the light conditions, for example, and thinking about cytokinins, it's known that actually cytokinin signaling is affected by light. So actually it would be very interesting to check this in the transformants in the inducible lines.
And next we will go to Mario.
Thank you. Thanks, Nayeli. That was a really good talk. I have a quick question regarding, again, about the shape of the plants. So the shape of the leaf also reminds me about some classical small RNA mutants, just like serrate and some other, because you not only have serrations, if actually those are serrations, but also the shape, it's kind of narrowed, like in RDR6 mutants. And have you looked for small RNA defects in your plants?
Thanks, Mario. Very, very interesting question. A long time ago, we started to do some crosses. We also thought they were related. Now we focus more in the transcriptome. We did not see something so general. I think now maybe it's more related to the COG genes that can also be regulated by microRNA. So maybe that's what it's affected. But I think we should look closer to our transcriptomes in any case. I think it's a very nice suggestion. Thanks.
Thank you, Nayeli. A beautiful talk and great questions, everybody. We move on. Mario will introduce the next speaker.
Thank you, Dan. Okay, well, it is now my great pleasure and honor to introduce our second and last keynote speaker of the day. We have Roberto Solano from the Centro Nacional de Biotecnología del Consejo Superior de Investigaciones Científicas in Spain. The title of his talk is Evolution of Jasmanates in Land Plants and the Role in Thermotolerance. Roberto, if you're ready, could you please share your screen with us?
Yes, okay.
Thank you.
Thank you, Mario. Can you see it?
Yes.
Okay. All right. So thank you very much for the invitation, Mario. It's a pleasure to be here, and I really enjoy the talks of the sessions today. So today I'm going to be talking about evolution of jasmonides and basically how we discover that the hormone that activates the jasmonide pathway is different in bryophytes and vascular plants, right? And I will be talking about the evolutionary implications that this discovery has to understand the evolution of jasmonides. So we have been, So we have been working on jasmonides for quite some time already, and because jasmonides are very important hormones for plant survival in nature, and because they regulate many aspects of growth and development, and that I have divided here in three main topics. So basically, jasmonides regulate fertility. Without jasmonides, plants won't set seeds, and so we basically won't see any plant on Earth. In addition to fertility, jasmonides are very well known for regulation of defenses, so against insects and also necrotrophic pathogens. And jasmonides also regulate growth. They inhibit growth at the same time that they activate defenses. So they are very important in this trade-off between growth and defense that has very deep implications in agronomy and in crop yield. One of the reasons why jasmonides regulate defenses is because they regulate the secondary metabolism, the accumulation of secondary metabolites, which are different in different species. And here are 4 examples that are very interesting molecules or are current anti-cancer drugs used in pharmacology, right? So these are vinblastine, vincristine from Vinca, or paclitaxel and Taxol from Taxus. So basically what we want to do in the lab is to try to understand jasmonide biology and try to derive from this knowledge biotech applications that may be interesting in agronomy or even in pharmacology, as I will show you at the end of the talk. Okay, I need to introduce very briefly the biosynthesis of jasmonides because it's going to be key for this talk today. And jasmonide come from different fatty acids from the membrane of chloroplasts, mainly linolenic acid, and through a series of reactions that I'm not going to detail today, give rise to this important molecule for the talk, OPDA. And OPDA travels to the peroxisome, and there, by the action of this key enzyme, OPR3, that reduces this double bond here, funnels the OPDA molecule to the beta oxidation pathway, and after 3 cycles give rise to jasmonic acid. And then jasmonic acid is conjugated to isoleucine in the cytoplasm by JAR1. This is another key enzyme, so keep it in mind, OPR3 and JAR1. And jasmine isoleucine is the real bioactive form of the hormone. Actually, not any epimer of jasmine isoleucine. There are 2 in the cell. The cis isomer plus 7-isojasmine isoleucine is the active molecule that inactivates after epimerization to this trans isomer minus jasmine isoleucine. And basically, this is the molecule that binds the receptor and activates the jasmine responses. In the nature, there is another molecule called coronatin, and coronatin is produced by certain strains of Pseudomonas syringae, which is a very neat example of how evolution has produced a molecule in Pseudomonas to cheat the plant in the route of benefit. And basically, I introduced coronatin because this is a molecule that mimics jasmine isoleucine, and we use indistinctly in our experiments to activate the jasmine pathway. So you will be hearing me to talk about coronatin or jasmine isoleucine indistinctly. Regarding the signaling pathway, this slide summarizes about or more than 20 years of research. Very long time ago, we discovered a family of transcription factors mix that activated jasmine responses through binding to this G-box element in the promoters of those genes. Now we know many other genes other transcription factors from different families that activate these responses, but these were the first ones. And in basal conditions, in all transcription factors are repressed by this repressor complex formed by the JAS repressors that through this adaptor protein ninja brings the co-repressor topless and histone deacetylases to the chromatin to lock the chromatin, right? In the presence of the hormone triggers the interaction between the two pieces of the co-receptor, which is the JAS protein itself, the repressor itself is also a co-receptor, and this COI1 protein, which is the F-box protein of this SCF type of E3 ubiquitin ligases. Right? So the interaction leads to the ubiquitination of JAS repressor and then the degradation by the proteasome. So this liberates the transition factors from the repressor complex that now can activate transcription through interaction to the GNR machinery through this subunit of the mediator complex. So in this couple of decades, we have identified not only these components, but also many different mechanisms. So I think I lost the all right, here. So many different, I was saying that we have identified not only the components, but also different mechanisms to regulate those components. And but I want to focus my talk today in two issues that were worrying me in the current model Arabidopsis thaliana. One of these issues is redundancy. All the components of the pathway, or even the new mechanisms that we are identifying that regulate these components of the pathway, are genes belonging to medium to large gene families that share partial redundant functions, right? And this difficult very much the genetic analysis. So basically slow down the rate, the pace of discovery, right? The speed of discovery. So since a few years ago, I was looking for a different system, much simpler, that could overcome this redundancy problem. And the other issue I want to discuss today is representativity, right? And with representativity, what I mean is that all these details of the pathway, we have discovered those, we and of course many other laboratories, by using one single model species, which is Arabidopsis thaliana. And of course, don't take me wrong, Arabidopsis has been a wonderful model system for plant molecular biology, but it's just one species out of the 400,000 species that are out there on Earth, right? And these species are very different. So for instance, if you compare Arabidopsis to a sequoia, it is very clear that these plants are very different, right? So I don't expect that the details that we discover in the pathway or in any other pathway are exactly the same in these different species, right? So if we don't want to end having a very narrow view of plant biology, we actually need to understand how these details are in different species. And we have actually a wonderful opportunity now because we have genome sequence already of many representative of these species or these lineages in the tree of life, right? So just to summarize this, I think understanding conservation of the pathways is very important to broaden our view of plant biology, not only to understand or broaden this view, but also because this could be used as an approach to find specificities or new mechanisms that are present in different species that may help us to overcome the redundancy, the problem I was introducing before, right? And even if we don't find these specificities, studying the pathways, our favorite pathways in different plant lineages, at least we will understand how current pathways appear in evolution and how they evolve, right? So with this simple idea in mind, I was starting to look for our favorite genes of the JAS machine pathway in different important steps in evolution, from Arabidopsis to lower algae. And with this simple analysis, what I found was something quite interesting to me, because I found that the whole set of components of the JAS machine pathway appear with land colonization, so they only appear in land plants, not in algae. And there was a species, Marcatia polymorpha, where all these components were represented by only one gene, one representative. Later in evolution, each of these functions became more complicated with gene duplication, etc., and became small to medium gene families, right? So this is why I became interested in this species, Markhantia polymorpha, because the simplicity, the low redundancy. Markhantia is a liverwort, and therefore one of the three classes of bryophytes, which are sister to all vascular plants and are separated by more than 450 million years of independent evolution. And this is very powerful in terms of evolutionary studies. At that time, I was very lucky to participate in this project of the characterization of the genome of Markhantia polymorpha, that was led by John Bowman, Takayuki Kochi, Ton Yamato. And actually, Mario, I think, was also participating in this project. And this was very interesting to our laboratory, because in addition to set up the model system in the lab, I learned that this low gene redundancy not only applied to the jasmine signaling genes, but all kind of regulatory and signaling genes in the genome of Markhantia. So Markhantia is a very neat model system for understanding regulatory processes, right? And in addition to that, and to this key evolutionary position, or the power of evolutionary analysis in Markhantia, all kind of tools for transformation, CRISPR, etc., Markhantia has homologous recombination, etc., were very well set up already, right? So this was a very nice system to start working in the lab. And so we established this Markhantia in the lab as a new model system. And so in addition to, so the first thing we did was to analyze the conservation of our favorite pathways. As I was saying at the beginning, all the components of the signaling pathway were conserved. Now, when we analyzed the conservation of the biosynthesis of the hormone, turns out that the chloroplastic part was conserved. So we expected that OPDA could be produced, but these two key enzymes that I was introducing at the beginning, OPR3 and JAR1, were not conserved in the genome, were not present in the genome. So it was very unlikely that this peroxisomal and cytoplasmic part of the synthesis occur, and therefore it was very unlikely that Markhantia could synthesize jasmine solution. And actually, we measure these compounds after bonding, which is one of the stress signals that activate the accumulation of these compounds in biopsies. And we found that OPDA accumulated very nicely and transiently, very quickly in Markhantia, but we could not detect jasmonic acid or jasmine solution, right? And not only that, Markhantia does not produce jasmine solution, but also does not respond to exogenous treatment of jasmine solution or jasmonic acid. So in fact, none of the three classes of bryophytes, liverworts, mosses, or wholeworts, respond to jasmine solution, whereas they all respond to OPDA inhibiting growth, right? So Markhantia, or bryophytes in general, do not produce jasmine solution and do not respond to exogenous treatments. Oops. And so this raises this very interesting situation in which we have a group of plant bryophytes that contain all components of the jasmine signaling pathway, but they don't contain the hormone to activate this pathway, right? And therefore, we decided to try to identify the molecule that activated this pathway, but first we needed to make sure that the signaling pathway, these components, were functionally conserved, right? And for this, we set a series of combined approaches. We first identified or localized all components of the pathway in the right compartments. We tested all possible interactions between the components of the pathway to make sure, or based in the information that we had in our biopsies. And we also obtained mutants of all components to make sure that the phenotype that we show were consistent with the information that we had of those homologous genes in our biopsies. And basically, this is the example of just mutants. These are two alleles, and they are string dwarfs, which is consistent with a constitutive inhibition of growth by a constitutive activation of the pathway when we eliminate the repressor. The same thing with the transcription factor. If we eliminate the transcription factor, then we don't get response to the hormone, and these mutants in the transcription factor are insensitive to the hormone. And the same thing with the receptor. We obtain different mutants in the female or male background of the receptor COI1, and these mutants were completely insensitive to the hormone compared to the wild type male or female plants, right? And once we have these mutants, we could ask whether the processes regulated by this pathway are the same as in Arabidopsis. And here you can see that growth, inhibition of growth, is regulated by the COI1 pathway. Defenses are regulated by the COI1 pathway. You can see here that the COI1 mutant is extremely susceptible to insect feeding compared to wild-type plants, and they are also much more susceptible to fungal infection than wild-type plants. You can see here these more affected COI1 mutants than the wild-type plants by fungi. But what is different between Mercantia and Arabidopsis is the regulation of fertility, because fertility is not affected in this mutant. So we can cross COI1 male with female plants and obtain perfectly fertile sporophytes that set spores in same numbers as in the crosses with wild-type plants. So what this means is that this pathway was co-opted later on in evolution to regulate pollen development, which is what is basically regulated in Arabidopsis. But the gamete development itself is not affected by the pathway. And therefore, of course, Mercantia doesn't have pollen, so fertility, pollen development does not affect the fertility in Mercantia. And in terms of gene expression, the type of genes or gene ontology terms that are regulated by this pathway in Mercantia are pretty much the same as in Arabidopsis. So the same gene ontology term as responses to stress, wounding, NGA, secondary metabolism, etc. These are the same terms that we obtain in Arabidopsis. But probably the best proof of functional conservation of the pathway is that we can complement the COI1 mutant, which is, as I was saying, insensitive to the treatment with OPDA compared to the wild type. We can partially complement this defect by expressing the receptor from Arabidopsis. Now, the striking or the interesting result here is that now these plants are able to respond to coronatin or jasmine-isoleucine, which are, as I was saying before, the wild-type plants are completely insensitive, or COI1 mutants, right? So what this means is that the whole pathway is conserved. The responses activated by this pathway are the same in Mercantia and Arabidopsis, but the receptor is slightly different and determines the specificity or the molecule that is going to activate the pathway, right? In the case of Mercantia receptor, OPDA, or as I will show you later on, a derivative of OPDA is the ligand activating the pathway, whereas in Arabidopsis is just jasmine-isoleucine or coronatin, right? So to try to identify the molecular determinants in the COI1 protein that determine this ligand specificity, we complemented the COI1 mutant with chimeras that express different parts of COI1 from Arabidopsis or Mercantia. And what we found was that the C-terminus of COI1 was determining the ligand specificity, because if we had the C-terminus from Arabidopsis, for instance, in this case, then we get the same response at the full-length Arabidopsis COI1, so response to both OPDA and jasmine-isoleucine. But if we have the C-terminus from Mercantia, then we get the same response as the wild-type full-length Mercantia COI1, right? So we focus on trying to analyze this the C-terminal part, and of course, using the all sequences that we could find in this 1KP database. So there are hundreds of sequences from bryophytes, COI1 from bryophytes, and also about 900 sequences from vascular plants. And of course, we found many differences in this C-terminus part, but we focus on a particular amino acid that was this one here, an alanine in all COI1s from all vascular plants, right? So always an alanine in this particular position, but never an alanine in this position in any COI of any bryophyte, right? Most usually valine or isoleucine, valine in the case of MP-COI1, Mercantia COI1. And this was a very interesting residue because actually this residue contacts the hormone, is in the binding pocket and contacts the hormone. So we thought that this could be relevant for determination of the specificity of the ligand. So what we did was to complement the COI1 mutant with a mutant version of Mercantia COI1 in which we substitute the valine in this position to an alanine, right? And this protein was functional because could complement the insensitivity of COI1 to OPDA, but now the transgenic plants also responded to jasmine isoleucine and coronatin, right? So what this means is that one single amino acid was sufficient to switch a specificity or to broaden the specificity from the Mercantia COI1 receptor to the Arabidopsis COI1 receptor. And probably the reason is that being smaller, alanine here in this position makes a bigger binding pocket that allows coronatin or jasmine isoleucine to fit in here. Whereas in the case of valine, which is slightly bigger, doesn't or makes a smaller binding pocket and doesn't allow the fitting of this molecule. What is interesting here is that this bigger or smaller binding pocket suggested that the molecule binding or the hormone, the ligand of the Mercantia COI1 should be smaller in size than the hormone in Arabidopsis. We knew that OPDA was not the ligand. This is the typical pull-down experiment that we use to show that a ligand is really a ligand of the receptor. So we try to pull down, we use extracts of Arabidopsis plants that express COI1 flag and try to pull down this COI1 flag using a just gene fused to MVP in a column, right? So in the absence of any molecule, there is no interaction, so we can't pull down COI1. But if we use jasmine solution or coronatin, then they trigger the interaction and we can pull down COI1. OPDA was not a ligand of COI1, just in Arabidopsis, and none of these molecules were ligand in Mercantia. So we thought that, of course, the ligand in Mercantia should be a derivative of OPDA, and to try to find out what this ligand was, we actually searched in the literature for all possible molecules that we thought that had been described in any system, in any plant, and that we thought could be derivatives of OPDA. We ordered those in different catabolic pathways, and thanks to a collaboration with Max Humber in the Karolinska Institute, he synthesized most of these molecules for us, so we were able to test the activity of these molecules and also to use them as standards in mass spec experiments to quantify the accumulation after bonding of this molecule. So what we were looking for was a molecule specific of Mercantia and not present in Arabidopsis that was induced after bonding, accumulated after bonding. And of course, we didn't find any. Actually, what we found was a very well-known molecule for us, dinoro-PDA, that because dinoro-PDA is, we know it because it's a precursor of jasmine isoleucine in vascular plants. This molecule was present both in Mercantia and Arabidopsis, but in Mercantia had this isomer, dinor-iso-PDA, that was specific on Mercantia, not present in Arabidopsis. And this isomer accumulated very nicely after bonding, after 5 or 30 minutes of bonding, very transiently, and more than dinor-PDA or OPDA itself. And when we treat plants with these molecules, actually with all the molecules that Max Humber synthesized for us, we found that only dinor-iso-PDA or dinor-OPDA inhibited growth similar to OPDA in a COI1 dependent manner, right? So these molecules were not inhibiting growth in COI1. So this suggested that these molecules could be actually the ligands of COI1 that we were looking for. And using different kind of protein-protein interaction assays, we were able to demonstrate that actually dimer-iso-PDA is the strongest ligand of COI1, although dimer-thiso-PDA can also be a ligand of COI1, although we think that the real final molecule accumulating and activating the pathway, the real ligand of COI1 is dimer-iso-PDA, because as you will see later on, is the end product of the biosynthetic pathway, and is also accumulating more than any other molecule, any other isomer in the cell, right? So with this, we found the molecule, the ligand of COI1 in Mercantia to be dimer-iso-PDA, and here is the reason why the Arabidopsis COI1 can complement the Mercantia COI1 mutant. And this is because dimer-PDA can trigger the formation of complexes between Arabidopsis COI1 and Mercantia JAS, right? However, what is interesting is that when we use a JAS from Arabidopsis, in this case JAS9, but we have tested all JASes, dimer-PDA is unable to promote this interaction. So this experiment was suggesting that the specificity for the ligand is not only dependent on COI1, on this residue that I introduced before, but also depends on the nature of the JAS protein that, or the co-receptor JAS, right? And this is not only based on this in vitro assay, but also in vivo, we can completely switch the specificity of the response of the pathway or the hormone by switching both the COI and JAS co-receptors in a double mutant COI1 JAS, right? So as I was saying before, in the case of the COI1 mutant Mercantia, we can complement the specificity, the defect, the insensitivity to OPDA by expressing the Arabidopsis COI1, but still there is some response to OPDA, right? It's not complete, but there is some response. If we change also the JAS gene, the JAS repressor by in this case JAS3, then we don't get we get plants that do not respond at all to OPDA and now only respond to JAS myosin or conactin, right? So we completely switch the specificity from the natural hormone to the hormone from vascular plants by switching both, only by switching both pieces of the co-receptor. Okay. We also wanted to understand how these molecules are synthesized in the cell, because in the case of Arabidopsis, OPDA, linolenic acid that I was introducing at the beginning, give rise to OPDA, and then OPDA followed this pathway to produce jasmonic acid and jasmine in the presence or requires OPR3, right? But in Mercantia, there is no OPR3, so therefore we thought, well, maybe there is a direct conversion of OPDA into dimer-OPDA or dimerized-OPDA. And to try to test this hypothesis, we use deuterated derivatives of linolenic acid and OPDA and measure the deuterated products that after exogenous treatment. And just to give you simply an example, if we treated the plants with deuterated OPDA, then we saw accumulation of both dimer-OPDA and dimerized-OPDA after very quickly, right? But not OPC6, which is a marker of this other pathway. And if we treat with deuterated dimer-OPDA, then we only saw dimerized-OPDA, deuterated dimerized-OPDA. So basically what this suggested is that this is the pathway for the biosynthesis of the hormone, and this dimerized-OPDA is the final product of the biosynthetic pathway. And now this has important implications in terms of Arabidopsis also biology. This is not a specific of Mercantia, and this is, I think, a good example of how understanding processes in a different plant can also help when you go back to Arabidopsis to identify new processes that were previously hidden or at least unknown for us, right? And the example is here. So at the same time that we discovered this biosynthetic pathway in Mercantia, Andrea Kini in my laboratory was trying to understand why OPR3, which is this mutant that blocks the OPDA conversion and the synthesis of jasmonic acid, actually accumulated some jasmonic acid and was able to activate defenses to different kind of pathogens. So, and what he discovered was that also in Arabidopsis, OPDA can convert into dinor OPDA, but in this species, instead of going further to dinorized OPDA, dinor OPDA is transformed to tetranor OPDA and 4,5-dihydro-GA, and then this molecule is reduced to jasmonic acid in the cytoplasm by different OPR proteins, right? So with this, Andrea actually discovered a new pathway for the biosynthesis of GA in Arabidopsis that was previously unnoticed. And this pathway actually turns out to be the ancestral, we think is the ancestral pathway, the original pathway for biosynthesis of GA, because when he analyzed the presence of these intermediates, tetranor and 4,5-dihydro-GA, in different algae and bryophytes, he found that this was the only, the intermediates were present in this species that he analyzed, and this was the only pathway to produce jasmonic acid in this species. So what we think is that the original pathway, the ancestral pathway for jasmonic acid, was actually a catabolic pathway from OPDA by beta oxidation to get energy. So at that time, jasmonic acid was not a hormone, was just a product of the catabolism of the degradation of OPDA, and then later on in evolution, OPR3 was invented, and therefore appeared this other, what we thought was the canonical pathway, actually is a newly invented pathway in evolution to produce jasmonic acid in a more efficient way. And okay, so just to summarize what I just said, what we discovered is that dinor OPDA is a key molecule that gives rise to the hormone dinorized OPDA in bryophytes to activate this pathway. And later on in evolution, with the invention of OPR3 and JER1 in vascular plants, this hormone switched to germline solution, which required this complication of the biosynthetic pathway to produce jasmonic acid and germline solution more efficiently, right? And this also involved a switch of one amino acid in COI1 and also a modification in JAS proteins that restricted the capacity of this JAS to recognize dinorized OPDA. Right? Now, the other question that was also interesting from these conclusions was that dinor OPDA is actually present in all plants, right? So we know that dinor OPDA is not a hormone in vascular plants, but we knew that dinor OPDA was also present in charophytes, right? Because actually we measure, for instance, in the case of Cresomedium, is a charophyte, we measured these molecules and found that Cresomedium can synthesize dinor OPDA. So the question or the situation was actually the opposite, as I was introducing at the beginning. Now we have a series of some plants that have a hormone or a precursor of a hormone, but do not have the signaling pathway that this hormone activates, right? So the question was, what is the function of dinor OPDA in these plants? And to try to identify this function, what we did was to study or to analyze the transcriptional response of Clepsydridium and also Mercantia and Arabidopsis COI1 mutants, because we wanted to avoid the COI1 dependent responses. So the response of these three plants to dinor OPDA, so exogenous treatment to dinor OPDA. And what we found common in all of them was geoterms that have to do with different kind of stresses, but mainly temperature or heat stress, right? So we thought that maybe this molecule had an ancestral role in activation of stress responses or even in thermotolerance. And to try to test this, we set these conditions in which we can kill Mercantia, wild-type plants, by growing the plants about 2 hours at 37 degrees. The plants die, and this is also happening in COI1, so this is a COI1 independent response. But these plants survive if we pretreat the plates with OPDA or dinar-OPDA, right? And this not only happened in Mercantia, but also in Clasarmedium. So Clasarmedium dies at 45 degrees, but not if we pretreat the plants with, or the plates with OPDA or dinar-OPDA, right? So just to summarize this, what we think is that dinar-OPDA was a molecule present in the ancestor of all streptophytes that invented this molecule for activation or to cope with different stress responses, including thermotolerance. Later on in evolution, when algae conquered the land and gave rise to the current land plants, they invented the signaling pathway, and dinar-OPDA acquired a new function as a hormone activating a particular receptor. And later on in evolution, this hormone switched to a different one by complication of the biosynthetic pathway, right? And this is all I wanted to tell about the evolution of jasmonites. And since I have a few minutes, I would like to explain or to share with you a project that we have been working on in the last year. So I don't need to explain you how bad has been the pandemic of COVID-19, right? So with over almost 5 million deaths already worldwide. Now in Europe, we have a lot of, again, new infections. So the situation, even with vaccinated, highly vaccinated population, is still very bad. There haven't been, until very recently, any drug for treatment of the COVID-19. Remdesivir has been the only one that was not very active, actually. And a couple of weeks ago, I heard about the approval of a new drug from Merck and another one from Pfizer that is coming. But anyway, still, at least 1 year ago, there was a need for drugs to treat the COVID-19. And 1 year ago, I was thinking, as a plant scientist, how could I contribute to fight the disease, right? Could we do something in our lab to try to help humanity, right? And well, as I was introducing at the beginning, although liverworts are very simple in terms of regulatory processes, they are very complex in terms of secondary metabolism. And liverworts have an enormously rich diversity of secondary metabolites. So for instance, they produce more, or have been described, more than 1,600 terpenoids in liverworts. And well, Mercantia is one of them that produces many different secondary metabolites. So we thought, well, maybe we should search for antiviral compounds in Mercantia. And I was very lucky at that time because I could collaborate with a group of virologists in our institute, in the CMB in Madrid, that had set up these conditions. We had a high security lab in the center, so they could make infections with SARS-CoV-2. And these are monkey cells grown in plates in which, well, they had this color when they are monkey infected. But when they infected with SARS-CoV-2, the cells die and have this lighter color, right? So when we pretreat the cells, or we treated the cells with different extract from our favorite plant, or with mutants, wild type or different mutants, actually very low concentrations of the extract were protecting the cells and avoiding the accumulation of the virus. And actually, we could measure the RNA from the virus, and the reduction was huge and comparable to that of the Remdesivir, which is this approved drug for the COVID-19. So we decided to go ahead and and fractionate the extracts to try to find out what this antiviral was. And you can see different fractions without activity. Some of the fractions concentrate the activity. Actually, the activity was very good, reducing the amount of infection. And something that was important was that cell number or cell viability were not affected. So these antiviral or these fractions were very safe, were non-toxic for the cells. And just to cut a very long story short, we keep fractionating, and actually we ended with a fraction in which there was a huge accumulation of a compound that was less accumulated in other less active fractions. And we could determine the exact mass of this compound. And with the exact mass, we derive this formula and the idea that, or the hypothesis that this compound could be FeO4 by day. And as you can see here, FeO4 by day is basically a catabolite of chlorophyll, so not a secondary metabolite as we expected, but a primary metabolite. This is chlorophyll A without the magnesium that becomes magnesium of chlorophyll A, so present in all plants. And so FeO4 by day was commercially available. So we bought FeO4 by day to confirm that this was the molecule with antiviral activity. And using different kind of either monkey cells or human lung cells, we were able to demonstrate that FeO4 by day, in fact, is a potent antiviral that is very safe and does not affect cell viability, with an activity that is very close or very similar to remdesivir. And not only that, but FeO4 by day has a broad spectrum antiviral activity against other viruses like hepatitis C virus or other this cough coronavirus or West Nile, etc. So different kind of virus. So in conclusion, we are very excited that to have identified this FeO4 by day, the chlorophyll derivative, as a potent antiviral against SARS-CoV-2, at least in vitro in culture cells. FeO4 by day has a broad spectrum activity against plasma-stranded RNA virus, and we think it's a good candidate for antiviral therapies against COVID-19 because actually FeO4 by day has already passed clinical trials in humans because it has been previously used in anti-cancer therapies because it's a photoactivable molecule and therefore has been used in photoactivable therapy. So it has been already proved that it's not toxic in humans, and therefore I think the application in the clinic could be kind of straightforward. And with this, I just want to, sorry, I don't know why, yeah. I just want to thank you very much for your attention and acknowledge the people that has done this work. So in the case of the evolution of jasmonate, Isabel Monte and Andrea Kini and Sophie Nisho had a prominent role. Guillermo has done the work with COVID, the FeO4 by day. And of course, I want to thank our collaborators in the antiviral platform of the CMB and all our collaborators in the jasmonate field. Of course, I want to thank the funding agencies and you for your attention. I will be very happy to take any question that you want to place. Thank you very much.
Thank you, Roberto. That was amazing. So we have time for questions. Okay, so I'm going to read one from the chat. That was the first one that came in. It's from Nayeli Marsh. Great talk, wonderful work. Thanks. I would like to ask, what do you think that was the adaptive advantage of changing to jasmonic acid in plants like Arabidopsis? And also, how do you think that DNOPDA protects organisms from heat stress?
Yeah, okay, so these are good questions. And the first one is what I would like to know, and I can only guess, right? So So there is one very key important issue, which is that jasmonic acid and jasmonate solution are more polar than dimer or PDA, right? So what we think is that, of course, the hormone was originally dimer PDA. In plants that are not vascular, then that's okay. But in plants that invented vasculature, having a more polar molecule that could move through vasculature could have been a very important advantage, right? So we think that this may be the key. Of course, we don't have any data to support this hypothesis, and I would like to design an experiment, but it's not easy to do. And regarding the second question, how dimer PDA protects organism, I haven't explained that, but this is because dimer PDA has particularly dimer PDA and also OPDA in the structure, chemical structure, has a double bond and an alpha beta insaturation, which make this electrophilic character of the molecule. So these molecules are electrophiles. And I think probably what they are doing is to modify molecules by directly binding to these molecules. So we are actually trying to find targets of this molecule modified by different chemical methods. So probably they could modify transition factors, but also transport proteins or anything, right? So they are actually, this is also very important because these molecules are very reactive, right? And this is why the plant don't want to use dimer OPDA or to have OPDA free. And this is probably why the plant invented dimerized OPDA, which is much less reactive and much less electrophilic. And so this is probably why dimerized OPDA is the hormone in bryophytes and jasmonate solution in vascular plants. And dimer OPDA and OPDA are molecules that need to be controlled.
Thank you.
We have a question from Patricia Leon.
Hi. First of all, congratulations. It was a great talk. And I have two general questions. One of them is, in many cases, the proteins in Mercantia or the open reading frames are bigger than those that we can observe in Arabidopsis. And I was wondering if this is the case for the jasmonic acid biosynthetic pathway that you analyze.
So you mean bigger and bigger?
Bigger in size.
Yeah. Actually, the proteins are not bigger. The proteins in the genes are much bigger. You're right. But the proteins are not bigger. So what is bigger are the introns and the 5' and 3' non-coding regions. But so the genes are really big, but the proteins are pretty much the same, are not much bigger.
Okay. And my second question is regarding to the secondary metabolites. Do you have an idea if most of these metabolites come from the plastidial terpenoids, or they're coming from the cytoplasmic terpenoids?
I don't know. When actually, Mercantia, there are in liverworts in general, there are specific organelles that accumulate all terpenoids, and these are the oil bodies. So this doesn't exist in Arabidopsis and are specific for liverworts. And this is the biosynthesis and the accumulation occur there in oil bodies.
Okay, but the oil bodies are inside cytoplasm or are inside the plastids?
Sorry, say it again.
They are in the cytoplasm, the oil bodies, or they are inside the plastids?
No, are in the cytoplasm.
Okay. So they probably coming for this mevalonic pathway isoprenoid and not from the MEP pathway from the
Yeah, well, actually, what I don't know is whether all terpenoids are accumulating in oil bodies, or there may be part of the terpenoids accumulating elsewhere, right? So, but yeah, most or many are accumulated there. They are especially organelles.
Okay, thank you.
Thanks to you.
Okay, so we're now running into lunchtime. I just have one quick question regarding the activity of OPDA, or the derivative. Do you know if any of these molecules can induce epigenetic changes?
Not that I'm aware. You mean by directly by modifying something, or?
Yeah, or by indirectly by changing, for example, if they are so reactive, probably by altering some protein that might be involved in DNA methylation or histone modification.
This is perfectly possible. Yeah, this is perfectly possible. We don't have data yet because we are, as I was saying, we are trying to identify targets by purification, different kind of purification methods, but we still don't have good results. And so we don't know the nature of the proteins that they are going to be modifying, but my guess is that there will be many targets. And of course, proteins or chromatin related proteins, etc., are perfectly good targets, yes.
Perfect. Thank you so much, Roberto. I think that we will close our session. And thank you again for accepting our invitation, and thank you again for your great talk.
Okay, thanks to you, Mario. Thank you very much. Bye-bye now.
Okay, so we have now lunch. So I'll see you back at 2:45 for the first flash talk session of the day. Thank you, everybody, and see you later. talk session of the day. And if everything is ready, let's go. Thank you.
I'm going to present genome evolution and phylogenetic relationships in Opuntia tehuacana. As background, Opuntia has multiple taxonomic problems, mainly due to hybridization and polyploidy. In Mexico, there are approximately 93 species, from which 15 are in the Tehuacan-Cuicatlan Valley. Only 4 of these species have been included in phylogenetic analysis. Furthermore, their chromosome numbers remain unknown. Opuntia tehuacana is a wild species endemic to the Tehuacan-Cuicatlan Valley. It has variable characteristics throughout its distribution, such as the flower color, the shape of the stem, and the length and number of the spines. The aim of this work was to infer the phylogenetic position of Opuntia tehuacana in the context of its sympatric species and analyze their genome evolution by estimating ploidy levels and genome size in 5 populations. What we found in a broad phylogenetic context using 3 plastid markers, the species from the Tehuacan-Cuicatlan Valley are in the clades Nopalea and Basilares. Opuntia tehuacana is nested within Basilares, and all of its terminals form a monophyletic group. About the genome size and the ploidy level in Opuntia tehuacana. Opuntia tehuacana is on decaploid and dodecaploid, the highest reported so far for the genus, and our results show significant differences between the 2C nuclear DNA content, and this is probably due to allopolyploidy, satellite DNA transposable elements, or ribosomal genes in different genomes. Finally, about the chromosome pairing and gene flow. In Opuntia tehuacana occurs aneuploidy, which may implicate gene flow from other species, and the pollen mother cells present rings and univalent, which have been observed in neo-allopolyploids. Finally, we can conclude that multiple polyploidization events in Opuntia tehuacana are related to their ongoing genome size evolution. And this differentiation between the localities could also be related to their highly variable characteristics, but further studies should be performed to confirm this hypothesis. Finally, I would like to thank to all the institutions and people who helped to this work. Thank you.
Hi, my name is Luis Gerardo Ortega. The topic is characterization and pharmacological evaluation of Callistemon citrinus leaf herbosomes. Callistemon citrinus leaf extract has shown its potential effect as a possible anti-obesogenic agent. The biological activity of Callistemon citrinus has been mainly attributed to phenolic compounds and terpenoids previously reported in vivo studies. The phytoconstituents, due to molecular weight and intrinsic instability in some physiological fluids, phenolic compounds passage through biological membranes is very limited. Carriers like phytosomes are promising systems to optimize oral absorption of encapsulated extracts. The phytosomes is a technology developed to complex standardized plant extracts or water-soluble phytoconstituents with phospholipids to produce lipid-compatible vesicular structures named herbosomes or phytosomes. The aim of this work was the standardization and pharmacological evaluation of Callistemon citrinus leaf herbosomes was evaluated. The Callistemon citrinus leaf herbosomes at dose 200 mg per kilogram show vesicular structures under the light microscope, and the weight analyzed particle showed a size from 150 nanometers with great similarity in terms of the size of the vesicular particles. Regarding solubility, it was found that the solubility of Callistemon citrinus leaf herbosomes was much higher than that of Callistemon citrinus leaf. Finally, it was found that the encapsulation efficiency of the herbosomes was the 80.49%, represented by the concentration of the unbound Callistemus strinus leaf extract. With these results, the optimization of the herbosome preparation technique was obtained in terms of conditions such as reaction time and temperature. The results highlight the potential of Callistemus strinus compounds as supplements or pharmacological tools, suggesting a promising and safe phytosomal formulation containing bioactive agents of Callistemus strinus that could lead to health benefits during obesity. Thanks for your attention.
Hi, my name is Alfonso Sierra and my PhD project is titled Americano truncatula MIR2199 Coordinates Environmental Responses. Well, we know that plant microRNAs participate in multiple processes including development, stress responses, and their own biogenesis. Their activity is exerted through cleavage of target mRNA or by translational inhibition. Several plant microRNAs regulate transcripts encoding for transcription factors, thus amplifying the microRNA effects through regulation of downstream genes. We previously identified MIR2199 in Medicao truncatula and other Fabaceae species. In Medicao truncatula, 4 VHLH mRNAs encoding for basic helix transcription factors are targeted by Medicago MIR2199. One of them, SAR1, is a key transcriptional activator of saponin biosynthesis, a pathway previously shown to enhance symbiotic nodulation. In this work, we determined that Medicago MIR2199 increases its abundance in roots and leaves, while SAR1 mRNA abundance is reduced upon water deficit. We are employing genome editing, heteroroot transient transformation, and mutant lines to determine the molecular and phenotypical consequences of affecting MIR2199 and SAR1 levels, and in turn identify downstream genes networks regulated by this module involved in water deficit response and during root development. Our aim is to understand how SAR1 silencing mediated by MIR2199 is involved in the plant response to water deficit and root development. SAR1 role in saponin biosynthesis is known to promote plant bacterial symbiosis, which represents an expensive process for the plant in terms of carbon usage, while at the same time this element becomes one of the first affected nutritional fluxes under a water deficit condition. How these processes are coordinated could be addressed by studying the gene expression changes caused by MIR2199 and SAR1 regulation. Well, I want to say thank you for listening to my talk, and I will be glad to answer all the questions. Thank you and bye.
Currently, bioinformatic tools allow us to simulate molecular processes in a reliable way. However, before being used as a suitable method for the specific research, it is necessary to validate its feasibility by experimental data. This work aims to evaluate the use of simulations to analyze the effect of the mutations and the delta-delta G to be used as a selection method for target potential sites to increase the catalytic activity of the enzyme lipoprotein synthase or LOS in the hydrocarbon production process. LOS is the key enzyme for the lycopene biosynthesis in the microalgae Botryococcus brownie I arrays and has been demonstrated to be substrate promiscuous. We have adapted an in vivo system that allows us to evaluate the role of specific residues and the substrate binding process of LOS based on the hydrocarbon production rate using E. coli as a host. In this slide, I show the estimated delta-delta G of each amino acid of loss mutated to 20 amino acids. To validate this method, we analyze the hydrocarbons and discover that low delta-delta G increase the total hydrocarbon production, and high delta delta G decrease the hydrocarbon production. The lowest delta delta G was estimated on the serine 276. Interestingly, we noticed that high delta delta G increased the amount of lycopodioctane in the samples. With these results, we were able to demonstrate the reliability of the selection of in silico target sites and in a fast, easy, and economical way. We are at this moment working on the evaluation of in vivo hydrocarbons from other sites selected by delta delta G analysis, and we are confident that these sites will be determinant in the pursuit of the highly efficient LOS versions. Thank you so much for your attention, and thank you to everybody on the DevRNS lab.
Hello, my name is Julian Peña, and I'm going to present this research work by Lucy Medina, Pillada Junta Wong, and Blanca Barrera. We titled this Submergence Stress Affects the Expression of the Evening Complex Genes and Produces Altered Transcriptomic Outputs of the Diurnal Cycle. We are very interested on the impact of flooding on crops, which is brought to the field by hurricanes, and Mexico and the USA are in the middle of one of the most active sites of hurricane activity. Submergence and drought stress compete every year to be #1 stress causing economic damage on crops. So to learn more about this phenomena, we use Brachypodium distachyon. We submerged this model grass at CT13, leave it underwater, and started collecting plants the next day at early dawn, middle of the day, evening, middle of the night, and the next dawn. We did RNA-seq and gene ontology analysis to catch some genes like flowering locust T, which has a normal peak in the night, but under submergence is strongly downregulated. For that, we use sensitive and tolerant ecotypes such as BD21, as you can see, is the sensitive ecotype, and BD21.3, which is the tolerant ecotype. When we quantify this, we can see that BD21.3 stays green for longer periods of time when compared with BD21. So the first thing we observed when we did gene ontology analysis was that the evening complex genes, in special PRR5, which is the master inhibitor of the clock genes, is very upregulated. So if the master inhibitor is upregulated, then the rest of the clock genes could be expected to be downregulated. And such was the case, as you can see here. Let's do another zoom on circadian clock associated and giantia, and these genes are also downregulated here in the gray line. What was the result of these alterations? For example, some of them were exclusive of the sensitive ecotypes, such as cellulose synthesis. So this plant continues growing while the other one keeps the energy. The tolerant ecotype had, for example, pyruvate kinase upregulated, which is very important to keep the plant extracting energy for carbohydrates. We found many others in other time of the day, for example, polyamine biosynthesis and phenylalanine catabolism genes upregulated in the tolerant ecotype. These both are new actors. We could also find older actors, such as nitric oxide regulation by means of upregulation of hemoglobin in the tolerant ecotype. So our conclusion is that submergence stress affects the expression of the evening complex genes, especially PRR5, and produce an altered transcriptome of the diurnal cycle. Some outputs are exclusive of the sensitive, some others of the tolerant ecotypes, and we are learning what all these results mean for plant survival. Thank you all.
Flash talk, analysis of pitaya fruit exocard transcriptome toward identification of cuticle biosynthesis genes. The plant cuticle protects the fruit during development and ripening of harsh environmental conditions and extends the profile shelf life of fruit by reducing the water loss. Cactus are plants adapted to survive in environments with low water and low relative humidity. Cuticle plays an important role in the adaptation to these conditions. Nevertheless, there is no information about transcripts or proteins that plays a role in cuticle biosynthesis in cactus. Stenocereus turberi is a cactus endemic from Sonoran Desert, which produces a fleshy fruit named pitaya. In this work, we carry out the sequencing of the pitaya fruit exocard transcriptome to elucidate the molecular mechanisms of cuticle biosynthesis. This information could help in designing protocols to reduce water loss in other fruits. Trinity default parameters were used to assemble 243 millions of reads with a minimum Fred quality score of 25. The assemble includes 174,000 transcripts with an N50 value of 2,110. 85% of complete transcripts and only 11 and 4% of fragmented and missing transcripts, respectively. Homology searching by alignment to different protein database shows that the percentage of homologous ranges from 37 to 49 for the protein database includes in this analysis, and for proteins from model plants and commercial fruits. In the case of cactus, percentage range from 44 to 66%, with the exception of Illucerius polyrhizos. All the cactus database included in this analysis show the highest amount of homologous transcript of strawberry transcriptome. This could be due to a higher conservation of sequences among the Cactaceae family. 19% of transcripts neither show open reading frame nor homology, which represent a valuable resource for searching long non-coding RNAs. We have already identified orthologous genes playing a role in cuticle biosynthesis in other species and reference genes for the quantification of the expression by real-time PCR. This information will be helpful for further studies about the molecular pathway of cuticle biosynthesis and response to abiotic stress in cactus. Thank you.
Hello, my name is Oliver Rafil Magaña Rodriguez, and the name of this project is Antipancreatic Lipase Effect of Edible and Medicinal Plants. Strategies in the prevention of obesity can be controlling food intake or altering lipid metabolism by inhibiting fat absorption. Pancreatic lipase is the most important enzyme in the digestion of 50 to 70% of fat diet. It is responsible for the hydrolysis of triacylglycerols to monoacylglycerols and free fatty acids. Orlistat, a derivative of lipstatin obtained from Streptomyces toxitrini, is the only pancreatic lipase inhibitor currently approved for the treatment of long-term obesity. Its use can result in up to 10% weight loss when used in combination with diet and physical activity. However, this drug can cause adverse liver effects. One of the options to combat obesity is finding plants that have a strong activity to reduce the digestion of lipids from the diets. The main of this study is evaluating 3070 ethanolic extract of plants, some edible, medicinal, or belonging to a family that have the inhibitory activity of pancreatic lipase to found a new anti-obesity agent. We collect the plant and separate the plant tissue, grind and macerate it in ethanol, evaporate the solvent to obtain the pure extract. To proceed to spectrophotometric anti-lipase assay in each extract. The degree of the inhibition of pancreatic lipase in vitro was compared using orlistat as a control. Results revealed that 9 plants had a low percentage of inhibition of lipase activity, 41 or less percent. 9 extract with moderate percentage of inhibition, 41 to 50%. 8 extract in high inhibition range, 51 to 60%, and 11 extract with the highest percentage of lipase inhibition greater or equal to 61%. Ethanolic extract of hibiscus racesinensis of dry leaf display the highest inhibitory activity of pancreatic lipase, 72% at 400 milligrams per milliliter, very similar to orlistat. When performing the enzyme kinetic of hibiscus racesinensis, it was found that it has an uncompetitive inhibition. That is, the extract can inhibit both the enzyme and the substrate enzyme complex, unlike the orlistat, which presents a competitive inhibition. As a conclusion, the plant's analysis have pancreatic lipase inhibitory activity. Hibiscus racesinensis showed a very high anti-lipase capacity, very similar to orlistat, so it could be a natural alternative for the prevention and control of hyperlipidemia and obesity. Thank you.
Hello, my name is Maria Guadalupe Pérez Muñoz. I'm a student in Universidad Autónoma de Zacatecas, and I'm doing my thesis in Tecnológico de Monterrey. So we have this fruit that has for name in silico characterization of one carbon metabolism genes during the post-harvest ripening of climateric fruits, Persia americana and Solanum lycopersicum. So let's start with folates. Folates, known as folic acid, they are essential in algae cells, and they are related with several aspects in plants, just like development. And they're important because they donate one carbon for the biosynthesis of purine, stimulate, or amino acid, just like methionine. So they assist the ripening process in climateric fruits. So we have this folate that donate one carbon for the production of ethylene. And there's a previous work that say that ethylene affects the pools of folates in climateric fruits. So we have this table, so we can see at least one autologous gene for one of Arabidopsis thaliana. And these sequences in this alignment, we can see a signal peptide for subcellular localization. So we have this phylogenetic tree. So we can see that they come from the same family, and we predict the subcellular localization. So we can see with this little images. And right here, we have this alignment. So we compare these sequences, and we can see they are highly conserved because the black part means that they're equal. So we can see the identity, and for methionine, it's more than 72%. And for SAMs, it's more than 77%. So right here, we have the expression of the genes, and we get this information from the transcriptome from Ibarra-Laclet, and we can see how the genes are expressed in every stage in the ripening. So they're upregulated. And this is the same for tomatoes. So we can see the impact for methionine and SAMs in the production of ethylene. Just to finish, we have this conclusion. So we get at least one autologous gene from avocado and tomato genomes, fruit genomes. And these plants have one cytoplasmic and one organellar plastidic or mitochondrial predicted methionine form. And these proteins are highly conserved, just like we see in the alignment. And methionine and SAM expression was modulated temporarily in the ripening of avocado and tomato fruits during the preclimateric stage. Methionine synthase 1, the cytoplasmic isoform and SAMs were expressed according to meth and SAM requirements for ethylene synthesis. Finally, in the climateric and post-climateric stage, ACS and ACO were expressed. Thank you so much, and I hope that you like it.
Hello, my name is Luis Ángel Choca Orozco. I'm going to present the work titled RNA-seq Analysis of the Dilemicus Dipsashi Allium Sativum Interaction. As collaborators, we have Donaldson McLeod, Carlos González, Julio Amezcua, y Alejandra Rugón as corresponding authors. Dilemicus is a phytoparasite nematode considered within the top 10 more damaging nematodes for crops. Nematode is capable of surviving severe freezing in humidity conditions, as well as being able to survive for a long time in soil and the host absence. This nematode causes significant economic losses in garlic cultivars. Garlic contains various substances with antimicrobial activity and even with nematicidal activity against some nematodes. However, this defense mechanism does not work against the invasion of the Dipsashi. That is why our aim was to analyze the Dipsashi garlic interaction to elucidate which are the mechanisms both host defense and nematode attack. For this purpose, we isolated, identified, and cultured nematodes inside garlic bulbs. Mock and nematodes were included in healthy garlic slices and incubation times. The inoculation was carried out with 10,000 nematodes, mainly in juvenile stage. Tissues were measured in liquid nitrogen followed by result RNA extraction. cDNA pirate libraries were sequenced from the Illumina platform. Expression analysis involving comparison of transcript of Allium versus Allium plus nematode and the different time points. We found that in the host, some of the differentially expressed transcripts that are related to disease resistances are induced in time zero, which was taken 10 seconds after inoculation, whereas at 24 and 72 hours they are in the basal state. Likewise, some transcripts related to the alliance biosynthesis have the same behavior. For the nematode expression profiling, the result shows that many transcripts related to effectors are induced in the early stages as well, and as infection progresses, they reduce their expression, whereas others have the opposite behavior. Finally, many candidate genes related to defensive garlic, antiparasitins, and ditilencus were found in this work. We found that the many overregulated transcripts are shown in early stage of the integration in both organisms. Further analysis are required to confirm the role of candidate genes during this integration. Thank you.
My name is Norma Cecilia Morales. I am PhD student at the Colegio de Postgraduados in the States of Mexico. Playground in gel are dependent on the photosynthetic fixation of carbon and optimal allocation broadly in syn tissues. However, in common being in reproductive states, produce fruit with different signs. Several evidence indicate that part of the allowed carbon is stored in pericalized storage, suggesting that pericles play important role on reserve of carbon. We wonder in planes the change in variability will modify the distribution of photosimilate in fruits. In this study, we explored the carbon distribution of planes of common being genotype OT during pollen filling state under 3 levels of variability. Using 14 carbon pole chains, the carbon distribution were measured collecting plane at 1, 3, and 7 days. However, the fruits were divided in 3 sizes. Here we can see the total carbon distribution. The accumulation of the carbon-14 label in the pericarp chains during time almost absent in the pericles of the small and large fruits. In medium, the pericarp decreased while in the seed increased with the age of the fruit. These results show that pericles play the important and dynamic role in the responsibility to moisture retention. In the pericarp, the fructose concentration doubled that of the glucose in decrease with the pole age. The sucrose decreased only in the little pericarps on day 7, as well as the starch concentration that decreased by half in the 50% fill capacity condition. The level coincided with the highest amylolytic activity as evaluated in naval health. We concluded moisture restriction modified the distribution of photosimilate and obstruct their allocation to early fruit that understood just the reserve stored in the pericarp and soluble sugar and starch. In this article, you can review all the complete research. Finally, we would like to acknowledge all the institutions and the people who made this research possible. Thank you.
Okay, that ends our Flash Talk session. We're going to end with the last talk today from Dr. Prasinkevich from the University of Calgary in Canada. You probably all know his wonderful work on all sorts of wonderful mathematical modeling and phyllotaxis, but the talk today is Phyllotaxis Beyond the Standard Theory. So we're looking forward to your talk. You can take it from here, Dr. P.
Thank you very much. Can you hear me well?
Yes, we can hear you.
Okay, let me share the screen. And okay. And just a check. Can you see also my opening slide?
Yes, the slide looks great.
Okay, thank you very much. So thank you again for the introduction, and thank you very much for the invitation for me to present. I will talk today about phyllotaxis, and it is a work which has been done jointly with Dr. Paula Eloma from the University of Helsinki and her group there. So what is the problem? And before getting to the problem itself, let me to introduce a little bit the key ideas pertinent to phyllotaxis. So phyllotaxis is the arrangement of organs on their supporting axis. And often it has a particularly interesting geometric feature. For example, in this case, we are dealing with disticus phyllotaxis, where individual florets are arranged in a single plane to the left and to the right. And here there is another example of the cassette phyllotaxis, where leaves are issued in pairs, and each pair is rotated by 90 degrees or is perpendicular to the previous one. And this is yet another example, which is called spiral phyllotaxis. And if sort of the most prominent cases of spiral phyllotaxis are found in flower heads. So you can see some spirals here, and if you look more carefully, you can see that there are two families of spirals going clockwise and counterclockwise. And well, the spirals are called parastiches. And if we count them, in this case, it turns out that there is 21 parastiches going one way and 34 parastiches going the other way. Now, there's lots of history going to this work on phyllotaxis. In particular, Leonardo da Vinci is credited for having been the first who observed the presence of spirals there. And Johannes Kepler was the first one to pay attention to the numbers, and he discovered that very frequently the numbers of these spirals of parastiches are consecutive Fibonacci numbers. So what are Fibonacci numbers? They are described by this kind of formula, which may look a little bit cryptic, but all it means is that we start from two numbers, 1 and 2, and then keep adding the last two numbers to get the next one. So 1 plus 2 is 3. Now we take two last numbers, 2 and 3 is 5. Again, 3 plus 5 is 8. 5 plus 8 is 13. 8 plus 13 is 21, and so on and so on. And you can see that these numbers which we have seen in sunflower head, 21 and 34, are represented here. So this leads to the key question: how do plants create spiral phyllotaxic patterns? And the basic model is based on the idea that existing primordia in the meristem are inhibiting the emergence of new primordia nearby if they are too close. So let's look how it works. This is shoot apical meristem, and in the shoot apical meristem, we distinguish if a structure is called active ring. This is where new organs or young organs, primordia, may emerge. Let's suppose that we have well, the first primordium to start with. As the meristem grows, this primordium inhibits the presence of other primordia nearby until to the growth it is too far from those created before. This enables another primordium to be formed. And the process in this way continues. So now another primordium is going to be formed either here or there. This is called breaking of symmetry. And it is easiest to see how things work in a simulation. So first primordium has been created, and the strength of inhibition is the color. So if the whiter it gets, the less inhibition it is, and the new primordium can appear when a threshold has been reached. And you can see that new primordia are emerging in a spiral pattern. So this is kind of the basic mechanism, which has been figured out relatively a long time ago. This particular simulation was done by my then postdoc, Johannes Batjes, in 1996. And if this model over time has been perfected quite a lot. In particular, it has been the mechanism of inhibition has been explained in terms of plant hormone auxin. So it turns out that inhibition is not some factors which is positively inhibiting, but this lack of auxin which is absorbed by existing primordia. And then if it is not enough of it, the new primordium cannot be formed. Without going into these details, which have been described among others in a paper by Richard Smith and others in PNS 2006. You can see here the microphotograph of shoot apical meristem of Arabidopsis. And here is the result of simulation, which, well, at least visually shows that it is possible to get close to modeling phlotaxis via this inhibitory mechanism at the level of meristems. And we can go further by looking at the mature structures. And again, here is photograph, and on the right you can see the model of Phyllotaxis Arabidopsis, now in more mature plants. It turns out that this mechanism can be further simplified and expressed in geometric terms. And the simplification belongs to Hofmeister, Wilhelm Hofmeister, who observed or who postulated that new organs emerge at the circumference of the growing apex, so in this active ring, where there's enough space for them. And then later, Snow and Snow added that this new meristem, new primordia appear as soon as there is enough space for them. So basically, this is how this mechanism works. New primordia are created at the tip, and then as the apex grows, a phyllotaxic pattern is created. So it would seem that everything is now clear. However, well, as it is often the case, nature is more complex than this. This is the scanning electron microphotograph of a young Gerbera head. And you can see a phyllotaxic pattern, but the point is that this pattern is emerging from the outside in. And this is, well, a common phenomenon in all flower heads. And it is very fascinating how this pattern can be formed from the outside in in violation of the mechanism which we discussed so far, what is supposed to be happening near the tip of the meristem. And in particular, these inhibitory mechanisms, which I mentioned before, cannot work as well if in the large head because, well, there is this empty space in between, which sort of prevents communication between primordia across the circumference. So this is a key question, which, well, Paula Eloma and I and our groups set up to solve. However, this question has some history as well, and in particular, in the 1990s, the late Professor Paul Green from the Stanford University was very fascinated by the same problem, and he proposed a biomechanical theory according to which this emergence of primordia takes place on the, well, in the active ring due to buckling, that is to the fact that if sort of due to disproportions of growth rates, ondulations mechanically occur, and then they propagate inwards. And Paul Green created a model which explains this, and the model works, except that it explains the emergence of whorls and not phylotactic patterns, in especially with Fibonacci numbers. So this problem remained unsolved. And with the Helsinki group, we looked at this in more detail using Gerbera hybrida as a plant model to consider. And there were several reasons for choosing this plant, but one important reason is that, as you can see, flower heads are supported by very long naked stalks, which suggests their phylotactic pattern is emerging de novo and is not inherited from the leaves underneath. And this phylotactic pattern has nice spirals, and actually, well, it turns out that the numbers of primordia can come close to 1,000. So it is a very elaborate pattern. So in order to see what happens, we looked carefully into the development of heads. And here you can see the developmental sequence. So those are micro-CTs of heads in different developmental stages. And those are the kind of, well, virtual cross-sections. So you can see the progression of shapes. And sort of coloring shows the part which is covered by primordia. So this process can be divided into three phases. In the first phase, well, first is the meristem, or the young flower head, is naked. There are no primordia there. And then first primordia appear on the rim. In the next stage, new primordia are being inserted between previous ones as the head grows. And we can see that there are some, that the newest primordia are a little bit offset towards the center of the head compared to the older ones. So there is almost kind of like, well, the pattern is no longer one-dimensional as it was on the rim. It has two-dimensional aspects. And then if they gradually, in phase 3, the head is filled, this is when parasticus can be observed, and their number decreases according to the reverse Fibonacci sequence. And eventually, this active zone basically disappears, or convergence in the center, and then near the center, the pattern becomes chaotic. So the most intriguing part was this emergence of primordia on the active ring, on the rim of the young head. To understand it better, Teng Zhang from Paula's group has taken a number of confocal microphotographs in which presence of auxin was reported using the year 5 reporter. So you can see here these heads, and it turned out that several things were interesting. First, that if among these random numbers, majority of heads showed Fibonacci number of primordia. This indicated that primordia are inserted in bursts. And also, basically, both chiralities occur, so the spirals kind of could be oriented well to the left and to the right. However, if we reflected them, it turned out so that we abstracted from these differences in chirality. It turned out that the development is highly stereotypical, and also we could not follow the development of individual heads over an extended period of time. We could see we could reconstruct the progression from actually either individual heads cherry-picked or by superimposing heads in the same developmental stage. So what we see here is that this is a process in which patterning is basically taking place on the rim of the head. However, the numbers of primordia jump according to Fibonacci sequence. And the question is, how is this possible? So, well, the first thing is to start straight Hofmeister's hypothesis. Well, to assume that if new primordia are emerging on the rim whenever there is enough space for them. So this is a simulation of this process. Whenever there is enough space between existing primordia along the rim, a new primordium occurs. So, yes, we have increase of number of primordia on the rim, but if the numbers do not increase according to Fibonacci sequence, but just increase according to power series 1, 2, 4, 8, 16, and so on. As actually, it is very intuitive. So this was a question. If, well, something different must be happening. And in order to find what is happening, we looked at these heads in further detail. So this is how we analyzed it. This is a head with one primordium. There's, well, basically just a single, we can say that there's a single interval, single ring kind of around there from this head, from this primordium to itself. Now, when we have two primordia, interestingly, the pattern is asymmetric. We have a shorter gap between primordia here and longer gap there. We can symbolically write it as short, long, if we scan this head from this first primordium going in the counterclockwise direction. Now with three primordia, we have SLL, or LLS. And then we proceed while constructing this in the same way, getting a sequence of sequences of Ls and Ss. So the question is whether there's pattern to this. So here I wrote the first 8 sequences, and here I superimpose them on sort of the ring, which is kind of straightened. So it is kind of, well, this is the ring, this is the ring, but it's drawn as a straight line. And we see that what happens is that if we have a long gap between primordia, then in the next developmental stage, it's going to be divided into short and long. But if we have a short gap, then at the same time, it is just going to grow and become long. So we can describe this process using rules that long gap produces short and short just becomes long. And it turns out, as you can see here, that this process described by these rules always needs to Fibonacci sequence that can be mathematically proven that it is the case. And however, there is a caveat there. We can see that, for example, here long segment has been divided into short and long, first short, then long. But this long segment has been divided into long and short. So the question is that sometimes short is on the left side, sometimes on the right side of during the division. So the question is how this can be captured. And it turned out that it can be captured by assuming that the intervals have polarity, and kind of further analysis shows that shorter gap is always at the stage, at the side of the older neighbor, older primordium. So now the question is how this kind of asymmetry can occur. And to analyze this, we looked into time-lapse confocal microscopy of heads, and we could only do this for a short period of time, but you can see it here. And looking at this part there, which is magnified here, we found out that this maximum of auxin expression originally appears halfway between both primordia, but then propagates, tends towards the older one. So this is how asymmetry between intervals occurs. So here is going to be short interval, and here is going to be long interval. And we created a model of this process and superimposed on the images. So you can see that when primordium is introduced, it always tends towards its older neighbor. And in such a way, Fibonacci numbers of primordia occur. Okay, so this is phase one. On the rim, primordia appear in Fibonacci numbers. Now the question is, what happens next? So we looked into the distribution of the Gerbera Clavata3 gene, which we and others assume that define defines the position of the active ring. And we can see that the expression of this gene first grows with the head, but then gradually shrinks. So consequently, we introduce this mechanism to the model. First, things are happening on the rim as before, but then the active ring starts to move inwards. And you can see that now new primordia are being displaced a little bit inwards compared to previous ones. Now let's focus on this pattern. And if we just connect the centers of primordia, we can see a zigzag line. And it turns out that this zigzag line corresponds exactly to the distribution of this L and S segments if in the model in the rim. And since the number of L and S segments are two consecutive Fibonacci numbers, as this zigzag provides a template for the subsequent formation of parasticus in the third phase. So those are the symbols S and L, and this zigzag pattern, which has Fibonacci numbers of both this kind of intervals and this kind of intervals, serves as a template for the formation of parasticus. So let's look at this. This is again the template, and now the active ring grows further. And according to Hofmeister rule, new primordia are formed whenever there's enough room for them, and the pattern has emerged. And if we look at this, there are Fibonacci numbers of parasticus as it can be observed. Moreover, as we move toward the center, we can see that the numbers of parasticus decline. That is a well-known phenomenon, occurring in models and of course in reality as well. And in the center, the pattern becomes chaotic, again, as observed in real heads. Moreover, if it turns out that this active ring does not have to be circular, and the pattern can work fine as well. This is interesting because, as I'm going to elaborate towards the end of my talk in a few minutes, the standard theory of phyllotaxis is very much preoccupied with the radial symmetry of everything which happens there. However, it is not essential. We can have perfectly nice patterns, even though in this case, active ring at some stages is not circular. So we further tried to create, to show that we really understand what's going on. We tried to create these patterns on the model of actual growing head. And to this extent, we look again at this profile of the heads and superimpose them and extracted contours shown here as colorful lines of different colors. And then we interpolated between them. And in such a way, we got a model of growing head where the red color indicates the position of the active ring. So it is obtained by, it is a data-driven model obtained by interpolating between the measured shapes of the heads. In addition, we knew how the individual primordia develop, and we combined these elements together to obtain a model of a growing head, which is three-dimensional, not just flat, and with primordia taking the same forms as they would be seen in an SEM. So you can compare this as SEM of actual head as a way of validation. There is more validation in the paper, which I referred to earlier by Teng Zhang and all, which was published earlier this year. However, it is not the end of the story, because it turns out that some heads are not round, can be actually even quite elliptic. And so those are examples of wild type heads in Gerbera, which sometimes are like this. So it turns out that the model which I just described can capture this kind of patterns as well, because nowhere in the model there is assumption of circularity present. And here you can see, well, I just showed here the last stage, how this pattern can develop. The interesting thing is what happens when this pattern collapses, and well, we assume that in this case there is necessary to have some repositioning of primordia in the process of growth. Anyways, we can recreate that kind of pattern, and you can see here that these parastiches kind of have different form, kind of more elongated versus more curved. And again, we have Fibonacci numbers of parastiches. This kind of patterns actually appear in different plants. And well, actually last summer, I have seen lots of fleabane flowers, so this is erigeron, which have this kind of patterns. So here is an example. And it interestingly has a very kind of linear pattern here. And this is the model. And this is, well, a smiling face pattern. And here is the model. So to conclude, phyllotaxis in flower heads is no longer a mystery. If we found out that it is can be explained as a superposition of three phases. The first one is patterning on the growing rim, which is basically a one-dimensional process with a lateral displacement of primordia. This lateral displacement is critical for Fibonacci numbers emergence. Then a zigzag pattern occurs due to the gradual shrinking of the active ring. And then as the active ring propagates further, a centripetal pattern occurs and fills the head. Moreover, we observe the patterning does not need to be radially symmetric, and this offers a key to understanding the question of patterning in fasciated heads. And also, this means that well, some key notion of phyllotaxis have to be updated, because well, even the terminology is so very much based on the assumption of circularity, the presence of the center of the head, and so on, that it is difficult even to describe verbally, let alone mathematically, what is happening when this assumption of circularity is abandoned. There are also interesting questions. How does fasciation emerge? What is the dynamics of the active rim propagation in fasciating heads, and how it is controlled? Why it is fasciated? How these processes are controlled at the molecular level. And there are also some kind of details related to what mathematically does it mean that there must be enough space for the Hofmeister's model to work. Hofmeister said that there must be enough space for new primordium to be placed in the meristem, but actually how this availability is measured. So we are looking forward to address it, and I would like to thank my collaborators and my own group in particular. So here is Paula and myself, of course, and here is Teng Zhang, who performed pretty much all this experimental obtained all this experimental data in the scope of his PhD thesis, which we defended not long time ago. And here are two of my students, Andrew Owens and Mick Cieślak, who did a lot of modeling this. And other students are part of collaboration as well, but they were not so much involved in the work which I described today. And here is more thanks to organizations which supported us and paid for all this fun. And thank you very much.
Thank you for that wonderful presentation, Dr. P. While people start asking questions, I'll start off really quickly with a question. A long time ago, we approached you for your cell and auxin-based models to ask questions about leaf asymmetry, how the source and sink relationships affected the asymmetry of leaves through auxin. And I was wondering, because for your new very L systems based model of how this ring propagates, it's really fundamentally on that initial asymmetry of how it starts, the long and short, right? And also the asymmetry plays a role there. Do you think if, as you go forward exploring the molecular mechanisms or applying your cell and auxin-based models, that these sink source relationships might be very fundamental to setting up this pattern?
Yes, so it is excellent question, and yes, I think that it is going to be very essential, very much essential. Looking at the distribution of pins in heads is critical to answer this question, to see what exactly happens. And but we are working on having Gerbera heads or Gerbera plants with pin reporter, but it is a long process. So we are still working on this. But it is very much expected that that this asymmetry can be related to the inherent directionality of auxin transport via pin exporters. Incidentally, it is also interesting if we look into the fact that this primordia appear in sort of asymmetric context. There is short space and long space there, whether this may have some impact on the asymmetry of organs, petals or leaves. And this would be extension of the work which has been done several years ago by I believe it was Chris Kuhlmeyer's group, where it was observed that leaves are not exactly symmetric, and this was related to the chirality of the patterns. So yes, this is very interesting, and we started looking into this, but unfortunately in Gerbera, bracts are very symmetric. It is difficult so we need something which would have more asymmetric bracts to find out whether this asymmetry can be related to this pattern of long and short intervals.
Thank you. Dr. Dabrowski.
Yes. Hello, Przemysław. Thank you very much. That's really amazing work you showed us. And your proof of that the patterning starts from the periphery to the center is rather convincing. Still, we can think there is in center, there is some invisible signals that instruct periphery in certain way. So did you try to damage central part and see if the patterning is normal?
So I haven't, but Paula Eloma and Teng Zhang have. And the pattern, and I believe that is also related to the earlier experiments by, I think, Palmer and Hernandez. And it turns out that if the center of the head is damaged, for example, if a ring is cut in the head, then patterning up to this damage is normal, but after this is not. Okay. So, so this indicates that that the pattern really is progressing from outside in. Regarding signals, however, it is interesting because, well, we believe that the signals going from the center of the head, especially the presence of auxin, which seems to be there, but it's not clear what it's doing, may be important for the development of vasculature in the heads. But this is just well, something which we're still working on.
Okay, okay. Thank you very much. That's very interesting.
Thank you.
Thank you again for a wonderful talk, Dr. Przlinkiewicz. Let's move on to our last session for the day, the flash talks from students. Thank you, Dr. P.
Hi, my name is Angelica Jara, and I'm going to present my project about the buffalo grass microbiome. Buffalo grass is the plant that you can see at the bottom, and it's a plant that has been introduced to deserts around the world and has wiped out endemic plant species. To do so, buffalo grass uses a mechanism known as allelopathy, which consists on the secretion of allelochemical compounds that arrive to a recipient plant and affect its development. Allelochemicals from the root exudates or from the aerial part of buffalo grass also seem to impair the growth of the same buffalo grass. However, we don't know the effect that these compounds have on the rhizospheric microbiome, and we also don't know which are the microorganisms that attach themselves to the roots of buffalo grass. So we used soil from the Sonoran Desert, and we grew buffalo grass plants in PVC tubes, and we treated them with root exudates or with leachates from the aerial part of different buffalo grass individuals. We took samples after 90 and 110 days of growth, and we evaluated the microbiome composition. All our samples had a taxonomic profile dominated by actinobacteria, proteobacteria, and acidobacteria, which agrees with previous reports of other desertic shrubs. We did not find strong differences among the allelochemical treatments and the control. When we ran a CAP analysis, we saw that time is the main factor shaping the microbiome, since all our samples from the first period are located at the bottom, and the samples from the second period are located at the upper left section of the graph. And this is probably related to the development of buffalo grass, which entails changes in the exudates pattern, and therefore the bacteria that are recruited are different as the buffalo grass grows. This has also been reported for other plant species like wheat or watermelon. And accordingly with this, we found 4 taxa enriched in the first period and 4 different taxa enriched in the second period. And finally, even though the allelochemicals did not significantly influence the microbiome composition, we detected 12 taxa that are enriched in the different treatments. Some of those taxa, like Saccharothrix, are capable of metabolizing phenolic compounds, an allelochemical that is produced by buffalo grass. Others, such as Caulobacter, are capable of promoting growth in plants. So far, our conclusions are the composition of the rhizospheric microbiome of buffalo grass under different conditions, the fact that it's time and not the allelochemicals, the factor that shapes the microbiome composition, and, well, the microorganisms that buffalo grass recruits are capable of thriving under allelochemical conditions. Thank you so much for listening.
Hi, everyone. Thank you very much to the organizers for the opportunity to present my work today. I'm Neftali. I'm a PhD student in Nick Talbot and Frank Menke Lab at the Sainsbury Laboratory in Norwich. Today, I'm going to talk to you about the fungal pathogen Magnaporte rice. This pathogen is a serious problem in every single country where rice is grown and causing losses of up to 30% of the global rice production. Here, I show you a picture of how rice plants looks like during infection. After a few days, this pathogen kills the plants completely. Magnaporte rice infection starts in the surface of the rice leaf. To penetrate the leaf, Magnaporte spores germinate to form this dome-shaped structure called an apresorium. In our lab, we have the null mutant of the MAP kinase PMK1. After germination, this mutant never differentiates into an apresorium, and therefore, it doesn't generate infection. It is well known that PMK1 is involved in apresorium formation, but how this protein regulates these processes remains unclear. Because PMK1 is a MAP kinase, we decided to study the Magnaporte phosphoproteome during apresorium formation. Basically, we decided to compare the phosphoproteins present in germinated spores of the wild-type strain XII and the null mutant of PMK1. Using a combination of discovery and targeted proteomics, we identified 30 proteins that could be involved in the PMK1 signaling pathway. These 30 proteins were differentially phosphorylated. One of these putative targets is the SAN domain containing protein BTS1. We identified that BTS1 is phosphorylated at 3 different residues, and by yeast-to hybrid and co-immunoprecipitation experiments, we found that it associates to the MAP kinase PMK1. Because BTS1 hasn't been characterized in Magnaporte rice, we generated a null mutant to study its role during infection. When we compare apresorium formation between wild-type spores and BTS1 null mutants, we found that some apresoria have aberrant structures like these ones I'm showing you here. And when we used BTS1 null mutant spores to infect rice, we found that around 85% of these null mutant spores are less pathogenic compared to the wild-type strain XI. So taken together, we have identified a novel component of the PMK1 signaling pathway using a phosphoproteomic approach. At the moment, we are defining the role of BTS1 in this cascade. Thank you very much.
Good afternoon. I'm Joel Colchado, master's student at the Botanical Garden of the UNAM. I'm presenting preliminary results of a meta-analysis studying the variation of rhizosphere prokaryotic communities in halophytes and xerophytes from arid environments. Rhizosphere is the soil region influenced by the secretion profile of root metabolic activity. This favors and controls the establishment of micro-plant interactions, which are important for resource acquisition, thus affecting plant survival under stressful conditions. Due to this impact on plant survival, root microbiomes of arid plants have been recently studied, yet no work so far describes their assemblage of variations at a microgeographical scale. In this ongoing work, I described these patterns using previously published data from 4 countries, representing 10 host species from 4 families, with diverse ecological strategies and life history traits. A question to answer is, how is variation across microbiomes related to plant host traits and its environment in arid communities? For this, I extracted the sequences from SRA and elaborated a pipeline in QIIME2, using Silva database for taxonomic annotation and R for data visualization. Here I present some preliminary results. Microbial communities are taxonomically diverse at the class level. Here the 21 main classes are represented. Many of these were previously reported on the source work as globally abundant, such as Protobacteria or Bacilli, while others are site-specific, like the Pakistan-associated Archaean class, Halobacteria, or the interesting POC43F haline bacteria, before unknown to inhabit rhizospheres. Based on the calculation of 3 alpha diversity indexes, ACE for species richness, face phylogenetic diversity and Shannon evenness, we observe host-specific differences, panel A, and country-based patterns, panel B. But, according to Crystal Wallis' test, these differences don't seem to be related to the host ecological nor life history strategy, as shown in these box plots. Finally, beta diversity was calculated using Jacquard index and plotted with NMDS. The axis 1 seems to be related to pH, while the axis 2 correlates with temperature and precipitation. This pattern separates xerophytic from halophytic communities, as well as distinct halophytic communities. Overall, these results show that the abiotic environment and host identity are the main key drivers for prokaryotic communities composition. It remains to be tested if there is a correlation between phylogenetic distances of hosts and their communities. That's all for now. Thanks for watching, and I will be extremely thankful for any feedback to improve this work.
Good evening. Today I will present the talk titled Chemical and Physiological Perturbation as a Dissecting Tool of Arabidopsis Circadian Clock. The plant circadian clock, which is comprised of transcriptional and translational feedback loops, receives environmental stimuli as light and temperature, and as an output, the clock controls the plant physiology to a plethora of mechanisms. A circadian rhythm persists under constant conditions, either this light or continuous darkness. In this study, we use promoter luciferase reporters to evaluate gene expression under these conditions. Using a chemical biology approach, we found that hormones, vitamins, antibiotics, and other chemicals that induce oxidative stress affect clock period either in darkness, red light, or blue light conditions. From this screen, we focus on salicylic acid, and we found that the expression of 5B was affected and that the period of it was reduced. Furthermore, the effect of salicylic acid on period shortening was restricted to the first hours of the morning. We also found that the effect of salicylic acid on period length was inhibited on the presence of sucrose with a concentration of 3%. Therefore, we assessed the effect of sucrose on period length. We found that the effect of period was dependent on the genotype. As we can see here in the wild-type background Basileska, the period is reduced upon the addition of sucrose. However, in the mutant Gigantea, the period is increased. As an example, TOC1, a short period mutant, has a short period upon addition of sucrose, but not without sucrose. And in a media without sucrose, salicylic acid reduces its period under red light conditions, but not blue light conditions. We can conclude that the compounds that alter the redox status affect the circadian clock periodicity in different ways. Also, salicylic acid reduces the period length in the presence of sucrose in the media. As a perspective, we hypothesize that salicylic acid may act as a sign number to the circadian clock through the action of phytochrome B.
My name is Adolfo Aguilar Cruz. I am a PhD student, and I'm studying the function of the dicer-like1 gene in the liver wort Mercantia polymorpha. Mercantia polymorpha can reproduce asexually by the formation of gemmae. Gemmae are vegetarian propagules that can form a complete thallus, and gemmae are formed inside the gemma cups. The gemma development initiates when a cell from the floor of the gemma cup elongates and differentiates into a gemma initial cell. The gemma initial cell undergoes two asymmetrical divisions. The first one gives rise to the basal cell and the start cell, and the second one gives rise to the gemma primordium cell. The gemma primordium cell continues dividing to form a complete gemma, while the basal cell and the start cell do not divide anymore. And this is how a wild-type gemma looks like. It's a flat disc with two apical notches, but this developmental process doesn't happen like this in the dicer-like1 mutant. We use the CRISPR-Cas9 system to target the dicer-like1 gene in Mercantia polymorpha, and mutant plants produce gemmae that show differentiation of gemmae directly from the dorsal and ventral cells of the gemma surface. And this phenotype resembles the floor of the gemma cube because we can see different stages of gemma development on the surface of the mutant gemma. And here we can see the development of ectopic gemma directly from the surface of the mutant gemma. In land plants, dicer-like-1 controls microRNA's biogenesis. And in the dicer-like-1 mutant of Mercantia polymorpha, the biogenesis of microRNAs is abolished. We also performed RNA-seq, and we found upregulation of targets and precursors of microRNAs. We also found upregulation of genes involved in auxin biosynthesis and signaling, and an overrepresentation of auxin-responsive genes. We use a reporter line that marks the site for auxin biosynthesis in Mercantia polymorpha, and when we express this marker in the dicer-like-1 mutant, we found new sites for auxin biosynthesis, and these new sites are the ectopic gemma. We also perform auxin immunolocalization. In wild-type gemma, the auxin maxima territories are the rhizos precursor cell and the apicarnoches, and in the dicer-like-1 mutant, new sites for auxin maxima territories are formed, and these new sites are the ectopic gemma. And what we propose is that the epidermis of the gemma in the dicer-like-1 mutant acquires the cell identity of the floor of the gemma cube. And in conclusion, dicer-like-1 is a major contributor for the biogenesis of microRNAs in Mercantia polymorpha. And dicer-like-1 regulates cell fate specification through a mechanism driven or influenced by auxin during gemma formation. Well, this is it, and thanks for watching.
My name is Aranza Angulo, and the title of my work is Expression Analysis of Sugar Transporter Genes Induced by Mycorrhiza Colonization in Tomato. Arbuscular mycorrhiza is an important symbiotic association between plant roots and arbuscular mycorrhizal fungi. In this symbiosis, the plants provide the fungus with the carbon compounds, while the mycorrhizal fungus helps the plants to improve its nutritional status. Nevertheless, there is not much information about the sugar transporters involved in the carbohydrate movement from the leaves to the roots colonized by this fungus. So, we studied the expression of some sugar transporter genes, such as STP1, 2, 16, and SFP7, described as inducing leaves of mycorrhiza colonized tomato plants, and the genes encoding for sucrose transporter SUT1, 2, and 4 were also analyzed. The relative expression of all these transporters were determined in leaves at 3 different developmental stages in mycorrhiza colonized and non-colonized tomato plants. Regarding monosaccharide transporters, STP2 showed induction of expression in leaves of non-colonized plants compared to colonized, whereas for sucrose transporter, only SUT1 showed differential expression. This was observed in mature leaves. We think that since the monosaccharides in the apoplast, the sugar may be then used to form sucrose in the apoplast to be translocated to the root with the possible participation of SUT1. On the other hand, the sucrose transporter gene SUT4, which encodes for a protein that has been located in vacuolar membranes, was repressed in young foliar tissues of mycorrhizal plants. So we propose that since SUT4 expression is lower in young leaves of colonized plants, the sucrose is transported from the cytoplasm toward the vacuole of the leaf cells, so more sucrose could be directed to colonized roots. The glucose content was also determined. Although no significant difference were observed in foliar tissues between colonized and non-colonized plants, a significantly lower content of the sugar was found in colonized roots. This supports the idea that glucose is the preferential transporter sugar from the plant to the mycorrhizal fungus.
Hello everyone, my name is Seungling Li. I'm a PhD student working in Professor and here's Hilde Bugun's lab at the University of Freiburg in Germany. The title of my presentation is the novel phytochrome interacting protein YA58 is a repressor of light-induced cis-germination in Arabidopsis. From East Traversicum library, we identified YA58 as a new interactor of phytochrome A. Later, we verified the interaction between YA58 and phytochrome B. ER58 belongs to the AP2EF transcription factor family, which has several homologs, including ER15-5. With a combination of physiological, genetic, and molecular assay analysis, we proved that ER58 is a negative compound in Phi A and Phi B mediated cis germination. In the cis germination experiment, we observed that ER15-5, 58 double mutant showed a higher germination rate than the wild-type Columbia in phytochrome off condition. Well, the opposite was true for ER58 O impression lines, which exhibited a lower germination rate than the wild-type Columbia in phytochrome active state. PIF1 and SAM are two important proteins involved in Phi A and Phi B mediated cis germination. We found the expression levels of PIF1 and SAM are upregulated by ER58. Both in vivo and vitro assay evidence that ER58 associated with the promoter of PIF1 and SAM and enhances the expression. Furthermore, we found that the binding capacity of ER58 to PIF1 and SAM promoters were disturbed by light activated phytochromes. So we observed that more activated phytochromes less ER58 binding to PIF1 promoter and SAM promoter. The phytohormonal ABA is an important inhibitor of cis germination. The inhibitory effect of ABA on cis germination is reduced in ER15-5, 58 double mutants, while it is increased in ER58 O expression lines compared to the wild type. At the molecular level, we found that ER58 inhibits cis germination by directly regulating ABA anabolic genes, increasing ABA level. In parallel, as reinforcing feedback mechanisms, ABA upregulated ER58 transcription level and protein stability. Moreover, ER58 represses cis germination partially dependent on ABI5 and promotes JA catabolic genes involved in JA regulated cis germination process. With that, I want to thank you very much for your attention.
Good morning, my name is Karina Cortez Fonseca, and I'm going to present the work entitled Antifungal Activity of Mexican Mesonthus Leaf and Flower Extracts Against Fusarium. As the co-authors, we have Carlos Luna, Veronica Moreno, Gustavo Hernandez, and Luis Choca as corresponding author. Mesotol is a parasitic species that frequently grows at expenses of trees, causing serious forest damage in Mexico, as well as being considered the second agent of forest destruction. The advantage of mesotol is that it absorbs a large amount of nutrients of the host, which makes it a candidate to evaluate its antifungal capacity in vitro, where it extracts against a phytopathogenic fungus of our interest, which is Fusarium. It has antioxidant, antihypertensive, and antimicrobial properties. Fusarium, it has a cosmopolitan distribution and is endemic in corn production areas throughout the world, causing great losses of crops that are of nutritional importance for humans and animals. In our methodology, we use a mesotol that was obtained from the product and natural area of the host Palo Dulce, while the fungus was insulated from the wind-growing area of the region. The samples were separated into leaves and flowers, and the processed to dehydrate them for subsequent grading. All samples were subject to soil liquor extraction using methanol acetone water. After this, the antifungal evolution was made of 3 different concentrations, which were mixed with PDA medium and inoculated with the phytopathogenic fungus Fusarium. Each treatment was done in tropicate, and all treatments and controls were incubated at 28 centigrades in the dark until the control present 100% mesothelial growth. The percent of mesothelial growth and the final correlation were calculated, as well as preliminary tests to determine the combination with the extracts. As results, we obtained that for both mesotol extracts, the highest inhibition was present in treatments 1 and 2, with the highest inhibition for the leaf extract being 91%, while the flower extract was 96%. Regarding a final solution, it was obtained that treatments 1 and 2 of flower and treatment 2 of leaf present a reduction of 2 logarithms, while treatment 1 of leaf was the one that had the greatest reduction, presenting 3 logarithms less, which present 99.7% reduction in solution. In addition, preliminary tests have been conducted where the extracts has been found to inhibit carmination. However, such tests are still being deployed. It was found that both a flower extract and the leaf extract present antifungal activity to inhibit the development in vitro in Fusarium. There was a great inhibition of mycelial growth using the flower extract, while the great reduction of spores was using the leaf extract. More research should be done to determine the possible mechanisms of action to inhibit the different phytopathogenics using the different mycelial extracts. Thank you.
Good day to the audience. My name is Fernanda Rosiles. The project I'm working on is protein characterization of MBF1 family in Solanum lycopersicum. It is supported by Instituto Potosino de Investigación Científica y Tecnológica and Laboratorio Nacional de Genómica para la Biodiversidad. First, tomato, or Solanum lycopersicum, represents one of the most economically important crops worldwide due to the higher content of important metabolites. Nevertheless, the tomato is also considered a model plant for the study of response to stress, especially those linked to transcriptional activation. What we know of tomato MBF1 family is that multiprotein breaching factor 1, or MBF1, is a family of proteins described as transcription cofactors. It is characterized for breaching transcription factors and the basal transcription machinery. Until now, 5 members of this family have been identified in tomato: A, B, C, D, and E. The MBF1 family is ubiquitously expressed in tomato plants. They are directly related to stress response in plants, but its function is not well characterized yet. Specific detection is key for protein characterization. For this, we did gene cloning on PCR8 vector and protein projection using PTAT17 vector. And then, we did protein affinity purification. And to gain more information about the MBF1 family, we produce and purify specific antibodies against these proteins. Finally, we did Western blot analysis for the antibody validation. For demonstrate that the specific antibody detects our protein, we did Western blot analysis. Here, we can prove MBF1E purified antibodies have the ability to bind both non-denaturing and denaturing structure of the protein on the expected weight. And then, the work to do. Once we validate the antibody, this will serve for carry out immunolocalization experiments in different tissues, and also co-immunoprecipitation experiments that will help finding other protein partners for the MBF1 proteins. Finally, all the recovered information will help us to complete the characterization of these proteins in tomato. Thanks to all.
Hi everyone, my name is Andrea Romero Reyes and I will be talking to you about how the increased trehalose synthesis in wheat plants can benefit photosynthesis and growth under drought stress. Trehalose is a non-reducing sugar and its accumulation provides desiccation tolerance in organisms like Telaginella lepidophila, which is a resurrection plant, as well as in Saccharomyces cerevisiae. Trehalose is synthesized via the TPS-TPP pathway by 2 enzymes, which are trehalose phosphate synthase and trehalose phosphate phosphatase. Many studies have shown that overexpressing these enzymes can enhance tolerance to drought and other abiotic stresses in crops like rice, tobacco, and tomato. So we worked with transgenic wheat plants overexpressing a bifunctional TPS-TPP enzyme. We subjected these plants to a drought stress for 5 days, and we analyzed the photosynthetic performance of the plants, and then we rewired the plants to analyze their biomass production. The drought treatment caused a reduction in the leaf relative water content, both in the wild-type plants as well as in the transgenic lines. When we analyzed the photosynthetic parameters, we found that the transgenics were less affected by the drought stress compared to the wild types, and they had a higher CO2 assimilation as well as a better efficiency of photosystem 2 under the drought stress. Here you can see a photo of the plants 5 days after we rewatered them. You can notice how the transgenic lines had a greater biomass compared to the wild type plants. And this was maintained until the final harvest stage, when even if we saw a reduction in the aerial biomass caused by drought in the transgenics, they were less affected by the drought stress compared to the wild type plants. So these results suggest that trialose synthesis can support a better photosynthetic performance and protect plant growth during and after drought stress. And here I leave you my contact information in case that you have any question or comment. And thank you so much for your attention.
Hi, today I'm talking about the project OPS-DHD2 and acidic dehydrogen ASK3 isolated from Opuntia streptacantha. The genus Opuntia represents an important economic source. The common named cactus pear is used as forage, fruits, and green food, and it's adaptable to growth under arid and semi-arid environments. On the other hand, the hydrins belongs to group 2 of Lea protein family and highly hydrophilic and intrinsically structured proteins. Its expression induction was observed under drought, low temperatures, salinity, and ABA. The aim of this work is the characterization of the dehydrogen 2 gene of Opuntia streptacantha. Previously, in our research group, the dehydrogen 1 gene was isolated and characterized. The dehydrogen 1 gene showed an induction in response to abiotic stress, and its overexpression in Arabidopsis improved the tolerance to cold stress. Then the sequence of the dehydrogen 2 gene from Opuntia streptacantha was obtained, and it was compared with amino acid sequence of the dehydrogen 1 and the 10 dehydrogen of Arabidopsis and grouped with the ASK3 class. A clustral alignment confirming the presence of the characteristic motifs in the dehydrogen 2, the S-K segment, and the nuclear localization signal. We carry out an expression analysis in 3-month-old plantals at 5 and 10 days of stress in cladode and in roots. We applied treatments of saline stress, drought, heat, and cold. And we observed an increase of expression of dehydrogen 2 in cladodes treated with salt at 5 and 10 days. Similarly, in the drought and cold conditions. Finally, the transiently transformed Nicotiana ventaniana leaves were analyzed under confocal microscope. As observed, the fluorescent signal is located in the cytosol and the nuclei. We performed the overexpression in Arabidopsis and the BIFC assay for the dehydrogen 2. Thank you for the invitation and for your time.
Hi everyone, I'm Adrián Pulido from Dr. Dinkova's lab at UNAM. Today I'm going to talk about how plants regulate gene expression under extreme temperatures, specifically the eukaryotic initiation factors EIF, 4E, family-mediated responses. Translation initiation in plants take place in 2 main processes: formation of ternary complex and mRNase activation. 4E family proteins are important because they interact with 5' end of mature mRNAs, recruited the messenger RNAs, and allowing the translation pre-initiation complex assemble to start the protein synthesis. Arabidopsis thaliana has three main isoforms of 4E family proteins: the canonical isoforms 4E and iso4E, and no canonical isoform 4EHP. By measuring the phenotypic damage, we found that the absence of either canonical isoforms 4E or iso4E results in susceptibility phenotype of 5-week-old Arabidopsis thaliana plants under freezing stress, and altered selective stress-related mRNA translation. 4EHP null mutant was affected in freezing tolerance too, and this happened even in acclimation response to low temperatures, as we can see when we previously treat the plants with acclimation period followed by cold stress. For this reason, we decided to explore transcription modification in 4EHP null mutant, which has altered transcriptional induction of important cold responsive genes as COR15 or TCF. Finally, under high temperatures, the absence of 4E or iso4E did not affect early seedling development, but surprisingly, the 4EHP null mutant seems to exhibit an improved response to heat stress when we compare them with wild-type responses. 4EHP null mutant also present altered accumulation of heat stress marker genes as these two chaperones. 4EHP orthos have been reported as no canonical since they are not directly involved in translation, but rather participate in other aspects of RNA metabolism. Our current aim is to understand how regulatory networks mediated by no canonical 4EHP isoforms work under stress to achieve a tolerance response to extreme temperatures in Arabidopsis. Thank you for your attention.
Hello, I'm Edgar Pascual. I come from the Biotechnology Institute in the UNAM in Mexico. I want to talk to you about the role of lorelei protein in the nodulation process in the common bean. Lorelei and lorelei-like proteins have an anti-catalytic function. However, they have in the amino terminal a signal peptide that mobilizes them to the endoplasmic reticulum, and in the carboxyl have an hydrophilic amino acid tail that is replaced for a glycophosphatidylinosytol anchor. In the reticulum, interact with the receptor-like kinase, and together through the secretory pathway migrate to the outside of the plasma membrane and form part of the lipid rafts. Some phenotypes associated with LLG mutants impair cellular interaction process. For example, lorelei mutant ovules are unable to recognize pollen tube that reach them, having low fertility rate. On the other hand, in the phytopathogenic interaction, LLG participate in the recognition of glutamous through its interaction with the flagellin receptor. LLG is also known to have rod head growth defect and low rod production. So, if lorelei regulates cell interaction process, could it participate in the mutualistic interactions? In beans, there are two genes that code for LLG proteins. One of them is expressed in the floral tissues and the other in vegetative tissues, particularly in the apex and in the rod hairs. This expression then correlated with the tissues where the promoter activity is present. Then we want to know what happened with LLG genes under nodulation condition. We observed that the inoculated rod present higher levels of transcripts of LLG, same to the mother nodules. Once again, this correlated with the promoter activity in the same tissues and in the infected rod hairs. To explore the role of lorelei in the nodulation process, we generate genetic construction to silence and overexpress lorelei. RNA roots have a rod height growth defect and also develop smaller nodules, while the rod overexpressing LLG form nodules even in the late stage of development. To test if the nodule development defects are related to the colonization process, we inoculate silenced and overexpressed root with a rhizobium tropiside strain that expressed Bull's reporter, we observed that in the silent nodules are colonized in a non-uniform way. In contrast, nodule overexpressed have a normal development. In conclusion, allergy play an important role in the development of nodules. A question to solve is determine if LLG interact with receptors that regulate the symbiotic process, and could LLG regulate other mutualistic interactions such as microregulation? And that is all. Thank you.
The title of my project is Volatile Organic Compounds Produced by Maize Plants on the Drought. Today, our agricultural model depends strongly on weather conditions, so climate change represents a serious problem to achieve high yield productions. One of the most important abiotic stress for the maize plants is drought, which causes decrease in water potential to grow chlorophyll, as well as in the availability and transport of nutrients. Furthermore, drought induces the accumulation for free radicals and ethylene biosynthesis. The bugs are organic compounds that at normal temperature and pressure have a high vapor pressure. These compounds play an important role in communication, stress protection, growth, and development in plants. In this work, we focus on knowing the change in the box profiles emitted by maize plants without and under stress due to water deficit. 3 stages of drought stress of 7, 9, and 11 days were induced in maize plants. The detection and identification of the bugs was carried out using the solid phase microextraction coupled to the gas chromatography mass spectrometry technique. We were able to identify 16 compounds from the sesquiterpene, aromatic, alkane, ester, alcohol, and fatty acid families. From the compounds identified, 3 of them show statistically significant differences in the percentage of normalized quantity. For example, emissions of the acetic acid 2-ethylhexyl ester and 2-ethyl-1-hexanol increased to a more severe stress, while hexadecane emission decreased. We observe that maize plants under drought stress have change in the box emission profile. This change can be seen in specific compounds or the majority of compounds of the same family. For example, the alkanes family stop their emissions in plants under 11 days of stress, while the aromatic family increase its emission in plants under 9 and 11 days of stress. Thank you.
Hello, my name is Amir Ortiz. I'm a graduate student at Tecnológico Nacional de México in Celaya, and I'm going to talk about the project titled Analysis of Amaranthus hypochondriacus seed development DNA methylation patterns by whole genome bisulfite sequencing. There is a great interest in amaranth due to its nutritional benefits, agricultural advantages, and the potential it possesses to produce a wide variety of food products. Molecular information regarding amaranth embryogenesis is very limited, and genomic data suggests that one of the molecular mechanisms that could be modulating this process is DNA methylation. The differences in the ovule and seed methylation profiles during amaranth embryogenesis could give indication of the molecular dynamics involved in this process. DNA methylation is produced by adding a methyl group at the cytosine carbon 5 mediated by a DNA methyltransferase enzyme. In plants, DNA methylation can occur in the CG, CHG, and CHH context. Detection of methylation is essential to determine the effects of these regulatory mechanisms, and whole genome bisulfite sequencing is a technique used for the analysis of genome-wide DNA methylation patterns at a single base resolution. The experimental model was amaranth plants grown under greenhouse conditions. The experimental groups were divided into 2 stages. Germinal tissue, early and late embryo were recollected from a total of 18 plants. Then, the amaranth plants were cultivated and recollected. The DNA extraction and whole genome bisulfite sequencing was performed. Subsequently, the alignment to the reference genome and analysis of the methylation levels were carried out, and finally, the level of methylation around all transcription start sites was analyzed. Regarding the results obtained, global DNA methylation levels for each tissue were very similar, but the differences in them could be found in the genomic regions where the methylation took place in addition to the context where it could have happened. Per context, results showed that in all three stages, cytosines in the CG and CG context were methylated, in contrast to CHH context where a smaller fraction of cytosines were methylated. Further statistical analysis confirmed that DNA methylation levels per context were independent of the type of tissue, as chi-square value was not significant, p was greater than 0.05. Around the transcription start sites in the CG context, a decrease in the methylation levels as the samples developed was observed. In CHH context, the ovule had the lowest methylation levels, while in the early and late embryo, a progressive gain in methylation during the successive stages of seed development was observed. In conclusion, the decrease in methylation levels near the transcription start sites in the CG context could indicate the transcription of some genes important for seed development in the region. Also, the increased level of methylation in the CHH context suggests that there could be a differential regulation of methylation during the transition of the seed development stages. Thank you very much.
Hi, my name is Eduardo Luján. I'm from the INCOVAS lab, and I'm going to show you some results about microRNA 528 and its regulation in a maize Mexican variety called Tuxpeño. MIR528 is a monocote-specific microRNA, and we characterized the accumulation profile of this microRNA in two particular models. During germination, we found that MIR528 decreases its accumulation as seedling establishment occurs. By the other hand, during maize somatic embryogenesis, we found that this particular microRNA is highly accumulated during the proliferative stage of embryogenic algae. After this, we experimentally validate targets that only have been predicted by computational methods. With this, we obtained the accumulation levels of these targets. Many of them exhibit a clear inverse correlationship with miRNA levels in total RNA in both the germination process as well as during embryogenic callus induction, which might suggest that miRNA-mediated cleavage is exerted over these targets. In addition, when performing sucrose gradient separation, we found that microRNA 528 is highly accumulated in heavy polysomal fractions. Here, we found that PLC1, a target that did not show an inverse correspondence in total RNA, displayed a negative correspondence with the distribution of MIR528, suggesting that this microRNA is also capable of regulating some targets by translational repression. So, after all of this, we decided to explore the upstream regulators of microRNA 528 using our germination model. So here we found that one of the precursors is present during the first 12 hours of imbibition. Moreover, we found that treatments with nitrate or exogenous auxins increased the accumulation of both pre and mature form of MIR528. In addition, we were able to corroborate that such increases resulted from Pol-2-dependent transcriptional event, as exposure to alpha-amanitin reverted the positive effect of both treatments on the miRNA gene transcription. And finally, we found that the promoter region of MIR528 harbors binding sites for transcription factors involved in the nitrate response and the auxin signaling pathway. So, this is our tentative model, which links the presence of both high nitrate concentrations and the auxin 2,4-D that increase MIR528 transcriptions, which results in the regulation of several mRNA targets.
Hi everybody, I am the Dr. Ernesto Vázquez Chimalgua from Universidad Michoacana de San Nicolás de Hidalgo. I'm going to present you the work titled The Volatile Organic Compound Dimethylhexadecylamine Represses Arabidopsis thaliana Primary Root Growth through alteration of cellular differentiation and the regulation of cell site in meristematic cells. Dimethylhexadecylamine is a volatile emitted by some rhizobacteria. In Arabidopsis thaliana, 16 micromolar concentration reduces root growth with respect to control condition. This inhibition is correlated with a suppression of expression of meristematic genes like shortroot and PRITORA1. The meristematic gene scarecrow is expressed specifically in endodermis tissue. Dimethylhexadecylamine downregulates its expression and duplicates endodermal cell layers, as is indicated by the arrowheads in the inset. Indeed, in this black and white contrast image, is visible that dimethylhexadecylamine modifies cell differentiation and regulates cell site control in all tissue layers of the entire root meristem. These effects are correlated with an increment of root meristem width. Then we prompted to investigate auxin because auxin acts as a morphogene. DR5-GFP synthetic marker showed an expansion of its expression domain, indicating an auxin accumulation in root tip after the dimethylhexadecylamine treatment. This overlaps with the expansion of WOX5-GFP expression domain, suggesting that dimethylhexadecylamine produces an agglomeration of auxin in root tip, and this is the grounds for alteration of cellular site and differentiation. Thank you.
Hi, good morning. My name is Bricia Ruiz Aguilar, and I'm going to present you our work entitled Arabidopsis Transcriptional Response Due to Exposure to Cold and Freezing Stress. The plants are exposed to two main types of stresses, the biotic and the abiotic, and within the layer we found the one generated by low temperatures, which in turn are divided in cold and freezing stress. The plants are exposed to this, there is a transcriptional regulation of low temperature response genes, which leads to stress tolerance. This might involve the synthesis of response molecules, such as compatible solutes, proteins with cryoprotective effect, and detoxification. But we wanted to know what more is happening inside the plant. To achieve that, we conducted an RNA-seq study in plants exposed to stress by low temperatures. We used Arabidopsis thaliana as a model plant, and seedlings of 14 days were exposed for 24 hours to 0, 4, and 10 degrees. And we are working now on the data analysis. We focused on the biological process with more enrichment in the treat temperatures. We found in the induction processes like RNA secondary structure unwinding, rRNA processing, and cold acclimatation. In these processes, we found proteins like helicasas, elongation factors, RNA binding proteins, dehydrins, and key transcriptional factors in the response to cold and freezing stress in Arabidopsis. On the other hand, in the repression, we found biological process related with photosynthesis and chlorophyll biosynthesis. We are also working on the characterization of genes of interest selected from this data, and in the identification and in silico analysis of the genes, analysis of expression, and in the characterization of mutant and overexpressing line. And that's all. Thank you so much.
Hello, my name is Mariana Sotelo. I'm here to present the work that we are doing in the Facultad de Agronomía, Universidad de la República, Uruguay, about root growth adaptation under water deficit. In our group, we are interested in genes that are associated with abiotic stress, and here I will briefly introduce the work that we are doing with the TTL mutant. TTL1 mutant was first isolated in Arabidopsis by Abel Rosado in 2006 in a screening population of sodium chloride. It was isolated with for the phenotype of root growth reduced length. TTL1 belongs to a small family of proteins with four members that are characterized for having 6 tetratricopeptide repeats that are located in conserved position among the members of the family. Also, in the C-terminal, they have a thioredoxin-like motif that is not functional because of the absence of a cysteine that is required for the reductase activity. During osmotic stress, also TTL1 presents a reduction in a root growth rate and a swelling phenotype in the epidermal cells of the elongation zones of the root. This phenotype was the starting point of our research because we wanted to know if these changes in the anisotropy of these cells were related to changes in the physical properties of the cell wall, and moreover, we wanted to know if these changes in the anisotropy of the cells were related with the reduction in the primary root growth rate. For this, we evaluated the physical properties of the cell wall in epidermal cells of the elongation zones in the mutant during osmotic stress. Moreover, we characterized the primary root growth during osmotic stress, characterizing parameters like cell production rate, length of the cell cycle, cell elongation in the proximal meristem, and time to enter the transition and elongation zone. At the end of the work, we were able to relate TTL1 with 2 of the hormones that control the balanced growth in the meristem of the root of Arabidopsis, and also we relate TTL1 with the cell anisotropic expansion that is needed to control cell division and root growth recovery during osmotic stress. I would like to invite you to read our work that was recently published in Genes. Thank you for listening.
Hi, my name is Eric, and today I'm going to talk about how to measure the shape of barley using topology, a project here at Michigan State University that mixes math, image processing, and a lot of barley. Barley is extremely adaptable. You have varieties that grow in Iraq, in Norway, Morocco, Tibet, and Scotland. And just looking at the shape of the seeds, you can tell that there must be an interesting relationship between morphology and climate adaptation. In particular, we are interested in studying the morphological properties of 20 different accessions corresponding to 28 very different climates across the Eurasian continent. So with our collaborators in UC Riverside, we got shipped large boxes of barley all the way from California here to Michigan State, where we have a 5-ton lead box that allowed us, with a very creative setup, to get almost 1,000 3D reconstruction of barley spikes corresponding to 28 different accessions. And not just that, doing a lot of image processing, we are able to also get individual seeds. And now with individual seeds, we can measure their shapes. So how do we measure the shapes? We can do it traditionally, say the height, the length, the width, surface area, volume of each of these seeds. But there's more than meets the eye. We need to be more comprehensive. So we do the Euler characteristic transform, or the ECT, which essentially measures the topology, the inner mathematics of each seed, by taking each seed and fixing a direction, say left to right, front and back, top to bottom, and chopping the seed across that direction and measuring how the topology, how the inner mathematics of this seed changes. And by measuring those changes in topology, as a collection of numbers, mathematically we can prove that we summarize effectively all there is to know about morphology. Does this work? Well, we can ask a computer. We tell the computer just morphological information of the seeds, and we expect the computer to tell us with some accuracy which accession the seed comes from. So if you tell the computer just traditional information, the computer will tell you 55% of the time the correct accession that the seed comes from based just on lengths and width and volumes. But if you tell the computer topological information, now the computer is able to tell you the correct accession 74% of the time. Even better, if you combine both traditional and topological information, now the computer is correct 86% of the time. So topology definitely is measuring something that we are missing in our traditional context. What are we missing? Well, we can do analysis of variance if there's a particular direction or a particular slice that tells us more information between accessions. And turns out that the most useful slices are those that correspond to the crease and the bottom of the seed. And with that, I thank my collaborators, organizers, and these are my details if you have any more questions.
Hello, everybody. I am Álvaro Colin, and I'm going to present some of my doctoral research, which is titled Acetogenin and Fatty Acid Dynamics During the Avocado Seed Germination and Leaf Maturing. So, acetogenins are fatty acid derivatives, and they can be found in the avocado fruit and also in the avocado leaves. They possess different biological activities, and this makes them attractive to some industries. However, their biosynthesis pathway is still unknown. In order to gain information about the tendencies that these lipids follow in the avocado plant, we sampled seedling and tree leaves, and also embryonic axis and cotyledon from imbibed seeds, and determined their acetogenin content and their fatty acid content. Some of the most interesting findings were that acetogenin seems to accumulate in specific periods of the seed germination, such as the time in which we can observe the root protrude from the seed. On the other hand, seedling leaves seem to accumulate more acetogenins as they develop. However, trees accumulate a lower amount of acetogenins. And also, tree leaves seem to accumulate almost exclusively persin, which is an acetogenin that is toxic to some herbivores and to some insects. It was also very interesting to find that acetogenins can be bound to different glycerolipids. Specifically, we found them bound to mono and diacene glycerols, and also phospholipids, as we can see in this graph. In addition, using all the data from this study, we built a correlation network in between fatty acids and acetogenins. We can see here that the polyunsaturated fatty acid cluster is the closest one to the acetogenins cluster. So this indicates a strong metabolic relationship in between them. Some of the conclusions that we can draw from this work is that acetogenins and fatty acid follow interlinked dynamics across all of the avocado plant life. For the first time, we determined that acetogenins can be bound to different glycerolipids, and also the correlation network that we built confirms a previous relationship in between linoleic acid and persin, but it also uncovers new relationships in between specific acetogenins and fatty acids. Lastly, the fact that we find acetogenins bound to phospholipids indicate that they may have different roles in the avocado plant, such as signaling. And well, that's all. I want to thank you for your time, and I hope you enjoyed this talk.
Hello, everyone. I'm Citlali Fonseca, and I'm a postdoc at UC Berkeley. And today I'm going to present to you some of the results I got in my previous postdoc at Instituto de Biotecnología UNAM in Mexico. And the title of my presentation is Metallothionein 1A regulates rhizobial infection and neuroorganogenesis in Phaseolus vulgaris. Metallothioneins are metal ion binding proteins that have important roles in plants. For example, they are involved in the regulation of cell growth and proliferation, protection against heavy metal stress, and responses to pathogen attack. In a previous transcriptomic analysis in your lab, we found a methylothione 1A was upregulated under nodulation conditions and downregulated under mycorrhization conditions. In addition, an in silico analysis and expression profile showed that this gene was most highly expressed in uninoculated and inoculated roots with rhizobia. So we hypothesized that this gene could have an important role in common bean and rhizobia symbiosis. We identified 6 methylothione genes in common bean genome, and they were classified into 3 classes based on their homology with those methyllothioneins in Arabidopsis thaliana. We analyzed the expression profile of MT1A at early and late stages of nodulation in common bean, and we observed that this gene was upregulated at 7 and 30 days post-inoculation, but it was downregulated at 21 days post-inoculation. Also, we observed promoter activity in cortical cells near to deformed root hairs and in nodal primordia, as well as an infection zone on young nodules and periphery of the infection zone of mature nodules. After reverse genomics analysis, the downregulation of MT1A showed a reduction in infection events at 7 and 10 days post-inoculation with 50 and 75%. In addition, the total number of nodules was reduced at 14 and 21 days post-inoculation, and those nodules presented a reduction of 50% of nitrogen fixation capacity. Finally, an histological analysis showed that the number of the infection cells and the size of the cells in the epidermical and parenchymal region was reduced in MT1A silent nodules. So we can conclude that MT1A gene is required for the proper development and progression of infection event during the early stages of nodulation in common bean, and also has an important role in the nodule organogenesis in this legume. Thank you so much for having me, and see you next time.
Hello everyone, my name is Laura, and today I'm going to share with you my research called Functional Analysis of Flowering Locust T during the reproductive transition in Agave tequilana. During their life cycles, plants undergo various morphological and physiological changes that have been classified as stages. One of the most studied is the reproductive transition, in which several pathways involving endogenous and environmental factors can influence flowering time. And downstream of these pathways, there's the key element flowering locust T, which is a mobile signal protein that induces flowering time through the fluorin activation complex. This transition has been widely studied in monocarpic annual species and polycarpic perennial species. Unfortunately, there are few reports for reproductive transition in monocarpic perennial species such as Agave tequilana. This is an emblematic crop here in Mexico due to the production of tequila and mezcal, and moreover, this crop has acquired international relevance for fiber production and as bioenergy source. But what do we know about this transition in Agave tequilana? Well, based on transcriptome analysis from leaf and shoot apical meristem tissues of Agave tequilana plants from different developmental stages, a series of elements involved in the reproductive transition were identified, such as flowering locust T. As we can see here, there are 6 homologs to promote flowering and 3 to repress flowering. So the manipulation of the reproductive transition represents an advantage for commercial production and basic research in agave species. Hence, the purpose of the present study is to characterize the function of the FT homologs in Agave tequilana. In order to do this, clones were constructed with the FT putatively designated as promoter or repressors, and their ectopic overexpression is being studied in Arabidopsis thaliana. The overexpression of FT is also being characterized in Agave tequilana by establishing a transformation protocol mediated by Agrobacterium tumefaciens. In addition, in situ hybridization and fusions of the genes to yellow fluorescent protein will be developed to determine the spatial and temporal expression. The results obtained will enable us to understand the transcription and regulation of the reproductive transition and to improve strategies for genetical analysis in agave species. Finally, I just want to say thanks to Professor June, Emmanuel Avila, and to all the members to the Laboratory of Genetic and Molecular Analysis of Agave Species. Thanks to CONACYT for support this research, and thank you for watching.
That was a great Flash Talk session. Thank you for all the Flash Talk presenters, and we'll have many more Flash Talk presentations throughout the week. You can go to our website and you can see the names of the presenters for each of the sessions and also in the abstract book. So thank you to all the Flash Talk presenters. Thank you to all the speakers today. And also thank you behind the scenes to the organizers and the technical people that are making this run, especially Edgar and Cedric. It was a great first day to the meeting, and we will have the meeting until Thursday. There is also a business meeting on Wednesday, which is from 5 o'clock to 7 o'clock p.m. Mexico City time. That's Wednesday, 5 to 7 p.m. Mexico City time. Please stop by to that meeting if you would like to contribute to the organization of the next SMB ASPB meeting. Hopefully by that time we'll be in person and you might get to select a nice place in Mexico to have the meeting, fingers crossed. But plan to attend that meeting if you want to be an organizer, or go to the website and the email is listed there for you to get in touch with the current organizers if you would like to participate in the organization of the next meeting. So please be aware of that. And if there's no other questions or concerns, we will close this first day of the meeting. Thank you everybody who participated.